thank our speaker. Is Ilya Sakharov from the Psychological Institute in Moscow, right?
Yes, yes.
So, perfect. So, and for the attendees, I just want to remind you, we have our Q&A section right below your screen. Please send your questions there. And remember, Neuromatch Academy, Neuromatch Conference, actually, is a safe place, so you can ask as many questions as you want. Remember, there are no dumb questions, and the more you ask, the more you are gonna get, okay? So you are gonna talk about us connectivity, right? And
Yes.
TEG data. So welcome, and please go ahead.
Okay, so starting my screen. A second.
Yeah.
Okay. Okay, so can you be seen?
Yeah, great. Please go ahead.
Okay, so first of all, hello to everyone. I have to say at first that unfortunately the main presenter and the main author of this talk unfortunately got ill, so I will replace my colleague Timofey, and that's why maybe I will not be able to answer all the questions about the technical details of the analysis I'm going to present, but I will try to do my best, and I will be able to address you to all these questions to Timofey as soon he's getting better. The name of the present talk is Task Classification from Pre-Stimulus Functional Connectivity on EEG Data. And basically the background of the present talk is the importance of pre-stimulus functional connectivity and functional connectivity reconfiguration for task performance. First of all, it has been shown that functional connectivity characteristics are unique for different cognitive tasks. Next, it has been shown that actually functional connectivity reconfiguration before the task can be used to predict the quality of the performance. In the paper by Ekman in 2012, it has been shown that from functional fMRI data, it was possible to predict errors from pre-stimulus reconfiguration patterns. So in the present study, we were interested in whether we can actually predict different types of tasks from functional connectivity characteristics and its possible reconfiguration. What is our design? So our subjects were presented with three types of stimuli. It was either algebraic equations with and without error. I will show you how it looked. It was arithmetic equations with and without errors, and it were sentences in native language with and without errors. The participants' task was to indicate whether they could find an error in the present equation or a native language sentence. So 76 participants took part in our experiment. The age was from 17 to 34, with average about 21, 22 years old, and 30% of the sample were female. The experiment looked like this. So first of all, there were not only tasks, but before each task, every participant was presented with the cue indicating what would be the future task. So they knew that after 2-second wait, they would be answering the question either about arithmetics or about algebraic equations or about native sentences. For their task performance, there were unlimited time, but it was really quickly and took less than a second or two to perform the task. And the participants were shown feedback whether they succeeded in all these tasks. I have to say that the equations and the arithmetic equations and algebraic equations, as well as the native language sentences, were all pretty easy. So the average accuracy for every types of tasks were higher than 80%. So basically, all the subjects did really good in performing the tasks. And we were interested whether we could predict what task, whether we could predict the task from the 2-second wait period before the task. So our idea was that after the queue, the participant had to expect the specific task, and the neural system should start with reconfiguring itself to be better prepared for the specific task. And we used this 2-second period to predict what type of task will be shown later. So what did we do? We recorded 44 channel EEG and used 0.1 to 30 hertz data with 150 hertz sampling frequency. All the data was automatically pre-processed with auto-reject package for MNE Python. For each trial, we used 2-second pre-stimulus interval to estimate functional connectivity, and we used imaginary part of the coherency to estimate the connectivity between different EEG sensors. All the data was analyzed in 5 frequency bands separately. It was alpha, beta 1, beta 2, theta, and we used full range data for 4 hertz to 30 hertz, and we left all the data below 40 hertz because of the several papers showing that functional connectivity EEG data are unstable in lower frequencies, so only higher frequencies were used. For each functional connectivity metrics, we used a threshold. We took only 25% of strongest connections to build a graph, weighted graph in our case. And for each graph, for each participant, we calculated 5 global measures. It was median path length, it was associativity degree, it was average clustering of the graph, global efficiency of the graph, and average local efficiency of the graph, as well as 5 types of local measures for each EEG sensor. It was rich club coefficient, the degree of the node, clustering coefficient of the node, between the centrality of the node, and key core index of the node. So altogether, for each participant, we had more than 300 entities in total, which was used as a network fingerprint of the exact participant. We used the MNE Python package for EEG processing, as I said before, and for the classification, we used scikit-learn package for Python, and we used multi-class sparse logistic regression as a classifier with leave one out as the train test split to check the accuracy of the classification. Unfortunately, I have to say that we were not really successful in correct classification. Only from 31 to 38% for different types of tasks were classified correctly, which is below or just a little bit higher than the chance level for classification. So actually, there were no predictive data in functional connectivity in pre-stimulus time for the in our experiment. At the same time, we were interested whether it was just our algorithm that maybe was broken in some sense, and maybe it's just the problem of the classification approach. We checked that with another type of task. We were interested whether we could try to classify the participants and from the network fingerprints we obtained. And for these tasks, we showed that actually the algorithm was working, and from 61 to 90% of the accuracy was um, uh, we showed that the accuracy of the algorithm to predict different individuals was up to 90% for different frequency bands. Here, we think that these results show us that actually all the functional connectivity EEG data can be highly variable across individuals, and maybe the problem with our initial task classification was because we didn't properly address the problem of individual differences in the sample. So basically, the algorithm worked for individual differences, but didn't work for classification of the future tasks. And we had some limitations in our study. First of all, the topological our initial idea was taken from the fMRI study, which with much higher resolution, spatial resolution of the data, and maybe the 64-channel EEG might be just not good enough for this kind of tasks. Maybe the topological resolution is not adequate for the types of tasks we were interested in. Second, unfortunately, the graph construction is routines are highly variable across different types of studies, and we used only one synchronization measure for graph construction. It was imaginary parts of the coherence, and we used only one type of the threshold to calculate the weighted graph, 25% threshold. Maybe different types of synchronization measures or different types of threshold could lead us to different results. We want to look for that into our future analysis. And our last thing is maybe the averaging across the whole 2-second period could lead us to losing the temporal dynamics of network reconfiguration, and actually it's not so sensitive to the just this 2-second period, but maybe millisecond temporal changes in the network reconfiguration are much more important for the classification of the future tasks. So basically, these three issues we want to address in our future analysis. And basically, that's all. We tried the working method, but didn't succeed in good classification. And we want to go further, looking into further details of possible analysis routines. So basically, it's it. That's it.
Great, thank you. So, is there any questions from the audience? Please feel free to ask. So let's see whether somebody wanted to say something. But yeah, I actually wanted to ask you, you mentioned something like individual differences might matter. So does it mean that, for example, there were much variance in the, I don't know, grammatic, or I mean grammar, I think is Russian, right? Or you know, math level, math knowledge, or something like that? Or what did you refer to?
Well, actually, we think that there are a lot of things that can matter. Actually, this is a part of a bigger study that was related to studying math anxiety and its impact on the neural response to different types of tasks. And we know that there are a lot of individual differences in the sample regarding IQ level, regarding their math anxiety quotations, like so there are a lot of differences. And in our initial analysis, our first try, we will go on, we averaged, like it was averaged for the whole sample, and we were looking for the classification like without addressing the individual differences. Maybe if we will try to do that on the individual differences level of analysis, adjusting for some other variables, or just doing it within subject analysis, maybe it could lead us to better results.
Right, or just separate into groups the low and high performance.
Or separating or regressing out some other variables. So basically, we know that there are a lot of individual differences, and actually there are some studies that network fingerprints can be used to correctly identify the individual with more than 99% of accuracy. In our study, it was the highest level was 90% accuracy, but altogether network fingerprints are highly unique, not only for different types of cognitive tasks, but also for different individuals.
Yeah, great. So yeah, another quick question. How do you compare between tasks? I mean, because how I see it, that probably the algebraic equation is slightly more complicated or more complex than the arithmetic, or maybe for some people it's easier the sentence, or how do you ever compare that?
Of course, there are different difficulty level. However, this our accuracy was from in this 30% zone for all types of tasks. So it didn't really differ for different types of tasks.
Oh, okay, I see. That's great. And is there any reason you just only 30% of the sample were women? Or it's more like
it was just
I see.
Yeah, we know that there's an issue, like it's there was no particular reason for that, and we want to actually invite more women to the experiment, but at the time we just had that sample.
Yeah, have you compared? I mean, I know it's not fair because it's 30 and 70%, so it's not really a fair comparison, but just like a test, have you compared between women and men and see whether there is any difference?
No, not really. Yet. We like we were more interested in individual differences in general, like, and we want to address that regarding the gender. And maybe like I don't think that our sample is enough to really look into gender differences at all. We need a bigger sample for that.
That's right. Okay, let's see whether somebody else wanted to ask something. I think it's not the case. So I just want to thank you so much. Yeah, it was a wonderful talk. I think it's a really interesting study. So yeah, good luck with that, and just let us know when you publish your data, okay?
Okay, thank you. Thank you very much for your questions.
Okay, so thank you everybody, and yeah, see you next time. Bye.
Bye.
Just a few more minutes, just another few seconds. We'll log in. Not quite on the minute yet. Okay, so welcome to session 3010, room 3 at NeuroMatch. I'm Robert Rosenbaum, the MC for this session. As a reminder, you can type your questions into the Q&A. You can upvote questions you want to hear answers to, and you can communicate with each other and us via chat. Our first speaker is Deepthi Mahishi from University of Leipzig, and I'm going to hand it over to her now. Thanks.
Thank you, Robert, for the introduction. Hi, all. Welcome to my talk. Yeah, I'm from the Hutrot Lab in University of Leipzig, the Department of Behavioral Genetics. And we work on the fruit fly, Drosophila, and we try to understand the behavior and neurogenetics in this model organism. As you can see in the title, I will be introducing you to two specific behaviors that we are looking at right now. One is feeding and the other is locomotion. And most importantly, how feeding associated locomotion or walking in Drosophila is happening. And there are a lot of studies that talk about how food search related walks in Drosophila are guided by visual and odor cues. That is, the fly sees a food source or smells a food source and approaches it and feeds on it. And of course, the other interesting aspect is how the hunger state of the animal affects such food-related walks. And of course, it's known that starvation, a starved fly shows different behavioral dynamics or walking behavior as compared to a fully fed fly. However, all this is studied until the fly approaches the food source. How does the history of starvation impact longer periods of walking activity and behavior in Drosophila is not so well understood. And to specifically explore in this direction, we have come up with a very simple paradigm. We have two sets of flies, fed flies, which are raised on general dietary media, and another set of starved flies that have been starved for about 24 hours. And then we use our own novel walking activity measurement assay called the panopticon. So we measure individual fly activity and for up to 24 hours. Using such a simple yet robust mechanism. So also I'm going to introduce you to the setup. We have, again, a simple setup. We have IR illumination because we always record in constant darkness. And the arena itself is placed on a glass platform, and recording is done using a camera on top. The arena itself is quite simple. We use our regular 90 millimeter Petri plate, and we have 3D printed sector plate or a scaffold that we place as an inset in the Petri plate and cover the whole arena with a substrate. And as indicated here, we use 1% agarose for the outer arena. And the additional feature is, of course, the localized food patches, which have an additional 200 millimolar sucrose media inside them. And as you can see, each individual fly is loaded and recorded for activity. And tracking and analysis is done using custom written scripts in Fiji and MATLAB. So using such an arena, we have some results here. We look at hunger state of the fly for about 24 hours. We based on the hunger state of the fly, we see how it affects 24-hour activity. And as you can see here, we see a characteristic evening bump in the fed flies. And the colors here indicate the regions that we have selected for analysis. So what does the starved flies look like? Interestingly, the evening bump seemed to be quite reduced. This seemed to have caught our attention, and to further better understand how the fly interacts with the food source itself, we selectively looked at how much fraction of time each fly spends on the food patch only over 24 hours. We saw that the fed flies showed very sustained preference over 24 hours. But the starved flies showed an incremental increase in preference to the food patch. And as you can see, the food is already there as soon as we introduce the flies into the arena, which means the starvation state of both of these flies are basically comparable maybe after an hour. And yet the starved flies continue to prefer the food patch. And to further understand what activity really means here, we also looked at the distribution of distances moved, different distances moved by each of these flies over 24 hours. In the entire arena, as indicated here again, we saw that the starved flies show significantly more frequent, shorter walks. And that was also very characteristic to the localized food patch area here again. And this, we thought that maybe these short distance walks could be associated with feeding behavior. And to further understand this, we analyzed the first hour of activity. As you can see here, the starved flies showed comparably increased activity in the first 30 minutes as compared to the fed flies, which sort of becomes comparable in the next 30 minutes and reach baseline. What was interesting was also the number of stops that the starved flies make in the first 30 minutes is significantly more than the fed flies. So basically they walk also shorter distances, but they also make like very short pit stops or whatever you can call it. And also as concomitant with the activity plot, we saw that the stops were also comparable in the next 30 minutes. So the next thing we wanted to look at is the feeding rate. And interestingly, again here, we saw that the starved flies showed increased feeding rate in the first 30 minutes as compared to the fed flies. And all of this adds up to the fact that the starved flies show increased motivation, feeding motivation in the initial 30 minutes, which then equalizes in the next 30 minutes, making it comparable to the fed flies. So understanding all these fine dynamics also led us to questioning more. And of course, initial feeding motivation also drives foraging behavior. But what if we remove the whole component of foraging? How does the behavior look like then? And is this affected by the initial feeding state again? And to be able to understand this, we came up with a new paradigm in the same arena, wherein instead of localized food patches like before, we now have the entire arena, like every surface of the arena covered with food. And it's like really like 200 millimolar sucrose everywhere. So the fly has no escape but to be on food all the time, on very high sucrose diet all the time. And in this paradigm, we observed that the starved flies now do not show any reduced evening activity anymore. And this further indicates that that particular reduction we saw was very related to the presence of the food patch itself. And to make this more comparable, we also selected virtual regions in this omnipresent food arena. And we can confirm that this is not a very, this is a very homogeneous effect. And to summarize these points, we can say that flies with prior starvation experience show reduced evening activity in the presence of localized food and shorter walks. So it basically indicates feeding behavior. Does this mean they cannot afford to explore more like the fed flies? Also, the increased preference for the food patch could indicate an interesting long-term food deprivation dependent memory effect. And these are the two aspects that we are looking forward to exploring in the future. Thank you for your time. I would like to thank my PhD supervisor, Wolf Futteroth, and Tilman Tripan. He's my close collaborator, and he's my programming guide, including teaching MATLAB from the scratch. And Ricarda for all the flypad experiments, and Andreas Thum, Andy, for being my co-supervisor and for hosting us in his lab and all his lab members. Thank you.
Thanks. There's not currently any questions in the Q&A, but anybody feel free to pose a question if you'd like. Either type it into the Q&A or you can unmute your microphone and just speak up. I have a couple of questions. One, you said the flies were starved for 24 hours. Do you know how that duration compares to sort of the amount of time that they might go without food in natural environments?
That would be hard to comment, given that basically whenever they are hungry, they look for food. And I wouldn't predict that unless there are very dire, situations or extreme situations of no food source available in the vicinity, I don't think that is a very natural situation. But we, of course, make sure that we starve them for 24 hours so that we know that they are starved enough to be motivated to find food and feed. So
Right.
it's just an experimental aspect.
Yeah, that makes sense. Is it consistent with all of the data you presented that the starved flies are basically tired or low on energy, and then even after they feed, they could be potentially, I don't know what their digestive processes are, but if so, they're presumably low on energy when they first start foraging, which could potentially explain the shorter walks and stops. But they're also hungry, so they're looking for food. But then even after they eat, there's presumably some digestion process that might use some of their energy.
Yes, that's a very interesting question, and we are debating about that a bit. And my supervisor has this interesting idea of rest and digest. So once you have food, and this is known in very anthropocentric viewpoints, that flies, humans, feel lethargic after they eat. When you're very hungry and you eat, and you feel lethargic. And we don't know if this is true for flies, but we have looked at the number of the duration of stops that they make after feeding. Well, I wouldn't say there is a significant difference between the fed and starved flies, of course, but the number of short stops that they make is still, I would say, a bit more than longer stops. So yeah.
Okay. And these stops, I wasn't sure when you were looking at the stops, the duration and the frequency of stops. Does that include stops at the food source, or is that stops?
Yeah, sorry, I think I missed mentioning that. That's on the food patch specifically.
Right. Okay.
We are really, looking locally at the feeding behavior. We chose.
So you don't count stops. It's not just their stops while exploring outside of the food source.
Yeah, we have excluded that here.
Okay. Okay.
To make it very specific to feeding.
Okay. Yeah, that makes sense then. Any other questions? So I guess to follow up from that, since we still have another minute or two, are you sure when they're stopping at the food source, are they always eating, or do they sometimes just kind of stop there to hang out?
Yeah, that's
any way to know?
Well, first of all, we need good resolution to make, to really understand the very fine, the fine movements and fine behavioral, aspects, which is why we started with just binning the data into these logarithmic, bins. So the scale here is basically in log 2 range. So we thought maybe 2 to 3 seconds and 4 to 7 seconds, it's not so much of feeding, but maybe grooming or just, just very tiny movements. But I wouldn't comment too much on that because we haven't really visually looked at how that translates to. But it could as well have those aspects here as well.
Cool, awesome, thank you. Very nice talk. So then we'll move on, right on time. Lost my script here. Okay, cool. The next speaker is Ivan Estevez from Universidad Carlos Tres de Madrid. And I'll hand over the stage to him.
Thank you, Robert. I'm gonna share my screen. So, yes, I'm going to present a project that consists in the replication of 2 computational models of locomotion of Caenorhabditis elegans in the neuron simulator Brian 2. And this work was made with my supervisors at University Carlos III de Madrid in Spain. So I'm going to explain why we chose models of C. elegans, these two particular articles, and the simulator Brian 2. Then I will show a comparison of the most important results. So first, why we focus on the nematode Caenorhabditis elegans, or C. elegans for short? Well, this worm has been an animal model for systems biology and neurobiology for a long time. The whole animal is composed of 959 somatic cells, of which 302 are neurons. And this simplicity has allowed to reconstruct the genome and the connectome of the worm. The connectome is the wiring diagram of the connections between neurons. But despite this simplicity, this animal is capable of displaying a variety of behaviors governed by the complex dynamics of its nervous system. And in this work, we focus on the neural origin of locomotion in C. elegans. So for this purpose, we replicated experiments from two published scientific articles that are the ones cited here. I will be referring to them as the Izquierdo paper and the Cunard paper. And the reason for choosing these two articles is that both have computer simulations of part of the connectome of C. elegans and study the neural activity that leads to the locomotion of the worm. That is to say that they don't consider the body or the muscles. Also, both articles explore different neuron models outside the perceptron-based ones used in modern artificial neural networks, and are also non-spiking. These are point neuron models that consider continuous time dynamics and have two types of connections. Chemical synapses that model the communication between neurons that happens with the release of neurotransmitter. These synapses can be either excitatory or inhibitory. And electrical synapses, also known as gap junctions, where neurons are connected physically and electrical activity travels across this connection and becomes attenuated. Nevertheless, there are differences in each of the models that each paper uses. Izquierdo uses a non-dimensional mathematical model based on continuous time recurrent neural networks. And the parameters for neurons and connections are free. So a genetic algorithm is used to search for the right values. This limits the size of the circuits that can be used with this neural model. So Izquierdo focuses on a neural unit that repeats along C. elegans, which consists of 11 neurons and 26 synapses. On the other hand, Cunard uses a mathematical model with parameters obtained from experiments with living worms. These parameters, however, are considered the same for all neurons and connections. For example, all synaptic weights have the same conductivity of 100 picosiemens. And this simplification means that no optimization of parameters is required for this model, and this allows them to work with bigger circuits. In this case, it's almost the complete connectome with 279 neurons and 3,297 synapses. Another question to answer is why use the Brian 2 platform for the replications? And there are several computational tools available for neuron simulation. Some of them are shown in this image. Most of them focus on spiking neural networks that are used to model human brain. So we chose Brian 2. One of the main reasons is its equation-based modeling, which is well suited for replication. In this case, both papers present their model as sets of equations in standard mathematical notation. Another reason is its compatibility with Python. Brian 2 is available as a Python module. And the maturity of the project that is under constant and active development. It has extensive documentation and has been widely used in the field of computational neuroscience. Now I'm going to present some of the replicated results for each paper. I'm going to start with Izquierdo. This article tries to explain the origin of the oscillatory neuron activity that leads to muscle movement for the locomotion of the worm. For Izquierdo, oscillations come from multiple central pattern generators located along the ventral nerve cord of C. elegans. This image shows the locomotion unit that that is the object of study, and repeat 6 times along the length of C. elegans. This unit consists of 11 motor neurons that innervate the muscles responsible for forward and backward locomotion. Now I'm going to show you some of the most important results. This image shows the neural activity of the best circuit found after evolution. A, the image on top, are the traces of the best circuit from the original paper, and B, the bottom image, are the traces of the best replicated circuit. Although these two sets of traces are not identical, it can be seen that the replicated behavior complies with the requirements forced by the genetic fitness function, namely that the activity of neurons is oscillatory, 2, that ventral and dorsal activity are in opposite phase. This is a necessary condition for locomotion to happen. And 3, traces of B-type neurons in green take higher values during forward locomotion, while traces of A-type neurons in brown dominate during backward locomotion. So these 3 conditions were met by the best replicated circuit. This figure shows the circuits responsible for the traces from the previous slide. A on the left is the best evolved circuit from the original paper, and B on the right is the best circuit obtained in replication. The connections in green represent a different polarity from the original, excitatory synapses that came out as inhibitory or vice versa. This shows how different circuit configurations manage to fulfill the conditions posed by the fitness function. So there are many possible solutions for the proposed central pattern generators. Now I'm going to move on to the second paper by Caenorhabditis. And the hypothesis in this paper is that oscillations arise when the whole connectome is considered as a nonlinear dynamical system, and a strong sensory input induces a bifurcation in state space responsible for the oscillatory neural behavior that leads to forward locomotion. Now I'm going to present some of the main results. This figure shows the average neuron response by type under increasingly stronger input current to the PLM sensory neurons. These sensory neurons are located in the tail of C. elegans, and the worm responds to touch by going away from it, thus performing forward locomotion. The top panels, A, come from the original paper, and B, at the bottom, are the replicated results. The input starts at 1 nanoampere, where the average neuron voltage displacement is 0. At 1.5, the oscillatory behavior starts, and it becomes more apparent at 2 nanoamperes with stronger oscillations. We can see that the replicated results for this experiment validate the main hypothesis of the paper. Oscillatory behavior does arise from sensory input that propagates through the connectome to motor neurons responsible for locomotion. Another experiment performed in this paper was the ablation of some of the neurons. These kind of experiments are usually done on living worms, where neurons are removed to find out their function. With this method, it was found that there are neurons that are vital to the locomotion of the worm. These are the AVB neurons for forward locomotion and AVA for backward locomotion. The figure on the left, A, shows the original results of a singular value decomposition after ablating these neurons from the connectome. It can be seen that AVA ablation in green maintains the 2 dominant singular values that drive forward locomotion with out-of-phase oscillations of motor neurons. On the other hand, AVB ablation in red maintains only 1 singular value, preventing these oscillations and thus hindering forward locomotion. The figures on the right, V, are the results of these ablations during replication, and the outcome is quite different from the original, as the 2 dominant singular values remain after any of the ablations, as can be seen in the upper figure. So forward locomotion continues. And what's more, these oscillations intensify after these ablations, which doesn't make much sense. And this is indicated by the concentric circles in phase space shown in the lower figure. So this was the major discrepancy with the original results that we obtained. So to conclude, the objectives of the project were met. Experiments from 2 published scientific articles were successfully replicated for the most part. The main hypotheses of these 2 papers were managed to be reproduced, and the replication process was indeed helpful in increasing our understanding on how C. elegans moves and how its neural network works. Also, other types of neural networks, more biologically inspired, were explored. And lastly, I wanted to mention that the code generated for this project is publicly available on GitHub. So this is the end of my presentation. Thank you for your attention.
Thanks. Are there any questions from the audience? Nobody typed anything into the Q&A, but if you have any questions, feel free to speak up or type in a question. So I have one question. In the model with fewer neurons, the Izquierdo model, and that model, should we be interpreting each neuron in the model as a distinct population of neurons in the worm, or do they represent something more abstract than that?
No, each node here in this image represents one neuron in the worm. So this is an abstraction because this unit, as it is not found in the worm, but it's like a statistical average of the different units. So there are like 6, 7 locomotion units that innervate the muscles directly. So this is like an abstraction of one of those units that as if it repeats itself 6 times, but
Okay.
each of the 6 are different in the real world.
But in each one of the units, each node here is actually one neuron.
Yes,
not a
it.
population. Okay, cool. Yeah, it'd be interesting to see whether that can be translated directly to a model with, all 6 units using the more biological detailed neuron models or something like that, to see if it would reproduce similar results. Okay,
Yes,
are there, oh, sorry, go ahead.
No. What I was gonna say that in subsequent papers, they explored, they attached a body muscles models to this neural unit, repeating neural unit. But in this work, we focused only on the neural activity so that we could compare with other neural models.
Right. Okay, cool. Are there any other questions for Ivan? Okay, well, we're right on time then. So thanks to both of the presenters. Really enjoyed both talks, and hope to see you all in other sessions. Thanks.
Thank you.
Bye.
Okay, so it's almost time. Okay, great. Oh, so there aren't too many people yet. Okay, I suppose we start now. It's exactly 12. So in the interest of time, we start. Gelana, are you there? I guess we could start.
Yeah, we're live. We're ready to start whenever you guys are ready.
Okay, great. So let's start then. Welcome everyone to this NeuroMath session. This is going to be an interesting session of 3 talks, mostly with a bit of a clinical flavor. My name is Shrikant Ramaswamy. I'm a group leader in the Blue Brain project at the EPFL, and I'll be your host for this session of 3 talks. So our first speaker today is Mohammad Sobi from Cairo University. Interestingly, I met Mohammad when he was a student at a computational neuroscience school in South Africa. And when I looked him up, I immediately recognized him. So it's nice to see him again. So without much ado, the stage is all yours, Mohammed. So we would like to hear more about your work on altered brain function and structure in people with carpal tunnel syndrome. Okay, he just shared his screen.
My name is Mohamed Sobi. I am a PhD student in Cairo University, Faculty of Physical Therapy, and today I will present a topic related to altered brain structure in people with carpal tunnel syndrome, systematic review and meta-analysis. First, this study registered in Prospero database for complete protocol. You need to read the complete protocol before implementing this study. Carpal tunnel syndrome is a common entrapment neuropathy occurred in the upper limb because of focal pressure in the median nerve passing in the hand. The main symptoms are pain and or paresthesia. Prevalence of carpal tunnel syndrome in females is larger than in males. So the prevalence in females are 3.8% and in males 2.7%. The interesting thing that despite the carpal tunnel syndrome is a peripheral neuropathy, there are many reports showed evidence of central changes in carpal tunnel syndrome. Like there are extra-median spread of symptoms detected in people with carpal tunnel syndrome. Median nerve usually supply lateral 2/3 of the hand and lateral 3.5 fingers. Many reports showed extra-median spread of symptoms. I mean that the symptoms should be restricted to the area supplied by median nerve. But in many people with carpal tunnel syndrome, we showed like extra median spread, like in the shoulder, in the arm. There are sometimes people, you can find some people complaining from pain and paresthesia in the arm and shoulder. Also, some reports showed limited or disrupted descending pain modulation, what's called conditioned pain modulation, disrupted in people with carpal tunnel syndrome. Other reports showed presence of thermal and pressure hyperalgesia. Thermal and pressure hyperalgesia, both topics are signs of what's called central sensitization. I mean, central changes occurred in spinal cord and brain after this common peripheral neuropathy. These signs indicated there are some changes occurred in spinal cord circuit and brain gray and white matter. Evidence of central changes also, there are some studies showed changes in MRI and fMRI regarding the gray matter. They are reducing gray matter of somatosensory, pulvinar, and frontal areas. Also, the intercortical, interdigit cortical separation distance of index and middle finger showed some sort of reduction. Unfortunately, people with central changes with people with carpal tunnel syndrome and central changes showed limited response to conservative management and usually refer to surgery. So the purpose of this study is to confirm the all brain structure and function based on previous studies using imaging modalities, MRI and fMRI, to answer our question, which is, are there alterations in brain structure and function in people with carpal tunnel syndrome to confirm the central changes happened in these people? On these people, the method we searched like 5 databases in Bayes, Web of Science, Scopus, PubMed, and Cochrane, with eligibility criteria of studies should involve adult people with carpal tunnel syndrome. The stages of the disease in these people are subacute or chronic. Also, people should be diagnosed through clinical and or electrophysiological assessment using nerve conduction studies. The third eligibility criteria is those people with carpal tunnel syndrome and the healthy control group should be investigated using MRI or fMRI to investigate two main variables: inter-digit cortical separation distance and cluster size related to digits in somatosensory cortex. This is our flow diagram. The main important part is the included studies in qualitative assessment were 9, wherein quantitative and meta-analysis were 4 studies. We implemented risk of bias assessment using Newcastle Ottawa quality assessment, which is directed toward observational case control studies. We searched only for observational case control studies to compare between carpal tunnel syndrome and healthy control. And Newcastle Ottawa quality assessment used mainly for assessment of case control studies. We can show that our 4 selected articles in quantitative meta-analysis are of high, moderate quality. Yani 5 from 9 are good quality, not bad quality. But only one study represents a bad quality, which is for Haddouche et al. 2017. To illustrate my results, I quoted a picture from Nabado et al. 2006, illustrating center of mass of second digit and third digit, both second and third digit supplied by median nerve, as well as fifth digit, center of mass of fifth digit in somatosensory cortex. Fifth digit supplied by another nerve called ulnar nerve. Interestingly, our results showed that the distance between the second and third finger supplied by median nerve were significantly reduced in carpal tunnel syndrome people compared with healthy control group. Also, the distance between the second and fifth digits were significantly reduced in people with carpal tunnel syndrome in comparison with healthy control group. The final results were the interdigit cortical separation between the third and fifth digit were not significantly reduced. So here are my results. Interdigit cortical separation distance, we used what's called the standardized mean difference. That was 0.8. 0.8 is a big effect size here. The effect size here is not small, large effect size, indicating significant reduction regarding the interdigit cortical separation between the index and middle finger. Also, the interdigit cortical separation of index and little, second and fifth fingers also significantly reduced with the standardized mean difference of 0.84, also large effect size. While the interdigit cortical separation between the third and fifth finger showed no significant difference. Our second variable is the cluster size. Cluster size, there are two high-quality studies showed enlarged activation of somatosensory areas responsible for index and middle finger. It means while after inducing some sort of tactile stimulation in the index and middle finger, there are blurring excessive activation of somatosensory areas responsible for the second and third fingers. This can explain the reduction of the distance between those two fingers. Also can explain the reduction between the second and fifth fingers, interdigit cortical separation. So here are a picture that can show the activated or blurring or enlarged cluster size of areas responsible for index and middle finger. As we can see in the left picture, this picture related to health control group, and the right one related to carpal tunnel syndrome group. Simply, the red line represents the cluster size of index finger, while the green one responsible for or represents the cluster size of middle finger, while the blue line represents the cluster size of little finger. I think it's this picture shows the enlarged cluster size of index and middle finger in carpal tunnel syndrome compared with healthy control. Our conclusion: altered cortical morphology and function, as evident by increased cluster size and decreased interdigit separation of index and middle finger, proves the central mechanism involved in carpal tunnel syndrome. So, what's the implementation of this study? As I said in the first part, people with central changes usually showed limited response to conservative treatment and usually referred to surgery. This can increase the cost of treatment for those people and health insurance also. So, if we search for new interventions that can target the somatosensory changes occurred in such peripheral neuropathy, we can improve the outcome and the quality of life for this in those people. Also, we can try some sort of treatment like pain neuroscience education, cognitive behavioral therapy, like transcranial electrical, transmagnetic stimulation of somatosensory cortex, like direct current stimulation, somatosensory cortex. All of these methods can be used in the future, trying to improve the outcomes in people with carpal tunnel syndrome. So reducing the cost, health insurance cost, improving quality of life, improving the functionality for those people. This work cannot be done without the effort of my research team. So thanks so much. And I am ready for any questions.
So thanks a lot, Mohammed, for the interesting talk. Do we have any questions for Mohammed? So I have a quick question. So in your slide 13, you mentioned altered cortical morphology. So what exactly do you mean? Because with fMRI, you can't look at morphologies of neurons in the cortex. You probably mean cortical geometry?
No, some studies use diffusion tensor imaging to study gray matter and white matter, and they showed like reduction in the gray matter, but I couldn't this data in meta-analysis. So I will refer it in some sort of qualitative assessment, because of I can find only, I don't remember actually, 2 studies or 1 study represented reduction in gray matter and white matter using diffusion tensor imaging. but I couldn't add them in meta-analysis.
Okay, sure. Yeah, no problem. Yeah, that's good to know. So, any more questions, thoughts, remarks? No? Okay, so it's time then to move on to the next speaker of the session. Our next speaker is João Gustavo dos Anjos Moraes Oliveira from Brazil. He's from the Escola Bahiana de Medicina e Saúde Pública, and he'll talk to us about the endocannabinoid system and its role in neuropathic pain. I would just like to mention that João is in a bit of a rush, so he would not be able to take any questions. So he has to leave right after his talk. So no further delays, João, the floor is yours.
Hello, everyone. Thank you, Sirikant. My name is João Gustavo Oliveira. I'm a 30-year medical student from Brazil. And today I'm here to present this part of work I've been doing with my group. I put their names here because they are so special to me. And it is entitled The Endocannabinoid System as an Alternative Way for Neuropathic Pain Treatment. Since in this presentation I will cite treatments and medication names, I'll have to declare that I have no conflict of interest here. And so before we start, understanding which treatments are being used for neuropathic pain treatment and its efficacies, I would like to introduce some concepts that may make this talk easier to everyone. First of all, what is the endocannabinoid system? So the endocannabinoid system is a group of receptors, molecules, and enzymes that can be responsible for its biosynthesis and degradation. The name of the system comes from the fact that these molecules, these primary molecules, are endogenous and they have some structural similarity to the cannabis sativa molecules like THC and CBD. Also, those molecules are called the 2-AG and anandamides primarily. The 2-AG or 2-acylglycerol, and the anandamides is called as well the arachidoyl ethanolamide. They are both eicosanoids, which are synthesized on demand by those enzymes. In the case of the 2-AG, it is synthesized primarily by the diacylglycerol lipase, and the anandamide is synthesized primarily by the N-acyltransferase and the phospholipase D. They are removed from their sites and then taken by uptake processes and metabolized mainly in monoacylglycerol lipase and fatty acids amides hydrolase in case of the anandamide. This structural similarity that I talked about, it's why we can use products of the cannabis, such as artificial products like CT3 and other aglyamic acids and as well, for the agonism of these receptors, because they have some similarity on their structure. In fact, the receptors in a timeline were the first one discovered, and then the molecules that can agonize these receptors were discovered in a timeline. So these receptors are primarily two, the CB1R and the CB2R. They are distributed in different forms in our central nervous system and as well in other tissues. In fact, in this image from Fragua Sanchez 2018, we can see that CB1R is predominant in the central nervous system, but it can be found also in peripheral nerve terminals and other neuronal tissues such as stomach, sexual organs, vascular endothelium, among others. The CB2 receptors, they are mainly located in the immune system, but they can be found in the central nervous system, as we can see. In fact, in the limbic system, there are lack of evidence, but we can see it being expressed in the dorsal dorsal horn of the spinal cord and in the periphery of the nervous system. In fact, these receptors are inhibitory G protein coupled receptors, which regulates the adenylate cyclase and MAPK signaling pathways. It means that they can inhibit the calcium influx on the presynaptic CB1 receptor activation and thereby reduction of nociceptive transmission. That's important because they will not be, these presynaptic neurons will not be able to release those neurotransmitters associated with nociceptive transmissions, such as glutamates, GABA, and other ones. The CB2R functions just like the CB1R, but it can also decrease the release of pro-inflammatory cytokines. In this image, we can see as well that the CB1R is important in very process on the nociceptive transmission reduction. So when we are talking about the spinal cord level, the CB1R is responsible to inhibit the ascending of the nociceptive transmission. That means that the nociceptive transmission through the spinal thalamic threads and the pain processes associated with the dorsal horn of the spinal cord and the spinal cord dorsal ganglion are suppressed. It can also activate in a supraspinal level descending inhibitory pathways that can come from the periaquedutal gray matter and the nucleohalphus magnus, and they can modulate as well. Also, the CB1R is associated with the modification of the affective components of the pain, so it can be found in the limbic system as well. And you might be thinking now, which conditions cause the neuropathic pain? Well, the neuropathic pain is associated with neurons and immune cells, which are sensitized by signaling substances such as nerve growth factors, cytokines, and they become pharmacologically dysfunctional and hyperexcitable. That means that they can conduct pain easier, but they are not responding as so good to our pharmacs, to our medications. And some conditions can cause that, such as HIV, the virus, which causes the AIDS, the immunodeficiency syndrome, the multiple sclerosis, trauma, not only trauma of brachial plexus or lumbosacral plexus, but trauma from the spinal cord as well. And cancer can cause this condition of neuropathic pain. It's normally a chronic pain, not an acute pain, and that's why we have to look forward for some new treatments for it. So going now are some evidence that we have collected to now. I divided some topics to discuss. So first of all, a question that has been made a lot is solitary or combined administration. In fact, there are no conclusive comparative studies for that. There are no study that compare the use of the combined administration to a solitary administration of any cannabinoid at all. But there are, and as well, there are no difference efficacy between those cannabinoids, between THC to CBD, CT3, and other synthetic. Between studies, it seems that the combination have more benefits because the combination of THC and CBD, for example, it's good to reduce some adverse effects that can be present in solitary administration. Another topic that I like to talk about is the smoke cannabis cigarettes daily administration. So in mixed neuropathic pain, it improves spontaneous relief, but have not altered stimulus evoked pain. That's important. Some conditions have lots of difference between their responsibility to this treatment. And with doses of 3.56% of delta-9 THC, there were pain relief in patients with sensory neuropathy from HIV. In fact, this 3.5% was administered 3 times a day, for a short period of 2 weeks. And cigarettes with lower 1.29% and higher 7% content of delta-9 THC shown similar efficacy. So the dose is not very well pointed. In post-traumatic or post-surgical neuropathy, cigarettes with less than 9.4% of delta-9 THC contents produced no significant pain relief. So we see that probably there are more conditions that will have necessarily more dose up higher. So the vapor inhaled administration, we see that in nabiximols, a combination of THC and CBD, was used in vaporization devices and showed efficacy for multiple sclerosis. That's interesting because nabiximols, it's called as well as Sativex as in a commercial use, and it was the first cannabinoids medication approved in some places of the world. But it is normally used as an oral mucosal spray. But here it was used in a vapor inhaled administration and has shown some efficacy as well. There are new vaporization devices that can hit the plant material, releasing the active phytocannabinoids. That's interesting because it's better than the use of the smoked cannabis because it won't produce the carbon combustion harmful compounds. The studies evaluating inhaled cannabis did it for short duration, and that's probably a good critic because we don't have any studies taking a long-term duration. Sublingual spray administration of delta-9 THC and CBD, combination in a range of 2.5 and 120 milligrams a day was effectively analgesic in the case of the oral mucosal administration. The sublingual administration was primarily made with Sativex, the nabiximol I already cited, but there was another combinations that was used with different concentrations of THC and CBD. So this study is primarily not as well conclusive. There are studies showing no advantage of nabiximols over placebo in pain relief, but in other conditions. Those troches lozengos are indicated satisfactory pain relief in multiple organs of neuropathic pain, but those troches are formulated with different concentrations of CBD and THC. In the case of oral administration, the use of dronabinol capsules 2.2 to 10 milligrams a day showed pain reduction in patients with MS and spinal cord trauma. The nabilone in lower doses was effective in chronic open motor neuron in DPN, the diabetic peripheral neuropathy, and the azulamic acid CT3 was good to lower pain and had no major adverse effects in a different concentration between 7 days. We have other associations and treatments, so the cannabis can be associated with opioids and lower pain in several conditions. They can be associated with opioids and dronabinol, a synthetic delta-9 THC. Orally, that lower pain in oncologic patients, and preclinical students have provided good evidence of endocannabinoid degrading enzyme inhibitors. But in clinical studies, those inhibitors failed to produce any significant anti-nociceptive effects over the placebo. And in a trial for a fatty acid amide hydrolase inhibitor, the BIO-10-2474, there were catastrophic results with severe deaths and the death of one volunteer. The adverse effects are a lot. The primary severe are those three ones. And for our conclusion, I have to say that preclinical tests are not effective on the evaluation since they only evaluation hyperalgesia and allodynia, and more clinical trials are needed. There are still worries about the chronic use. This might be associated with some society taboo with the cannabis sativa. And even though nabiximols is approved in many countries, there are still a long road to travel in a lot of countries, including my own. Thank you for your attention. My name is Gustavo again. My email is here, so you can contact me for any questions.
Right. Thanks, Joao. But I guess you have to leave, or do you have a couple of minutes left?
Let me see. Oh, sorry, it's already my time. If anyone have any questions, do anyone have? You can send to my email, please.
Well, I have some questions, but I'll email you later.
Okay, thank you so much.
Okay, great. Thanks. Bye. Okay, so we are in a bit of a peculiar situation here because our third speaker, Zeynep from Istanbul, has not made it yet. And it's time for her talk. So I don't think she'll be able to make it if she hasn't made it until now. So unfortunately, I think we'll have to conclude this session without Zeynep. That's a bit unfortunate, but in case you have questions for Joao, I hope you all noted down his email address. Right, okay. So I guess we'll have to conclude this session now, unfortunately. And before we do that, let's thank Mohammed and Joao for their great talks. And of course, let's not also forget to thank Gelana Tostaeva for providing all the fantastic backend support, all the much needed backend support for this session. Gelana, could you please show yourself so everyone could see you, and also thank you, if possible?
Yes. Thank you, everyone. Great talks.
Enjoy
the rest of Neuromatch.
All right, so thanks a lot, and may the force of Neuromatch be with you all.
So hey everyone, welcome to our session. So today's session, we'll have talks on the role of reward in motor learning and also just computational models of motor learning. We have 3 speakers. I'm going to introduce the first speaker. Our first speaker is Nina Van Maastricht. She is a PhD student at the Faculty of Behavioral and Movement Sciences with Jeroen Smeets at the Free University Amsterdam. And she will be talking about the pitfalls in quantifying exploration in reward-based motor learning. Take it away, Nina. The rest of us can unshare our video so that it's just her screen and also audio. Hey, Nina, you're on mute.
Unmute. Yes, okay. Can you hear me now? Yeah.
Yes.
Good. Oh. Well, I would like to talk about pitfalls in quantifying exploration in reward-based motor learning today. And exploration is one of the sources of variability in our movements. But the most obvious one is, of course, motor noise. So imagine that you have to slide with your fingertip through the target and you cannot see your hand. You might end up at different positions on each attempt you do that. Imagine that you now ended up here, so you missed the target and you receive feedback that you missed the target. Then the next time you do this, you might add some exploration. So you add some variability in order to find out which movement leads to reward. And of course, if you have succeeded, you might add less variability or less exploration. And this is indeed what we see in behavioral data, for example, in the data of Peckney et al., that people are more variable following failure than following reward. And we question how can we estimate exploration? We ask how can we estimate exploration given that we are also noisy? So how can we separate those two sources of variability? Well, continuing on the line of thought I described before, we said we can develop an additional trial-to-trial change method. And we assume that motor noise and exploration are two independent sources of variability that can thus be, of which the variances can thus be added to the total observed variance. And we estimate this exploration as by trial-to-trial changes, which you see on the right here, following rewarded trials in green and following non-rewarded trials in red. And we consider the variability following rewarded trials, as motor noise, and we assume that following non-rewarded trials you have exploration, but of course still motor noise. And we consider exploration the additional trial-to-trial change following non-rewarded trials relative to rewarded trials. But the question is, how well does this method work? And in order to find out, we simulated reward-based motor learning with 4 models in which we varied the variability sources, motor noise and exploration. So the task that you can imagine is the task that I showed you before, and the learner has 500 trials to do this, but it's actually a computer who learns this. And from the model behavior, we then estimate exploration according to our ATTC method, and we compare this to the exploration that we put into the model, which yields the similarity ratio. And if the similarity ratio is 1, we perfectly estimate exploration. So how do the models work? They all assume that you're always noisy. So if you want to slide over the blue line, you aim somewhere and you end up somewhere else because you are noisy. And we saw before that you may also explore. So to this motor noise, you may add exploration. And all models assume that you explore more following failure than following success. One of the models, two of the models assume that you don't explore following reward. A model assumes that you explore less following reward than following failure. And another model bases his exploration on a trial, not on the previous trial only, but on the reward history of the 5 or 10 previous trials. So then how do the models learn? They learn by adjusting their aim point. So it's the black dotted line. So if you have succeeded, you might update or adjust your aim point with the previous exploration. And how this exactly works differs between models. So you might do this following reward or correct following failure. You might update with the full exploration or only a fraction of it, or you might use an estimate of exploration instead of exploration itself. So, what are the results? Here you see a graph of the 4 models in the 4 different panels. On the horizontal axis, you see the exploration that we put into the model, and on the vertical axis, you see the exploration that we estimated. And you see that the data points are not on the unity line. So although our method seems so straightforward, it's also problematic. And here we will describe 2 pitfalls in quantifying exploration. One of them is related to performance-dependent feedback, and the other one is related to the estimation of variability using trial-to-trial changes. So on the left, you see 2 graphs of this similarity ratio, which we would like to have it at 1. For 2 different sources of variability, motor noise on the left and exploration on the right. And you see that our estimate is influenced by the size on the horizontal axis, so the size of the variability. So this means that with performance-dependent feedback, exploration is, or our estimate of exploration is sensitive to the variability of the learner. So we then asked why this is, and we plotted here for one simulation consisting of 500 trials, the samples from the motor noise and the exploration distributions. So on the vertical axis, the exploration, and on the horizontal axis, the motor noise. And we see that contrary to our what we implemented in the model, that motor noise and exploration on those trials are correlated. So we implemented them as independent sources of variability. And then we discovered that if we then use random reward, so we randomly reward participants independent of their performance, that this correlation disappears. Well, we can visualize this again as follows. So on the left, you see, on the vertical axis, you see the similarity ratio again. And now you see on the horizontal axis, the ratio of exploration to motor noise. And we see that if we use the random reward criterion, the sensitivity of our exploration estimate is now reduced. But off to the second pitfall, we still have a problem because we have an underestimation of exploration. It should be at 1. In order to find out how this works or why this is, we simulated the simplest model, which assumes that you only explore following failure with random reward. And in this model, well, we didn't allow the model to learn, and we didn't put any motor noise in the model. So this means that the behavior of the model is fully determined by the exploration in the model. If we now look at our measure of variability, so the trial to trial change, we take the previous trial as a reference trial, and we calculate the change in behavior relative to this trial. And in this simplest model, or in this situation, we know that this change is directly influenced by the exploration, and this exploration is dependent upon the trial outcome on the previous trial. So with reward, you don't explore, and with failure, you do explore. If we now focus on this previous trial or this reference trial, and we plot the variances of these trials against, so that's on the vertical axis, against the exploration that we put into the model, we see that we have an underestimation. And then if we simulate the same model, but now say that you always explore, so following reward you also explore, then we see that this underestimation disappears. Well, what does this mean? It means that the trial prior to the previous trial, so the trial t-2, influences the trial t-1. And of course, this happens because the exploration depends on the reward on trial t-2. So this means that reward on 2 trials back also influences our measure of variability, so the trial to trial change. Okay, in conclusion, well, although our method seemed at least to us very straightforward, using trial to trial changes for quantifying exploration is problematic. And well, we identified 2 pitfalls. The first is related to the reward that you give. If your reward is performance dependent, this induces correlations between motor noise and exploration. Well, that's surprising because we implemented those 2, motor noise and exploration, as independent sources of variability. And the second pitfall is related to our trial to trial change for estimating variance. And that's problematic because reward on 2 trials back also influences trial-to-trial change, and this leads to an underestimation of exploration in the previous trial. So that was it for now. If you have any questions, I would like to answer them.
Great talk, Nina. Thanks for the talk. So the first question for you here is from Miguel, and he says, interesting talk. Have you considered exploration versus exploitation models? How are you going to validate your model? And like what kind of experiments?
Okay, I think the first thing to clarify is that our model, we don't really have a model. We have a method and we simulate it for models. So the 4 models that we used weren't, they were specific for motor learning. And in those motor learning models, I haven't come across the exploration, exploitation explicitly named models. So I think the answer is no, not yet, but maybe that's good to do. And what was the third part of the question?
Just what kind of experiments you would use to test the model?
Yeah, so that means testing which model is best predicts the behavior, I think. So then, there are many things that you can test. So what we now are looking into is whether we use the success frequency or the reward history rather than the only the reward or the outcome on the previous trial.
Okay, cool. Awesome. So let me introduce the next speaker then. Okay. I think also the people who are not speaking, we can like unshare the video as well in addition to audio. So our next speaker is Barbara Fuellner. She's a PhD student in computational neuroscience at Imperial College. And she's working with Claudia Klopath, and she will be talking about computational principles of motor adaptation.
Thank you. Let me share the screen. Can you see the screen now?
Yes, we can.
Okay, cool. Yeah, so let's start. The big question we are interested in our lab is like linking these 3 levels shown here. So first of all, we look at a certain learning behavior, like for example here, learning how to ride a bicycle, but it could be whatever learning behavior you wish in principle. And then we want to understand how changes in neuroactivity in, for example, like motor cortex give rise to this learning behavior. And then on the next step, we would also like to understand how this change in neuroactivity come about. Like what is the origin of this change? Is it because it's driven from different brain areas, like the different input signals to these neurons? Or our like working hypothesis here, we are interested in plasticity. So how changes in weights between neurons changes then the neuroactivity, which then leads to a change in behavior. The specific like task we are looking at in this project is motor adaptation. And just let me briefly explain this. So these typical experiments are done like this, like a monkey, for example, here controls this manipulandum. And by this cursor, it then controls the cursor on the screen. And it has to do a center outreach. So it starts at the middle at the screen, and then it has to reach to one of these targets here. And the monkeys can do this task very well then. And what we're interested in is then what happens if you apply an external perturbation to this very well-trained behavior. Because if you apply then the perturbation, like this behavior is perturbed, and the monkey has to learn to counteract this perturbation. So let me briefly tell you what exactly these perturbations are. So we're looking at 2 classical motor adaptation experiments. the visuomotor rotation and the force field perturbation. In the visuomotor rotation, you see that the monkey, like the cursor trajectory, which the monkey did by controlling this manipulandum, is offsetted by a certain degree, so rotated. So if the monkey goes straight like this, the cursor would go a little bit counterclockwise or clockwise. And for the force field, it's a bit different. Like there is an external force applied to this manipulandum itself. So here the monkey has to really produce an extra force to counteract this externally applied force to do the same kind of movement as before. And in the bottom here, you see hand trajectories. So that's the typical characteristic of this motor adaptation learning. Like initially in baseline, you see very straight reaches. And as soon as the perturbation is applied, like in the early trials after the perturbation onset, you see this very curved trajectories. This means that the monkey just thought it would be a baseline trial, starts the trial and then realizes, oh, that's not working, and then does this via online error correction mechanisms, this curved last minute correction. And that's very similar between both experiments. And in the later adaptation then, like when the monkey has learned to counteract these perturbations, you see that the reaches are more or less straight again. So that's the learning behavior here. So what about the brain areas, which might be important here? So we were very lucky that we could reanalyze some data from Matt Parrish, Juan Gallego, and Lee Miller from the 2018 paper, where they recorded in 2 monkeys in M1 and PMD. So 2 relevant areas for motor control. And during, like when monkeys were doing these 2 different motor adaptation tasks. So we got this data and we wanted to, first of all, quantify how much neuroactivity change there is between baseline and late adaptation. So let me explain what how do we quantify change here? So we have the spiking data, we align it to the GoQ. Then we calculate trial averages for each neuron and each target. And we smooth them. And then we basically have for every neuron and every target such a trace here. And then we calculate 2 measures of change. One is called dPSTH. That's basically a relative error or relative difference in the PSTH traces we calculated in this step. And we compare, like we do this for the baseline and the late adaptation, and then we calculate the difference basically. And another measure we were interested in is based on like how the pairwise correlation between neurons changes between baseline and late adaptation. So what is shown here basically is the covariance matrix in the baseline session. So each point here says how a neuron covariates with another neuron. And we do this also for the late adaptation. And then we ask how similar are these values in this matrix to these values in this matrix. And we quantify this by calculating the correlation coefficient then between all these values. So how does this look like now in the data? So what we found is that there are small but significant changes during adaptation. And let me unpack this first a bit. So here on the left, it's the dPSTH. And the black line is basically if you repeat the measurement, but now in a control session where the monkey's only doing baseline. Because there might be some fluctuation anyway, or because of trial averaging, there still be noise and things like this. So that's basically the control where no learning should happen in principle. And we see that for the visuomotor as well as for the force field, it's more changes are happening compared to the control session. And the same is true here when we look at the change in pairwise correlation. Like one fact we then also notice is that in principle for the force field perturbation, there are a bit more changes in neuroactivity compared to the visuomotor rotation. But in contrast, if we compare the 2 different brain areas, there we didn't find any big differences there. So this first result is that small but significant changes in neuroactivity and pairwise correlation between baseline and late adaptation epoch. So yeah. This is what we found in the data. But now I started out with like understanding this activity changes by plasticity. And that's of course unfortunate, like we can't really look at this experimentally for now, but we use a modeling approach instead to test this hypothesis, how plasticity in PMD and M1 would solve this motor adaptation test. And of course we are aware that like the kind of the mainstream idea maybe is that motor adaptation is also heavily due to like different input signals to these motor cortices. And the argument why people maybe think that plasticity is not involved here is that, as we saw, the neuroactivity changes are so small and the pairwise correlation are so preserved. And it's normally thought that if you have plasticity, these changes would be bigger. But yeah, I mean, we wanted to test this plasticity here. So, and test if this reasoning is really true, kind of. So for this, we developed like a recurrent neural network model where we have two recurrent neural networks, which we label as PMD and M1, but they are feed-forwardly connected then, and then the output is produced via this black box R model. Just a few words on this R model. Like we needed to be able to simulate a force field perturbation. So in some sense, we need a model where you have a force and you really, it makes sense to apply an external force. And how we did this then was like we had an extra data set where we had EMG value, like EMG data, force data, and position data. And we trained like this R model on this data set and until like very good degree. And then we basically took this model as it was, fixed it, and then used it here to produce basically the positions. And like this, we really could apply an external force and could test our motor adaptation. First of all, like if you look at the performance, here you see produced traces, like the black is the target X and Y position, and the red is the position which the network produces, and it's quite good. We quantify this with the mean squared error, and we see that also our model is able to relearn these motor perturbations. So in the middle line, the perturbation is applied, either the vision motor rotation or the force field, in the two different colors here. And the model in both cases can learn to counteract this perturbation and gets almost back to baseline level of performance. But now, like the interesting part is how different are the single unit activities before and after adaptation in our model, because what people maybe think is that they would be big because we have plasticity. But interestingly, we see that the changes here, for example, in the PSTHs is really comparable to what we see in the experiment. We also see that force field gives a bit bigger changes. So that's in line with the experimental data. Also now, if we look at the correlation, the pairwise correlation, they are highly preserved, even though we have plasticity in our model. And yeah, it matches experimental data quite well. So now the last bit of extra we can do in our model is to also look at the weight changes, because we can't do this in the reality. So we're interested here, how big are the weight changes for these motor adaptation tasks? And what we found is that the weight changes you need to solve these motor adaptation tasks are very, very small. So it's only about 1 to 2% of the initial weight value you need to change in order to counteract this perturbation. And you see here, like it's ordered according to like the pathway kind of, so from input to output. And like in all of these, it's basically very small. So like the second modeling result is then we found that plasticity in PMD and M1 results in small changes in neuroactivity during these motor adaptation tasks. And these changes are actually comparable to what we found in the experimental data. And if you look at the weights, they are very well within like the biological range, which is possible, which is known from in vitro experiments, for example. So to conclude, we started by looking at these adaptation behaviors, the vision motor rotation, force field perturbation. And then we reanalyzed experimental data to see how large these changes in neuroactivity are for these behaviors. And then with our model, we tested how the plasticity in PMD and M1 model, how big of neural changes this would produce. And we found that it's actually in line with what we see in the experiment. So our conclusion is then maybe these changes are just too small to notice. So it's not really the reasoning that plasticity is not happening because the changes are so small are maybe not valid, because these tasks, apparently you can solve these motor adaptation tasks with very little rate change and activity change. So with that, I would like to thank you, my supervisor, Claudia Colopan, my second mentor, Juan Gallego at Imperial College, and also our experimental collaborators, Matt Parrish, Lee Miller, and Ray Chaudhuri. Thank you very much.
Great, thanks for your talk, Barbara. So you have one question. I'll just read it out. It's from Caro, and they ask, how do you interpret the large variance in the FF condition? Have you looked at whether there is also large variability in FF movements compared to VMA movements? And this is driving the difference in variance.
I'm not exactly sure what variance. Is he talking about the variance here in the weight change, or in for the experimental data, like this here, for example?
I think they are talking about the variance in the FF movements compared to the VMA. Yes, the slide you're showing, yes.
Yeah. What comprises here these plots are mean values for sessions. So it's basically 16 sessions or so per like plot here per distribution. So there might be just one session where you find something else, and it's also highly variable actually how many neurons are recorded. So in some sessions here, you only maybe have 20 neurons. So there you can, like if you calculate then a mean value of this neuroactivity change, you're not maybe so close to the actual mean, but maybe you overshoot, because maybe these 10 neurons or 20 neurons you recorded are the ones which change maybe a lot. That's my take on this, but we haven't actually thought about this a lot.
Okay, cool. Let's take maybe one or two more questions. How do you set up your RNNs? What learning rules or plasticity rules do you use? Could you just retrain the readout weights of the network to perform the force field task, or do you need internal recurrent weight changes?
Yeah. So how it's set up, like it's fully connected here in our setup, and the recurrent weights are drawn from a random, like normal distribution, and we train it with backprop through time. And yes, like we played around with having different, only like the readout, for example, plastic. And it also works to some extent. Like you see, the best performance is really if everything's plastic, but basically you could have plasticity only in PMD or only in M1 or only in the output. And this also gives you quite good counteraction performance. I think like the way, like it's also very variable how you solve this task, because I think you can find a lot of different solutions and you get to kind of the same performance. Yeah. You're muted.
Oh, we should move on to the next speaker. So
Thanks.
You can continue trying to answer the questions in the Q&A tab while we move on. So I'll introduce the next speaker is Antoine Decoumi. He's a final year PhD student at UC Louvain in Belgium, working with Philippe Lefebvre. He will talk about the influence of target reward on motor planning in online motor decision. And Barbara, the rest of us can just mute and.
Okay, so can you see the slides?
Yes.
Yeah, okay, thank you. So As Nili said, I will be discussing about reaching movement and more specifically about how those rewards will interact with reaching movement while we're performing this movement. So let's start with an example. So you are in front of this basket of apple and you are hungry. So you decide to eat one of these. So to do that, the command view in reaching movement is that there are two parts of the process. The first one is to select which one of these apples you are going to reach. So this selection will be dependent on many parameters, and once the target is selected, you initiate the movement and start reaching for the apple you chose. So if we looked at both parts of this process, so the first one was target selection. So the target selection will be dependent on many parameters. For example, the position of the apple will have an influence. So you will be more likely to grasp an apple which is close by than another one which is far away. The expected gain as well. So if you are hungry, you want to eat a lot of food. So you will be more likely to go towards a huge apple than the smallest one. And the biomechanical cost could also have an influence. So if you, for example, if you have to grasp an apple which is at the bottom of the basket, it will cost you more energy because you will have to move the other ones around. So this is the first part of the movement. So you have to select, when you have different options, which one you want to do. And once this is done, you can start initiating the movement. And then this movement execution will also have influences throughout different parameters. So for example, the target shape. So this means that if instead of reaching for an apple, you are reaching for a grain of grape, you won't do the same movement because the target is smaller and it's more fragile. Obstacles will also have an influence. So when you execute the movement and suddenly you have a wasp that's hanging around the basket, you will have to avoid the wasp. And so it will change the way you execute the movement. And the last one is the impact of the reward. So if you are very hungry, you will have more interest in grasping one of these apples. So the movement, so it is shown from saccadic eye movements and reaching movements that when you reach for something which is more rewarding, you will have a more vigorous movement. So your movement will be faster with smaller execution time. And what we want to do in this study is we want to see whether this reward over here, so when you execute the movement, if there is a perturbation which is huge enough, for example, someone bumping into your arm or a wasp that lands on the apple you selected, you might have to change the target you selected and you have to change the apple. And you want to see how does the rewards, so how much each of the different apple will have so how much this reward will have an influence on the way you select a new option. So to quickly come back to this example, let's say you have selected that very apple, you start the movement, and then you have the wasp showing up. And what you want to know is how much of the reward of this apple, for example, will influence the way you select it or not. So this is the question we are asking, and the way we are addressing it is using this paradigm. So we had participants sitting on this skin arm robot, so they could perform 2D movements in by moving the handle over here. So we occluded the vision of their arm so that they only see this point, which represents the position of their hand. And we ask them to perform movement from these home targets to any of these three targets. So each of these one was associated with a reward. So this was points, and at the end of the experiment, they were paid depending on how much points they collected during the experiment. And we considered two reward distributions amongst the targets. So the first one in which all the targets had exactly the same reward. So there were no differences in between the targets. And in the second condition, the central target was more rewarding than the other two. So during movement, we also, in order to introduce the replanning and reselection of targets, we introduced some perturbation loads. So as the hand crossed this line, they could experience leftward or rightward perturbation with various intensities. And during the movements, we collected kinematics, so the position, velocity, and forces at the hand, as well as EMG activity of pectoralis major and posterior deltoid, which are the shoulder flexor and extensors. So if we look at first the kinematics, so here are the results that we obtained for a typical subject. So in the same condition, when all the targets had the same reward, we observe that when there is no perturbation, most of the time you go to the nearest target. And if you introduce a perturbation to the left, for example, you see that the larger the perturbation, the more trials will reach this target. And this is the same for the right perturbation. And if we now introduce the variation in reward, in which we decrease the reward of the lateral target with no perturbation, we observe similar behavior as we had before. But as we introduce the perturbation, we see that there is, at least for this subject, there seems to be way less trials that switch to this lateral target. And if we want to look at the behavior of all the subjects and of the average subject, here is what we obtained. So we reported the switch proportion, so which is how often one of the two lateral targets was reached with respect to the perturbation load. So you observe that this is the first line of the preceding graph. As we increase the force, on average, you are more likely to go to the lateral target, and the same for the other direction. When we introduce reward, we observe for all the subjects and even on average that the switch proportion will decrease. So if the lateral target is less rewarding, you will be less likely to switch during movement. So this first experiment kind of answered our question. Indeed, the reward will be taken into account while we perform this online motor decision. So why we have to select between several options, we will take into account this reward of the reward of the different options. And another question we wanted to answer is when we look at this condition, when all the targets at the same reward, for many of the subjects at every force level, we had trials that go to the lateral targets, but trials that also go to the center target. And we wanted to see whether before the perturbation, it could be possible to know what the subject is going to do. So to do that, we look at what's happening before the perturbation onset, and we gather together all the trials that go to one of the two lateral targets and all the trials that goes to the central target. So here are the traces for forward velocity and posterior deltoid activity for the lateral target. And what is surprising is that if we look at the same data, but for the trials that reach one of the central targets, so the central target, we observe an increase in these two parameters. And specifically, we observe an increase in the activity of the posterior deltoid. So this could suggest that for the subjects, we have, if the subject has a higher EMG activity in this muscle at perturbation onset, it is way more likely to answer the perturbation and go back to the central target. And in order to see, to go deeply in this effect, we designed a second experiment where the goal was to artificially increase the activity of these muscles. So to do that, we had a very similar experiment, except that in half of the trials, we had this background load. So the load was started when participants entered this home target and stopped when they reached one of these targets. And it was always pushing to the left. And then we also had perturbation when they crossed the line, so similarly to what we had before, but in this case, all the targets had the same reward rule. So we didn't play with reward anymore. We just wanted to see the effect we observed in the last experiment. So if we look at the no background condition, which is the same condition as the condition called same in the first experiment, we observe that the larger the force in one direction, the more trials will go in, will end up in this direction. And if we look at the same trials with the background load, there looks like there are some differences in the two conditions. So these are the same subjects. And if we want to look quick, to have a better look at that, so here we compare the two values here. So we'll compare the mean values at population level as well as SEM. And what we observe is that for the left target, there is a significant decrease of the switch proportion to this left target when we add the background load. So This is coherent with what we observed in the first experiment. That is, when you perturb the hand, so if you pre-excite the muscle pushing in the direction of one target, you will decrease the amount of trials that end up in this direction, which is very counterintuitive. And what is even more surprising is that we didn't observe any effect for the right target. So for the same background level, we didn't observe any significant effect on this target. So to pack this all, what we showed here is that first, when you have to select amongst different options during a movement, you will be taken into account the rewards of the different options. So the reward will indeed influence online motor decisions. And then we also observed that there is a correlation between the switch probability, so kind of the strategy you will be using, and the background EMG activity. So what does that mean? It means that when you have to perform witching movements and something triggers an online replanning of this movement, you can have two kinds of strategies. The first one will be a robust control strategy. So robust means that it's better at responding to perturbation, and then you will be more likely to end up in the center targets. And this strategy is characterized by higher reflex responses to perturbation. And the second strategy will be a more flexible one. So this strategy will have will correspond to the one with a smaller EMG activity, and it will be more prone to switch target during movement. So when you have a perturbation, you will be more likely to go to another target during this movement. So with that, I would like to thank my advisors and funders, as well as all the other members of the lab, and would be glad to answer any question. Thank you.
Great talk, Antoine. Thank you for that. We don't have any questions just yet, but let's give the attendees a minute to maybe post their questions. In the meantime, Barbara, if you're still there, do you want to answer a couple of questions that you had that we had to okay, there's a question for you now, I think. There you go. So the question says, did you check the triceps EMG activity, Antoine?
So in previous experiments, so if we go back to this slide here, so we checked in previous experiments the activity of the triceps. The problem is that participants were free to put the elbow as they want. So if we record triceps activity, we don't have stereotyped responses as we have for the pectoralis major. So we would expect behavior. So in the triceps, we would expect behavior similar to what we observe in the deltoid, but the problem is that with the position of the arm, it's not possible to, so depending on how you will put your elbow during the movement, you will have different responses. So we didn't look at it in this setup, at least.
Okay, cool. The next question for you is, how do you expect patients suffering from depression to respond to this task? Would the effect be more marked on the reward or on the perturbation?
So yeah, this is a good question. And one thing that we observe is that amongst participants we recruited, we had many different behaviors. So some of them were more on the robust strategy, some others were more on the flexible strategy, and yeah, we had many differences across subjects, and even in the subjects, depending on the block. For example, at the beginning of the experiment or at the end of the experiment, we had slightly shifting behaviors towards, like, if they are bored, they will be less likely to answer, just want to finish the experiment. So I would say it really depends on the person, but it could be in this case more relying on the flexible strategy and then just let it go when there is a perturbation.
I see. Okay, and one more question. So what was the reward protocol distribution? What was the reward protocol or distribution that was implemented? How was the reward implemented?
Yeah, so each target, so the large reward gave you 5 points, and the the smaller reward give you 1 point, and at the end of the experiments, so there were a scale, and participants knew beforehand, so they could earn from 10 to 15 euros, and depending on how much they scored during the movement, they get the compensation accordingly.
Okay, cool. Thank you. Since we actually have 3 minutes, Barbara, do you want to take those 2 questions that you answered like in writing in the Q&A tab on screen? Do you want to just address them? If you're still here, you can just, yeah.
Yeah, I can do.
Yeah, go ahead.
Let's see.
Yeah.
Just have to find them.
Yeah, so it's in the answered tab. So
yeah, so one question was if I could imagine a different mechanism instead of, I guess the question was instead of weight changes, that the neurons are implementing this change via synaptic adaptation or normalization, which I interpreted more as like change in excitability maybe of neurons. And I think, yeah, very well. I mean, I think take your adaptation mechanisms on hand and it could do the job. The question is, we don't know how this is done in a goal-directed manner. So if the network can do this, so it knows how to change its excitability of single neurons to produce a certain behavior, then yeah, of course. But I think it's unclear what really and how this is done in the brain. So yeah, hard to say. And the other question was, referring to my second hypothesis, which I didn't really show, where you have input, that the inputs to PMD are changed, and this is then what does the motor adaptation. So you don't change the recurrent rates, but instead you change the input drive. And we also considered this more thoroughly. So we implemented a model where we have an upstream network, and then motor adaptation is happening through change of a different drive to PMD and M1. And the results look very similar. So it's really hard to dissociate both things from just looking at the activity changes, because they are actually on the same level, which is maybe counterintuitive. So maybe one need really more experiments, I don't know what's possible about shutting down plasticity in PMD and M1 and see what happens, something like this.
Cool. So I think the session will end in a minute here. I want to thank all the speakers for a great session. Thank you, everyone. I'll just applaud on behalf of the attendees.
Hi. So should I start sharing my green screen now?
Sure.
Yeah. Okay, great. Wait. Is it good?
Yes.
Okay, cool. So should I start now? Cool. So hi, I'm Ken, and today I'm going to share our project, the effects of task on object processing reviewed by EEG decoding. And when you think about object processing under different task demands, there are two possibilities. First, we may generate one representation and then do different kind of manipulations on this representation. And alternatively, we may generate two different representations depending on the task demand. And in this study, we are trying to ask whether we represent an object in different ways depending on the task demand. So to answer this question, we created 16 artificial stimuli that can be categorized in 2 dimensions, texture and shape. So we have large dot objects here, and then small dots, and then round objects and cubic ones. Okay. And we imported our verification paradigm so that our Q is presented first, and then after 1 second, an image of the object is presented. And we asked the participants to judge whether this object matched with the Q. So for example, here is a round object. So the answer is yes. Okay. If the queue here is large or small, then it's a texture task because the participants need to make a judgment based on the texture dimension. If this is round or cubic, then it's a shape task. And for decoding analysis, we imported a linear support vector machine for the classification. And we selected 67 posterior channels out of the 128 we recorded. And we did it for each participant separately using a sliding time window and also a tenfold cross validation. We also performed the PCA to reduce the number of dimensions. For the analysis, we actually did two main analyses. For the first one, we compared the decoding accuracy of task reference and task irreference classifiers. For example, here, if I'm going to decode the EEG pattern of these two objects in the shape task, it is a task reference one, because these two objects have the same texture, which is large dots, but they have different shapes. So this is round and this is cubic. However, if I did that in the texture task trials, then this is a task irreference decoding. And our prediction is that the task reference classifier should have a higher decoding accuracy. However, our results found that there's low significance difference between the two types of classifiers across time. And then in the second analysis, we compared the accuracy of task reference and across task classifiers. For the across task classifiers, we trained the classifiers in one task and then test its performance in another task. So the purpose here is to test whether what the classifier learned in one task can be fully generalized to another task. And we predicted that there's low full generalization, so that the task reference classifiers will have a better performance. And we found that there are several time windows within the 220 milliseconds and 490 milliseconds in which the task reference classifiers have a better decoding accuracy. So in conclusion, we found that object representations were not more separable in the task reference dimension, but they changed qualitatively across tasks. And the timing of this effect happens within the time range of N250 ERP component, which is an important component in the object recognition studies, and is believed to be originated from the high-level visual areas. And thank you, and here's some contact details for my lab and also myself. Thank you.
Thanks, Ken, for the interesting talk. We have a question from Ilya, and the question is, did you do cross-participant decoding to uncover any common electrodes which were highly weighted?
First, we didn't do the cross-participant decoding, and then it may be interesting to try to do that. And also for even we did that, and under because we did the PCA for both the time and electrodes dimension, so we mixed the 67 electrodes and the 50 time points together, and then did the PCA to extract the components. So maybe we need to do the PCA on electrodes only to extract any heavy weighted electrodes.
Mm-hmm.
Yeah.
Okay, another question from Liu. Did you use ERP or also time frequency data to train the classifier?
We didn't use ERP to train the classifiers, but we also did the ERP analysis. Interesting is that there are also some effects, similar effects in the N250 components. So the results are comparable in terms of the ERP analysis and and the decoding analysis. And for the time frequency data, we didn't manage that because our focus is on the because our ideas is originated from the ERP analysis, so we didn't consider about the frequency domain.
Did you like low pass filter your data?
Low pass filter?
Yeah. Did you like in your pre-processing?
Yeah. Okay. So as I could remember, it's 0.05 hertz to 30 hertz. So a band pass filter.
30 hertz band pass filter. Yeah. Okay. Yeah, that's normal usually in visual task to.
Yeah.
So another question from Lydia. I was also wondering if you combined all your participants together into a super subject to see if the difference became larger. Were your participants very different from one another or around the same? So basically, first, how many subjects did you have, Ken?
36.
Oh, 36. Okay, so that's a big level of participants. So did you do the decoding analysis for each participant individually, or like you put them all together and performed it?
We did that individually. Yeah. But it may be good to try to combine them. Yeah. We've heard about that, but we didn't really consider that in our analysis. That's a good suggestion. And for the second question, I would say that there are few participants are very different from the others. So I feel I cannot say outliers because they are not excluded from the analysis, but the decoding accuracy was significantly lower than the others. Yeah, first perhaps around the others are like 70 something, and they are like 50 something. The percentage, yeah.
So there, I could see that there is a difference in your temporal decoding times. There is, but it didn't come significant, right? That's the thing.
You mean the first analysis?
The first analysis.
Yes.
There was, for example,
there's a numerical difference, but yeah.
But it's not a statistically, right?
Okay. Yeah, perhaps we can try to combine the subjects.
Yeah, like Ilya added that like 30 participants. That's probably like the question is less important, but like yeah, I agree. Like we usually get around 16 to 20 participants. So you had a good pool of participants. 36 is very good.
Yeah, we try to connect more because to ensure the power.
And the difference between your conditions are very small. Like for example, the small dots versus like large dots. It's really like the difference is visually small. That could be the reason that you don't get like strong signal differentiating task relevant versus task irrelevant conditions.
Yeah, that's
for shape, you are very different.
Yeah. Actually, the participants also reported that it's hard to differentiate this like the small large dots and the large small dots. So especially for these two, yeah, it's quite close.
Yes, they are. Okay. So I see another question from Cyril, if I pronounce the name correctly. Could it be also that the time window of differences are inconsistent across subjects but exist? So meaning that like the temporal dynamics of task effect is there for each participant, but they are not at the same time. Did you check that visually in your data?
Yeah, that's possible, but we didn't check that. I'm not sure how to check about that because currently the analysis the significancy is tested on the across subject base. So yeah, it's like a t-test. So you need to have subjects too.
So it will be quantitative. It will be qualitative. It will not be quantitative because you can just visually check the, for example, task relevant curve and task irrelevant curve for each individual participant. See if you see a difference, and maybe they don't match in terms of temporal dynamics. That's something for you to know and understand better, like why it doesn't come significant, for example. But I agree, for like to do a statistical test, you need a pool of participants. Okay, great. So let's thank Ken for the great talk.
Yeah, thank you.
And we can start the next talk by Ryan O'Donnell from Penn State University. And yeah, we are excited to hear.
Okay, hi everyone. My name is Ryan, and today I'm going to be talking about two different analysis techniques performed on a similar task and what we can learn about the time course of information processing through both ERP and machine learning classification analyses. But first, I want to outline the cognitive theory underlying our questions. So how does the brain build a memory for a visual event? So we know from ERP analyses that the time course of information processing is important to consider. So for example, when you look at an average ERP waveform, you'll see some early visual components like the P1 and N1 versus later higher level cognitive components like the P2, N2, and P3 that can be more associated with target selection and even target encoding. And all of these play an important role in understanding how a visual memory is built. However, we also know that the way information is presented can matter as well. So for example, if a target is presented in a rapid serial visual presentation format such that it's masked by distractors, or presented in isolation in what we're calling a skeletal display, you can see that the time course of the P3 changes based on the format, where it's occurring faster in the skeletal display, presumably because there's less visual interference and a greater ease of selecting the target for later encoding. However, while ERPs are great for correlating cognitive mechanisms to EEG data, it says nothing about what the actual underlying information representation could be. So what is actually it within the component when that component is being activated. However, neural decoding analyses can correlate item-specific information with underlying neural activity, and they've been growing rather popular in cognitive neuroscience with the increased computational abilities that we now have. So you can see in this study, the classifier was trained to distinguish a set size 2 versus a set size 6 memory display. I mean, it can do so both during early sensory stages, early visual stages, as well as during a maintenance period. So what would happen if we apply this machine learning analysis to the presentation format that we had previously discussed, an RSVP versus a skeletal display? So if we considered that a classifier was detecting early visual effects, we might expect that during the skeletal stream, it would show high classification early on compared to the RSVP, which would have a rather decreased and shortened time course due to the onset of visual distracting information. However, and a rather paradoxical prediction is that because the stimuli are masked and the task is more effortful and difficult to complete, information will need to be held longer within the brain. And this could actually increase the time course of classification accuracy during RSVP, as that information needs to be maintained more effectively to be able to complete the task. And so this is what we predict we would actually see in our classification analyses. So in the current study that I will highlight, we had participants complete 2 different presentation formats while EEG data was recorded. So first, in the skeletal or single presentation stream, participants were to detect one target, either a face or a body that was not followed by any distractors, compared to an RSVP stream where a target, again, either a face or a body shown for the same amount of time, was now flanked both by distracting chair pictures as well as a visual, highly salient visual mask. And then across both tasks, participants had to report the exact identity of the target. So behaviorally, what we can see is that the RSVP task was in fact more difficult, as participants were less accurate in completing the task for identifying the item, but just about as accurate for identifying the category. And we also see replications of the ERP analyses shown previously. So the P3 has an earlier onset latency for the skeletal display format as opposed to the RSVP format. You'll also notice in the RSVP an oscillatory effect. This is evidence that the distractors are being detected by early visual processing as well, creating an SSVEP, or steady state visually evoked potential. This is very common and can be thought of as little N1, P1 complexes occurring. So again, the distractors are attracting visual processing. So if we imagined that the classifier was picking up on this P3 difference, that we might see stronger classification accuracy in the single, what we in fact see is an equal time course of classification accuracy between both the skeletal and RSVP processing streams. Thus, the information that's represented in the brain for these different cognitive mechanisms is approximately the same amount of time, regardless of the presentation format, which is very interesting because even though the information that can distinguish faces versus bodies is probably undergoing different processing at each time, it's occurring for around the same amount of time. So it's rather mechanism invariant for the classifier. I mean, if we compare the actual time course between ERP and decoding, we can see that while there is a significant latency difference in ERP, there's no difference in SVM, and the difference between the latency difference between the two analyses is also significantly different. So finally, we used one last classification technique, temporal generalization, which can reveal novel insights about whether the information signal changes dynamically over time, such as this chain activation, which would be so high accuracy across the diagonal when the training and testing is at the same time point, versus a rather sustained activation where even when testing at different time points during the study, the activation remains significant over time. And what we can see from our 2 temporal generalization plots is what looks like a really strong diagonal for both presentations, but when we assess each classification point for significance, we see a much stronger dynamic representation in the RSVP stream compared to the skeletal stream, which shows some dynamic processing or dynamic representations, but a slightly more sustained activity as well. So to conclude, as we view visual information to remember for future use, signals from the brain can be analyzed using various techniques. From our results, the ERPs and classification seem to tell us 2 different sides of the information processing story. ERPs highlight the early onset of encoding in single/unmasked information because of the pop out of the target and ease of selection. But in classification, though we theorize different time courses, there's no measurable difference that can be decoded in the brain. So these differences highlight the importance of utilizing both analysis techniques. ERP research is not done yet. ERPs can highlight the cognitive control mechanisms that information undergoes, but is rather information invariant. And contrary to that, neural decoding analyses can detect the information representation over time, but is rather mechanism agnostic. It doesn't matter where in the visual stream that information is being processed, so long as it can be used to detect the difference between a face trial versus a body trial. So it's important to interpret both of these analysis techniques to get a fuller picture of the story. So thank you for your time. And I want to give a special thanks to everyone in my lab who's helped with this project, as well as the RAs who helped during the data collection, and all the participants who participated in this study. So thank you, and I will take any questions.
Okay, thank you, Ryan, for the interesting talk. we can take questions now from the audience. Okay, so you find that, let me ask one question. So when you do the ERP analysis, do you compare the face and body conditions?
I have looked, and I actually do have that data here.
Like, so this, okay, so, oh, okay. Can you explain this now?
Yes, and we have never analyzed this for significance, but it looks like there are slight differences in the PZ or electrode for whether a face or a body was presented in either presentation format. So you can see potentially an increase in amplitude for the P3 component and maybe a slight latency shift. And this would be what the classifier is picking up, but it's also important to take into account that the classifier is being trained across all 28 used electrodes. So even if we see differences just at this electrode, it may not be, this isn't the full picture that the classifier is picking up on.
So can you explain, like, what do you classify in your classification analysis? What are the conditions that you're classifying?
So we do a within presentation format classification where we train the classifier to detect either a face trial or a body trial. And then we test on, so it's a 90-10 test split.
Okay.
So,
because what you show me here is that, like, your ERP that cannot show the difference between the body and face, but your classification can show, right? Because you have a higher classification accuracy.
Correct, yes.
So maybe I'm confused, but like, what do you compare when you compare ERP analysis and classification? What are they comparing the same, like, are trying, for example, in the classification, you're trying to do a classification between two conditions. Do you do the same in ERP or no? That's something else.
Yes. So it is a little, it's a little confusing to think about what the comparison between the analyses would be. We're doing the latency of the area under the curve between the two techniques. So what's the 50% latency for the P3 component in the PZ electrode versus the latency of the classification, and that latency difference is what we're saying shows an early onset in ERP, but not for classification. I'm not sure if that answers your question.
I'm asking this because, like, in my experience, I usually find that classification analysis finds even subtle differences between conditions, whereas like in ERP, it's very difficult to get that, right? Like, so I find that the classification analysis is more sensitive to condition, to the like comparing conditions. So I wasn't quite sure why you don't see that in your analysis.
That's a good question. It could be because we're using single trial data. So I've seen that a lot of classification papers bin their trials, create little mini blocks. We actually found in our pipeline that didn't didn't show classification as we expected. It was rather oscillatory, and we just think that was noise. And we actually found a more coherent classification in single trial, but because it's a lot noisier, that could be where the difficulty is there. I don't honestly know why the mini blocking didn't work. I've tried asking researchers about it, and they seem shocked as well. So if you have any insights, I'd love to hear them.
Sure. Like as I said, like I need to know more about like your conditions, like what you're classifying. Then I would be happy to give you more feedback. And very interesting results, by the way. We have more questions from the audience, so I would like to ask them before like we are done with time. So Ilya asked, did you do any feature selection in order to potentially increase accuracy?
No, we did not. we tried to keep it as simple as possible to minimize fitting.
Okay. Or do you use all electrodes? I'm not familiar with which electrodes time points are best for memory. So, okay. So feature selection could help also classification accuracy. Then another question from Chloe. Did you do any classification of the distractors to see if representations are held at all in the RSVP paradigm?
That's a great question. And no, we haven't tried that yet. But we have a lot of time in this pandemic. So we could certainly test it out. We've also considered just classifying the exact presentation format, but we suspect that accuracy would be rather high for that as well.
Okay. Another question from Catherine. Interesting results. Do you think the ERP would also be information agnostic to other modalities of sensory information, such as vibration-based electrical tactile stimulation?
That's a great question. It is out of my area of expertise. I would need to test it to be able to give you an answer that I feel comfortable sharing with a crowd. But I guess I can say perhaps it's worth testing for sure.
Mm-hmm. Okay, so I think we are done with time. Thanks, Ryan, for the interesting talk. By that, I invite our next speaker, Ingrida Antonova from Nensky Institute of Experimental Biology to present.
Can you see my slides?
Yes.
Thank you for introduction and for organizing this session. I would like to present our recent results from Audible study. Audible task is one of the most popular tasks to evoke P3 wave, which is considered to be related to high cognitive functions and fluid intelligence. However, spatial temporal characteristics of this component are not well demonstrated, and we aimed to investigate them by applying ERP microstates analysis, which is topographic ERP analysis and can also show network activation. We asked several questions. What is microstates dynamics of stimulus processing in this free stimuli visual audible task? Do rare stimuli activate additional microstates which represent different networks and additional steps of information processing? Also, do common information processing steps differ between conditions in their temporal dynamics? And what are audible microstates correlates of fluid intelligence? To answer these questions, we recruited 41 healthy participants and recorded high-density EEG. Our task consisted of 4 runs, and in each run, stimuli were presented one by one randomly, but were grouped in mini blocks by 4 stimuli, and breaks between mini blocks were a bit longer than between stimuli. Our standard stimulus was letter P, while deviant and target stimuli were modifications of letter P. And participants were instructed to press response button only when we saw target on the screen. In separate psychological session, we collected fluid intelligence data and used Raven's advanced progressive matrices. And for ERP analysis, we were interested in duration, area under the curve, and mean global field power of microstates. Here on the left, on the right at the top, you can see 8 maps of microstates which represented our data best. Each of them has assigned color which shows where that microstate was present under a global field power curve. So top line is for standard condition, middle one is for deviant, and bottom one is for target. And you also can see at the bottom wave shapes of all three conditions at FCZ and POZ electrodes. So to better understand what show GFP envelope, you can imagine that you flip your ERP to the positive side, and it looks similarly. So we observed that there are three different processes in deviant and target condition: early, blue one, this olive one, and gray one. And they are not present in standard conditions. So they are additional to rare stimulus processing, and duration and area under the curve, both with global field power mean, so amplitude was higher in target condition. Also, we observed that there are some differences in common processing steps, namely microstates 5, this purple one, which represents early P3 component, lasted longer and had higher amplitude in standard condition compared to deviant and target conditions. So we asked question if we can see these differences related to Raven scores. For that, we grouped our participants into 3 groups based on Raven scores, and applied 3 by 3 factorial microstate analysis. So you see the same maps, and now in columns you see groups. The only significant difference was group effect for microstate 5, so this common processing step, and it showed that amplitude of this microstate was larger in group with lower Raven scores compared to medium score group and high score group. And interestingly, it did not differ between 2 groups with higher scores. Since we have this analysis, this difference in common processing step, we checked also if there are any differences in behavioral results. We did not find any differences in reaction time presented here at the top. However, there were significant differences in accuracy, and accuracy decreased from standard to target to deviant condition in all 3 groups. And also, low score group had significantly lower accuracy compared to medium score and high score groups, while as in microstate analysis, these 2 groups did not differ in accuracy. And these differences were driven by significantly lower accuracy in deviant condition in low score group. So to conclude, later P3 component associated in our data set to microstate 6 represented additional cognitive processes active during processing of both rare stimuli and possibly contributed to target discrimination from distractor, while the earlier P3 component associated with microstate 5 represents information processing step common across all 3 conditions and sufficient to recognize standard stimulus. And interestingly, this earlier P3 component also differentiated a group of subjects with lower scores of fluid intelligence from the 2 groups with higher scores of fluid intelligence, possibly indicating that this group of subjects with lower scores experienced difficulties to differentiate a rare target from both frequent stimulus and rare deviant stimulus, which was also supported by higher error rates in this group. So that were all findings, and I would like to thank you all for attention, and also thank my colleagues and collaborators and funding source. And if you have any questions, please ask, and you can also email me and discuss further.
Okay, thank you so much, Ingrida. An interesting talk. We have a couple of questions already in the Q&A. So Anka asks, how were the microstates computed? Please elaborate.
They are computed from mean subject ERPs and grouped based on topographic similarity of single time point topographies. So it takes values of all electrodes, builds topography like a map, and then combines similar ones. And then the most common topographies across all groups, conditions and participants recognized as this cluster maps. And then later we analyze our data using this cluster maps. So it's the simplest and shortest explanation.
Okay. So you do some clustering to identify those microstates.
Yes, yes. Okay. It was done using K-means clustering in this case.
Okay, so what do microstates represent biophysically? This is a question again by Priyanka.
They are suggested to represent neural network activation.
Mm-hmm.
And what can we see? For example, sometimes we consider ERP as one wave, but microstate analysis shows that within this wave, there are two processes. So something similar happens at the beginning than at the end. Like here, we have also this earlier P3 and later P3, and we can see differences. We plan to do some source reconstruction later to see what exactly is different.
And Levi sends us a paper related to this. I think this is like to answer Priyanka's question. Thanks, Levis. And Levis also asked a question for method clarification. So how were you able to drive an accuracy score to the standard and deviant stimuli? I assume that participants were only required to respond to target stimulus.
That's right, but sometimes they responded to a standard and deviant stimuli, and that was considered as errors. So we then computed accuracy scores.
Mm-hmm. Okay, thank you. So we still have time if there is any questions. So I don't see any more questions.
May I ask a question?
Oh, sure.
Yeah. So, what do you think about MS4, so microstate 4? Because it's quite interesting. It's earlier, much earlier than P3. So, what do you think it might represent?
It may represent the beginning of P3 component, which is a bit different in this deviant and target condition. So already recruiting some additional processing. But it's not easy to interpret it now. It would be easier when we have sources. But it shows that something additional already is recruited, then participants see rare stimulus.
Okay, Levis continues the question on standard versus deviant accuracy scores, and asks, why didn't you consider a sensitivity scores, like D prime, for example, which
talking about behavioral, yes, scores.
These are D prime?
Can you repeat question?
The question is, why didn't you consider a sensitivity a score instead of accuracy?
I see. We did not consider that, but yeah, thank you for suggestion.
Yeah, it's a good suggestion, I think, because then you can compare detection and false false alarms, and it's a better comparison. Okay, so I think that's it. Lewis has some suggestions that, like, again, like using the sensitivity scores rather than accuracy. And we are, like, it's 9:45, so we are done with the session. Thank you for attending. And let's also thank our speakers, Ken, Ingrid, and Ryan, for a very good and interactive session, and very interesting talk, and very good questions from the attendee. Thank you, and have a great day.
Thank you.
Keynote, and I am ably assisted by Yang Chu, who is our host on the back end. And it's a delight for me to introduce our speaker for our keynote, Diego Restrepo. And I'm happy and excited to be actually doing this and introducing him, because he's going to be talking about cerebellar circuits. And the cerebellum for so many years has been such a Cinderella of the central nervous system. There are more neurons there than in the cerebral cortex. And yet, all of us, me included, have focused on the cerebral cortex. So it's going to be really interesting to hear about these cerebellar circuits. The cerebellum has very, very interesting and idiosyncratic circuits. And Diego is going to be telling us about decision-making and activity in cerebellar circuits. So without further ado, Diego, it's all yours.
Thank you. And good morning from Denver. And I'd like to thank you. And it's wonderful to be in NeuroMatch. And so let me start by sharing the screen. And I'm going to be talking about decision making. So hopefully you see the screen. And what's important here is, so this, by the way, started for a sabbatical. I was doing a sabbatical with a little microscope that you see here that Emily Gibson and I generated. And I went to Isabel Llanos' lab. And I usually do work on olfaction in the front of the brain. And I became I was always extremely interested in the cerebellum because of what Cajal had done and a lot of people have done. And we started doing experiments and they became really, really interesting. So let me talk about this. And it has to do with go, no go, split decision making. And I'll explain what that is in a moment. And the work, of course, the important thing is the people that did it. And it was done by Mig Ma, who is a neuroscientist in the lab, Greg Futia, an engineer, and Beris Osbey, who helped us with the microscope as well, another engineer, and Fabio Simões de Souza from Brazil, who helped with modeling the cerebellum. So, and it's funded by the National Science Foundation and the NIH. So let me explain to you what go, no go is. And I'm going to show you Roberto Clemente, who was playing for the Pittsburgh Pirates in 1971. And I'm going to show you a very quick movie, 3 seconds, on purpose. And the important part is part of the 3 seconds. So let me just start the movie. And I'm going to turn off the volume. And it's only 3 seconds that I want to show you. So I'm going to start here at 1 second. and take a look at him hitting a home run. He hit 3 home runs in that game. And so he's looking at the pitcher and then hits the home run. Okay, that took place in a fraction of a second. And it's a very difficult decision, not just because you have to process the visual input, but because you have to process the history of what the pitcher was doing, what you're thinking, how you're feeling. And so it's a very difficult decision. And the cerebellum is involved in it. I'd like to call your attention to work with athletes, elite athletes, that was done by Owens et al. So I'm going to go to the next slide. And this is work where they worked with elite athletes, making split decisions, split-second decisions. And in cerebellar cortex, they found that in the medial and lateral cerebellar networks, there's prominent activity during the fast feedback stage of the task and before there's any movement. And so that calls our attention. That's a go, no go decision. Is the ball in the strike zone or not? So that kind of decision, of course, is done in the cerebellum. This is from Ramon y Cajal. This is the output cells, the Purkinje cells. And then we have the input, the sensory motor input, all the way through the mossy fibers, through the granule cells. And we're going to be focusing on these molecular layer interneurons. So we're going to look at those during the go, no go task. Now, in our case, the go, no go task is done with a mouse. And so I'm going to show you the task. So this is a mouse that is thirsty and starts roaming around the cage. And pretty soon they find that if they lick here, they get water. And then we start giving them 2 odors, 1 of 2 odors. And they have to lick for the rewarded odor. So that's the go, no go in this case. That's hitting the home run. So you'll see the mouse coming in, mouse licks, the odor comes in, that is the right odor, and they have to stay there for 2 seconds and lick, and then they get the reward. Then the mouse is going to come out and we'll have the unrewarded odor. And in that case, they do start licking, but then they stop licking very, very quickly. So what we wanted to do is, initially it was just because I wanted to record from the cerebellum since I was going to Isabel's lab, but very soon it became very interesting. What we did is we took PV Cree mice and we injected a virus expressing GCAM6 or GCAM7, and we recorded from the lobule 6. And that was because Giovannucci and Wagner had talked about neurons in this area, in that case, granule cells, responding to reward. So basically responding in a cognitive task. And also Rowan et al. Had suggested that these neurons gate for learning with valence. They actually respond to valence. That was a suggestion. So again, this is the input, sensory motor input, goes to the granule cells, and I'm going to be talking about these molecular layer interneurons that then inhibit the Purkinje cells, the output cells. So let's look at what happens. Now, this was now done in a head-fixed animal. So on the left side, you see the head-fixed animal, same task, the go-no-go task. And now we have odors A and B. The A is the rewarded odor. If they lick, and that's a dry lick, by the way, they lick for these 2-second segments, then it's a hit and they get water. If they do not lick, it's a miss. And then for odor B, that's not rewarded. If they lick, it's a false alarm and they do not get water. They actually get a delay in the next trial. And if they do not lick, it's a correct rejection. Now, what happened with the molecular layer interneurons? So you see now the field of view with a 2-photon microscope. And by the way, there's 2 types of cells, the stellates and the baskets. We went more superficial, so these are stellate cells. And what you see here is the response of those during the trial. So you actually see a large change in calcium, in GCAM6 fluorescence. And when we analyze this, we use actually something that's called Kaiman from Giovannucci et al. It's a constrained non-negative matrix factorization. And what that does is it actually pulls out the components that are independently firing, independently changing calcium in that field of view. When we do that, then we find the following. This is now a time course, and I'm only showing a subset of the regions of interest of the components. At the bottom, I'm showing the leaking. So whenever there's a little spike there, there's a leak. And these red vertical bars are when the rewarded odor was delivered. The blue ones are when the unrewarded odor was delivered. This was a mouse that was already proficient. So they had learned to respond to the rewarded odor. And what you see is really interesting. Basically, when you look at the rewarded odor, the red ones, the animal licks, of course, and there's an increase in calcium. So, and that happens in all of them. And when the animal receives the unrewarded odor, except for a small number of times, like in this time they actually responded, most of the cells do not respond at all. So we became very interested in this. This was initially just an experiment to go to Isabel's lab, but then we became interested in this, and we started asking, is this changing as the animal learns? So this is now trial number on the x-axis, and the y-axis is % correct, and it's computed in a 20 trial window that shifts. So at the beginning, the animal is naive. They do not know which odor is rewarded. Then as time goes by, they figure out which odor they're supposed to respond to, and at this point, they're proficient. They're 80% or better. So they actually have been able to respond to the odor. What happens with the calcium changes in the MLIs? Now I'm going to show for each trial. So I'm going to show an average, and I'm going to show it for each trial. On the right side is an average. This is when the animal is naive. At the top, we have delta F over F. So that's actually the change in fluorescence in the GCAMP. So calcium goes up, the fluorescence goes up. For S plus, which is the rewarded odor, or S minus, the unrewarded odor. And at the bottom is the leak frequency. So let's look first at leak frequency. This is the naive animal, and that is the proficient animal. So for the naive animal, the animal starts leaking. That's how they start the trial, by the way. They have to leak to start the trial. And then there's not much of a difference here. In fact, this is not statistically significant when the odor is given. This is the odor period. If you look over here to the proficient animal, the animal starts leaking, the odor is given, and for the S plus, the rewarded odor, they keep leaking. In fact, they increase the rate of leak, and then they get the water here. So, and they increase further. And if the odor is the unrewarded odor, they just stop leaking. So very quickly, they make a decision there. Now, what happens to the calcium? What happens with calcium is that it's not significantly different between S plus and S minus when the odor is given and the animal is naive, but it becomes very different when the animal becomes proficient. So the calcium goes up when the rewarded odor is given, goes down when the unrewarded odor is given. And this is showing the same thing, but per trial, for each trial. And we did this, that was in one animal. And we did this in 5 different animals. And you see the results for the different animals. This is % correct. So this is how well the animal is doing over there. And what you can see is that for the S plus, they increase, the calcium change increases, and it becomes significant. And for the S minus, it does not. So that raised the question whether this would encode for which odor we were giving the animal. Could we, from the activity of all of the neurons in the field of view, differentiate between the two stimuli? This is shown here, and it's shown at the top. So this is basically the coding accuracy. It's done with a linear deconvolution algorithm. So basically, we give the algorithm all of the inputs, all of the calcium at each time point, except for one. And then we ask, can we predict the one that's left out, placing a hyperplane in the n-dimensional space. So if this is the S plus and that's the S minus, we put a hyperplane in the middle. And what's shown here is this is the naive animal. This is again when the odor is given, that's when the reward is given. So the black one is odor, the red one is when the water is given. And when the naive animal comes in, the decoding accuracy is basically 50%. This area that's shaded, by the way, is just a shuffled area. So we cannot differentiate from shuffled. And then for the naive animal, when they do get the water, they of course make a decision. And from the calcium signal, we can actually differentiate between the two stimuli. This is very different when the animal actually learns. So this is the proficient animal. And you see that basically it's about 50% as it comes in, then the odor comes in, and then very quickly this rises above the shaded area, the shuffled. So there's a large difference we can actually decode, and this is shown on the right side, by the way. I'm going to move this out. Okay. On the right side, this is when the animal is learning, the naive animal. There's very difference. Well, there's actually no difference between odorant and shuffled. And when they are proficient, there's a big difference between odorant and shuffled. And of course, there's a difference at reward as well. So this was interesting, and it led us to look at the at what was there's a lot of different inputs to the cerebellum. There's sensory motor input, and one of the inputs that's been studied quite well is lick, and that animal is licking. So could this be just a motor reflecting the licking? And to look at this, we looked at 3 different phases: pre-odor, before the odor. We looked at the phase where we have the odor in there, and then we looked at the reinforcement area. So remember, during the odor, there's dry licking. They have to lick, but there's no water there. In fact, it's water with sugar, by the way. And then during the reinforcement, there is wet licking where they do lick and they get the water and the reward. And this shows the delta F over F, and this shows the lick frequency. This is for one example. In this example, the lick frequency just increased slightly, but it didn't increase too much. And what we look at is there a correlation for the slope of lick frequency? So the slope of the lick frequency here, in this case, and the slope of delta F over F, so the slope of the calcium change. And what we found is that this is shown for one animal here. There's very little correlation during the odor and pre-odor, but there's a correlation for reinforcement. So the lick is actually quite correlated during reinforcement. And this is now the correlation coefficient, showing that for odor, it's basically around 0. And for reinforcement, it's pretty, it's much higher. Then what we did is, of course, it's not just licks that are the input to the cerebellum. It's very a large number of motor and sensory inputs. And in this case, we looked at a subset of these inputs. So we did a generalized linear model fit to each of the calcium changes to try to see if we could predict. This is an example for a hit. That's an example for a miss. That's an example for a correct rejection. Could we predict the change in delta F over F from all of these inputs? And what were the inputs that we looked at? We looked at kinematics. We actually were doing a video of the mouse. We looked at the lick rate. We actually were detecting with a touch sensor the licks. We looked at velocity, acceleration, lick rate, derivative of lick rate. We looked at history also, the reinforcement history, the accuracy of the response, percent correct. And we also looked at the presence of the odor. And the black lines here show the fits for these examples. And so what we wanted to ask was, what percent of the variance can these different variables predict? So when we did this with the GLM, and we did it for all the animals, you see the results here. So it's basically the generalized linear model. And this is the kinematics. So this is, in fact, we put them all together. They each do about the same thing. They're not very different from pre-odor to odor to outcome. They contribute for about 30% of the variance. So they do about the same thing. If you look at odors or licks, then it's totally different, and different in a really interesting way. So in terms of odors, they do not contribute at all before the odor. They do not contribute at all for outcomes for the time when they get the reward. And they contribute about 80% when the odor is there. So it makes a large contribution to that change in calcium. In large contrast with the licks, it does contribute about 30% before the odor. When the odor comes in, it goes down, and then the licks actually contribute quite a bit during reward. So what this indicates, which is really interesting, is that these MLIs are actually responding to different things at different time points, which makes a lot of sense. Now, a question that came up is this something that is this a response to an odor, banana, or is this a response to the stimulus that's rewarded? And what we did here is we reversed the reward. So we had one odor being rewarded. In that case, it was banana. It was isoamyl acetate, fake banana. And then mineral oil is the unrewarded odor. So that's basically no scent. There's a little bit of a scent in mineral oil. And what we do is, this is percent correct. We reverse that here. And of course, the animals make a mistake. They go backwards, and then they learn again. And what we found was really interesting. Basically, what we found was, this is in 3 animals, is that the delta F over F, as I had shown before, is increased for the isoamyl acetate when it's a hit, when that's the rewarded odor. And it's a small change for the mineral oil. This is the change during odor. And it goes backwards. So this is a contextual, a response to the contextual odor. Is this odor rewarded or not? Now, a question that was important was to look at whether this really was licks or not. Even though we had done the GLM, we needed to be more careful about this. And this is work from Gaffield and Christie. And what they had done, this is done in Cruise 2, and Cruise 2 is well known to respond to licks. And what they had done is they had actually changed the reward. So they changed it from 8 microliters to 4 to 0. And what they then look at is the peak of the lick rate versus the peak of the change in fluorescence. And what they see is that there's a linear relationship between those two, because this is a response to the licks. That makes a lot of sense. So we did the same experiment, but in this experiment, on purpose, we did not reward one of the odors. We rewarded both odors 70% of the time. So this is not a go, no go task. This is a go, go task. So animal is at 50% if we just try to find out, are they responded to one versus the other one. And then we recorded the lick frequency, and we recorded the change in calcium with delta F over F. And the question was, is there a relationship between these two? And we changed the reward from 0 microliters to 32 microliters. And what you see is pretty interesting. So this is the dry lick when the odor is there. So they're just licking for what's going to happen in the future. And this is the wet lick when they get the water. And what we found is that for lick frequency, there is no relationship on reward volume during the dry licking. Makes sense. But there's a very clear linear relationship when they are licking during the wet lick, when they're getting the reward. They find out, oh, I'm getting 32, I'm getting a lot. In contrast, for the delta F over F, there was no relationship between the two of those with reward volume. So either dry or wet change in delta F over F. That indicates again that this is not just a lick response. Now the question is, these MLIs are responding, and we can actually predict which odor the mouse is being given, but what happens if we turn them off, or we actually decrease their activity? And we did this with chemogenetics. So what we did here is we used CNO, and we used DREADS, and we added HM4DI with an adenovirus. And then that HM4DI will inhibit the cells if you give them CNO. But of course, you have to do controls. And so for controls, we just injected a virus that did not have the HM4DI, and we expressed mCherry. And then for the experimental, we did HM4DI, and we did no CNO and CNO. And this is several sessions. So the animals naive first, and then they learn. And then after the next day, they learn again. So they're doing pretty well. Most of this is purple. This is mCherry with CNO, just to show that in this case, CNO is not changing the results. And the interesting thing, and this is HM4DI with no CNO, so they learn pretty well. But you see that there's a large change when we put CNO and HM4DI. So when we inhibit those MLIs, they have a hard time becoming proficient. And you can see that as an average here. Now, the cool thing, though, is that when we looked at the time course, it's shown here. So this is the frequency of licking. So this is the licking of the mouse. So they first lick at about 4 hertz, and then they come in, they have to lick a little bit to start the trial, and then if they get the rewarded odor, they lick a lot, and if they get unrewarded, they stop licking. And that decision is made fairly early in the trial, and this is shown here. And the interesting thing is that this is the control. So in the control, no matter whether we put CNO or not, there was a fast decision making. But with HM4DI, there was a much slower decision making. The dark blue is the one that's the one with the CNO and HM4DI. And in fact, there was a decision making that took several, it took about a little bit of like a second. Usually it's about 200 milliseconds. So inhibiting the MLIs, the molecular layer interneurons, led to a slowdown of that split second response. So there was a differential response of the odorants by MLI interneurons that develops as the animal learns to differentiate the odors. The MLI interneuron responses reflect the contextual identity of the odorant. It's not just the odorant. The mice are slower in the decision if we inhibit the MLIs. And then we concluded that in the vermis MLI interneurons, they encode for the valence of the odorant. Thank you very much. If there's any questions, I'll be glad to answer them.
Hey, thank you, David. That was absolutely fascinating. And we do have a question from the audience. Actually, it's from Conrad Cording. And I was wondering, Yang, maybe if Conrad may want to come on stage and actually ask that question himself, because we do have the time today. We don't have a time crunch like in other sessions. So if Conrad is willing, he can, because he may have a follow-up question to his question.
Hi, Conrad. You're muted.
Yeah, he is muted. Oops.
Oh.
All right. While he's trying to get online, let me ask you what he was asking.
Sure.
He said, I am curious why the calcium signals look so massively correlated. When I look at spike trains, that usually looks very different.
I was really curious about that, and I do not understand it. In fact, we computed dimensionality, and the dimensionality was pretty low. And in fact, when we started doing that decoding with subsets of neurons, we could actually predict with a small number of neurons. Now, why is that the case? Is that because this is the because of this task? So let me tell you. So the answer, of course, is I have no idea. Okay, but let me tell you what I think it is, and why we're doing something very different right now. That's a very simple task, the go no go task, where you're hitting that ball, and this is the one thing I'm doing right now. And so I think what happens is in that case, dimensionality goes way down because you're really interested in that. So what we're doing now, we're not doing that in the cerebellum right now. We're doing that in the olfactory bulb, but we're interested in doing it in the cerebellum. Is more complicated tasks. So the one that we want to do for the cerebellum, where I bet that dimensionality is going to go way up, and this is just me saying something, I have no idea if that's correct, is following an odor plume. Odor plume navigation is extremely different, very different, very difficult, because what happens is there's a turbulence, so the physics are different. And I bet in that case that we're going to have dimensionality that comes right up, and you still make decisions. But that's me just making up, to tell you the truth. I have no idea about that. So I bet that depends on the task.
Yeah, and just picking up on your point about the picking up on the plumes, and there has to be a really good dynamic response in the system to be able to track that and to be able to get that. Mm-hmm.
Yeah.
So you're thinking that the circuit, so the information goes from the olfactory bulb straight to the cerebellar circuits.
So the questions were yes. Mm-hmm. Yeah.
Okay, and that's my question. And so then the question is, it kind of leads into the question I had, it's a follow-up question about them. Actually, let me check. There's another question coming in because I may try and deviate you away from this discussion. But let me ask you. I'm going to come back to this. Let me ask you a question from Furazan Farahani. Since we're having trouble getting people on stage, have you tried to do the same experiment with a higher rate of reward while blocking MIL? And in order to know if MIL is modulating the value of reward or it has a gating role somehow.
Yeah,
So
that we did not do, but that's an interesting question, and we have not done that, but that's important.
Yeah, that's an important question. Thank you for asking that. So okay, and Conrad, okay, no, that's, yes, we've answered those. Good. No, so okay, so let me get to the question I wanted to ask you about, and that is, and I started going there because we started talking about, you know, the circuit in the brain potentially for that, and so there are studies of. Obviously, there are a lot of studies in prefrontal cortex with these kinds of tasks in many species. And so I'm going to put you on the spot. And I'm going to ask you, which is the cart and which is the horse? So of those two important axes, the cerebellar and the cortical axis, which one comes first, in your opinion?
Which one? That's a good question. There I have to again think about what I'm thinking. I bet that it's different depending on the task. Depending on how quick it is and what kind of decision you have to make, it might actually become cerebellar to supplementary motor. But very likely, you know, prefrontal, of course, is involved. And so I think it depends on exactly, you know, I come from physics, and when I came, I was totally naive about behavior. Goodness, the number of variables in behavior are amazing. And I think that what happens is that that's the case, that very likely it's different. Where the response is coming from, likely from PONS, the auto response. Noam Sobel has a beautiful paper in humans where there's cerebellar changes for odors. So clearly there's responses there.
I just resonated with your comment because I studied biophysics before I went into neuroscience, so I understand exactly what you're talking about. But just I'm interested in, because you made a comment about when you answered that question, which made me think of what you said earlier about it depends on the task, which makes a lot of sense. But I'm just wondering whether the type of task will also modulate the dimensionality of the system, which is what you were referring to earlier.
In fact, that's a really good question. And it would be very interesting to make this task a faster task. So Dima Ringberg did that years ago with the olfactory bulb. Basically with this kind of a task, and also with alternative force choice, you can have it so that the animal can respond ASAP versus the animal has to lick for, say, 5 seconds, something like that. Things may be very different depending on what you're doing, and also changing the reward amount. So one and the other.
Yeah. And the other thing, when you were talking about your experiment in the mouse, it made me think of two fascinating studies in macaque monkeys, actually, on metacognition in prefrontal cortex. And so that started me thinking along these lines. And one of those studies was by Yoshida et al., and the other one was by Middlebrooks and Summer et al. And they had two macaque monkeys, which they had trained for ages, and they had both of these studies had pairs of macaque monkeys interacting with one another and observing. So it was like a task with a rule that it was a go no go kind of situation where one animal had to do the respond while the other was watching. They both knew what the rules were, and not only did they examine the licking behavior for the reward in the monkey who was doing the task, but the monkeys took turns in doing it. The other animal had a response measured in what they were doing. So they were monitoring these errors, the partner's errors, if you will, and they were looking at activity in the prefrontal cortex. It was absolutely fascinating. So my question to you now is, what would happen if you had two mice, if it was possible to even train and have a mouse who was watching the conspecific doing the task, if they had both learned it? What do you think would happen in an instance like that?
That's interesting. So one thing is that I don't know, and it would be interesting to see what happens. It would be a little bit of a difficult experiment because they're really good at smell. So you would have to do it in 2 different places, which is fine. I do not know. Did the monkeys get rewarded depending on what the other one would do?
Well, they got the same. So if one got it wrong, the other one didn't get the reward either.
Excellent. That sounds like a cool experiment with the mice. I do not know. I bet that it would depend on which sensory inputs you give the mouse. Even auditory, they can hear 20,000 hertz to 40,000 hertz. Could be really, really interesting. So actually, it's a really cool experiment. Cool.
Well, I'll keep watching for your publications for when you actually do that experiment, because I don't know. I'm a human experimenter. I don't understand much about the many differences between the brains of say monkeys versus mice, and so I don't even know if that would be an appropriate experiment to do, because I don't know much. I know more about monkey prefrontal cortex, but I don't know if that would be those.
Yeah, what you're saying makes a lot of sense. There's a big controversy about monkey versus mouse. And there's some argument that initially that, they're just totally different. But then Schoenbaum came and showed many things that happen in prefrontal. And there's an argument that, but they're different, but they are doing something alike. So I don't know. Of course, we need to do experiments in different species.
Exactly. No, and that just underscores that we do that. Well, let me ask you then this, and this is again because it reflects my ignorance. Would that be an experiment that would be more likely to succeed in a rat?
So first of all, I'm more ignorant about. And my current thought is that the rat would be very different and interestingly different. But that's just the reason I started working with mice is because they can recognize each other by smell. They can recognize the MHC, 2 amino acids in the MHC, and the mouse, she will mate with the one that's different. Rats are predators of mice. They're very different animals. In fact, right now I'm interested in some ethologically relevant work. I don't know. Even if you try a wild mouse. So at some point we started some work with wild mice. They're totally different to a mouse. That for sure would be extremely, extremely different because these are congenic, we've had them in the lab for 200 years or so. So that's not a mouse. It's a very different mouse.
So talk a little bit about what makes a wild mouse different. Is it their repertoire of odorants that they would be sensitive to? What would be the main differences that you would see between them?
So their environment is totally different, and they have experience with predators. So I think that it's a totally different animal. So I bet that the brain development is different. It's both brain and plasticity, development and plasticity. We didn't do a lot of work with the wild mice. We did some work looking at the anatomy. And the bulbs were different anatomically. So I would expect there's a difference in development.
So the bulbs were more, the bulbs were much more, I would imagine, differentiated in the wild type? Would that be the case?
It was just gross anatomy. So they were different in terms of gross anatomy. So I don't know much about exactly what the molecular differences were. But that would be really interesting. You know, what's the receptors? What receptors are expressed, and what's the circuit like?
Right. Right. Yeah. And I wonder to what extent this go-no-go cerebellar kind of mechanism is also influenced by one's ability to respond to threat in the environment, because obviously what you were talking about with wild-type mice and their exposure to predators, I mean, there's going to be they're going to be the ones that are going to be freezing in the environment when there's a predator. And so I wonder to what extent there's an interaction between, you know, those threat circuits and the kind of go-no-go circuitry that you're talking about there. And I have no idea what I'm talking about. This is just something that I'm imagining.
That's a really interesting question. I would expect in the wild type mouse, we may see a very different behavior in the ones that survive. The ones that don't survive with the predator may have a freeze. And these other ones may escape, and that likely is some, I'm making it up, you know, some plastic mechanism, some plastic circuit.
So where would you see that circuit would be? Would it be mainly?
So amygdala should be involved. I'm playing with amygdala lately, so BLA for sure, a prefrontal BLA, you know. But that's again making it up.
No, and again, well, okay, that's okay because we're just speculating here. Does the BLA send connections to the cerebellum as well, and vice versa, that you know of?
Everything, I mean, most of it will come from pons, so it'll be not direct but indirect.
Sure.
And there is work on prefrontal, and I forget. I think Mimi and Luo was part of that, but I may be wrong. So yes, there is input to pons. So it would be input to pons. And I think that there's also amygdala to pons, I'm almost sure.
Oh, that's interesting.
Yeah.
That's interesting. Yeah, that's something that we haven't really explored even in the human brain. I mean, we're only starting to think about, you know, cerebellar circuits and also amygdala circuits in the sense that people are starting to break down, particularly the amygdala, into its different nuclei, and which is, I think, really healthy because I think that's keeping it as a unitary structure has been such a mistake for a long period of time. But I think that I find your work particularly exciting because it shows how the cerebellum can really influence highly sophisticated behavior. And that's something that people haven't appreciated. And, you know, the sort of work that you described today, which was really, really elegant, makes you think of frontal cortex, the kinds of the data that you showed. If I was blinded to what you were showing and I didn't know what brain structure it was, I would have said you were in prefrontal cortex.
I agree. I was very surprised. When we started doing this, as I say, we were just going to Isabel's lab and we thought we should do some recording before we go there. And goodness, it was fascinating.
So do you anticipate then doing, and I know this is difficult because this is a mouse, but is it possible to do recordings at the same time from, say, frontal cortex and from the cerebellum, do you think?
We've been doing some in hippocampus and frontal cortex. But yes, so in the future, I'm interested in that kind of a thing. How do these places talk to each other? And, you know, which one is faster? And could it be that one is faster in one condition and slower in the other condition?
Yeah, and also as a function of task, because we were talking about that earlier, right? That would be really, really fascinating to see what kind of differences. What would you predict if you had a crystal ball with the kinds of tasks, and if you could record from the two structures, what kind of differences would you expect to see? Have you an idea?
It will not be, I bet so there's something else that makes it very difficult, which is if it were with a more maybe outbred population or something like that, you're going to see differences between individuals. And they might actually correlate with which area is responding faster. even in the simple task. But that's me making it up. Which goes back to your question of, if you're in the wild, what do you do when there's a predator? Do you freeze? And a subset, we're going to do that, and a subset of them are going to jump out.
Right. Right. Well, it sounds to me like you have probably about 50 years work with this stuff. And you could do you're going to do some amazingly interesting studies and make us think about how important these cerebellar circuits are. And I would just like to thank you for such a stimulating talk and for putting up with my ignorant questions.
Thank you, Aina. And my questions are more ignorant, but that's what I tell my students. When they tell me something like that, that's the question I want. I had one student that did that, my first student, who solved something that was, it was beautiful, a beautiful PhD thesis from a quote unquote ignorant question.
Yeah, and that's what we've said here at NeuroMatch. There is no such thing as a stupid question.
That's correct.
That's what we've said, and people have been very, very shy at this meeting about asking questions, which is unfortunate. But that is the case.
Yeah.
And Farazan Farahani says that there are similarities here, and visual-based decision-making are interesting. There are inhibitory inputs from basal ganglia that are really important to the superior colliculus. So there's another parallel there for you.
Yeah. The visual system decisions, I've always looked at those as well. Yeah, that's interesting.
Okay. Well, I think what we'll do is we'll wrap it up. And thank you for such an illuminating talk today.
Thanks for the questions.
Okay. And everybody, have a good rest of the day.
Bye-bye.
Bye-bye.
At Brandeis University. And today I'm going to be moderating together with Kevin this seminar. And I'm very, very excited about this session because it's all about open source tools to facilitate our neuroscience research. And, since even though we all work on different problems and different organisms, we all face similar problems in terms of data analysis and neural simulations. So it's nice when there are open source tools available that we all can use. And with that, I would like to introduce you to our first speaker, who is Luigi Pitruca. And he's a PhD student in Portuguese lab at Max Planck Institute of Neurobiology in München. And he's developing Brain Globe, a Python-based software for the analysis and visualization of neuroanatomical data across species. And now he's going to tell us more about this tool. So please, Luigi, go ahead and you can share your screen.
Okay, everything's fine?
Yes.
Okay, so hello everyone, I'm Luigi, and then we talk about Brain Globe, a Python ecosystem for computational neuroanatomy. We all know very well that neuroscience is moving more and more toward the direction of collecting data sets that are bigger and bigger, and recent advancement and techniques allow us to really address neuroscientific questions at the whole brain scale, like from the whole brain connectomics and transcriptomic data that the Allen Institute is collecting to the whole brain imaging that we can perform in fish and even in mouse using the recent ultrasound imaging techniques. We're getting in a direction where we will need to make sense of more and more of those data at the entire brain level. And of course, we will need new software for analyzing this new kind of data that we are about to work with. And actually, in recent years, there are many packages that have been coming out to actually perform analysis and visualization and quantifications on such kind of data sets. A limitation of the software that has been developed so far is that most of those softwares have been developed with one single atlas or one single species or one single annotation of one single species in mind. And the most common one, of course, has been the Allen Brain Atlas. And actually, our story starts from one of those tools, which is Brain Render, which is a fantastic software that Federico Claudi developed at in London, which is a software for making very nice three-dimensional visualizations of different kind of data, from gene expression to connectivity data to electrophysiology data, using very simple Python scripts. And I saw this and it looked amazing, but you know, I work with fish, so I had little usage for something that was working with the Allen SDK for mouse. And luckily at the same time, Federico Claudi was actually reaching out in the community, wondering whether people might be interested in using his software outside just the mouse community. And I just saw this tweet and it looked amazing. And I was also already in contact with Adam Tyson, which is the third person we started this with. And so we decided to join forces and start to work together also on something that would make our software to work across different species and atlases. So we started looking around, and of course we found a whole bunch of atlases that like laboratories that are expert in neuroanatomy have been developed in the years. And basically every animal model that is common use in neuroscientific research has one or in most cases multiple annotations that are available. The problem is that many of those API, like those atlases, have interfaces that are web only. So you can go to the website of the atlas. You can search through the areas that describe. Sometimes they also serve data, like there is a zebrafish atlas that also serve single cell tracing data or transgenic line expression data. But those are web only. On the other side, there are tools that make the data available locally, like the Allen SDK package, which is great, but it's a very big package that serves both the atlas generation and also a lot of project-specific data that makes the Python package much larger than this specific usage. Which means that in general, this step, like if want to really generate atlas agnostic software that might work with different atlases for different animal models, one that is not really provided with one single interface for doing this. And we actually saw that, like, we are Python programmers, but we saw that in the R community, there actually something along those lines has been already developed, which is the NatVerse package, especially from the connectomics community. But we were missing a similar concept in Python, and we decided to come up with our solution, which was like in general an umbrella term to the standards that we're using. We like to call ourselves the Brain Globe organization, and in particular, we produce this Brain Globe Atlas API, which is a minimal, lightweight module with very little dependencies to use reference spaces in whatever Python package or you might be developed or your analysis pipelines. And the basic idea of distribution of atlases in Brain Globe is that there's us, the Brain Globe developers, who scratch data from different sources, which might be the LMOS brain reference, or atlases that have been developed for their online their web tools. We are interacting also with the people who actually generate those atlases, and we put those atlases in a standard format that then can be very easily accessed by the final Brain Globe users using a single class that is just instantiated with specifying different atlas names. How does it work? Well, installation is as simple as it can get in Python. And the usage is very simple. There is, for example, the shows atlases function that tell us what data do we have locally, what data is available for download. Actually, if we don't have an atlas, the moment we instantiate the atlas class, it will download it for us. If we have it already locally, it just generates the object as it happened now when I just generate it. And this Brain Globe Atlas object contains a full description of the atlas with the metadata, the source of the publication, the hierarchy that is comprehended in that atlas, and most importantly, the stacks. So the reference brain that you can use for aligning your data to an annotation and an annotation stack that contains the information of voxel-wise information about the brain regions. And then also pre-computed structure meshes that people can use for their own three-dimensional visualizations. So I go through a little demo of the class. So here we're just generating a Brain Globe Atlas of the Allen mouse brain at 100 micrometers resolution. Just printing it produce all the information about this atlas. And then we can very easily access the structures hierarchy that is described in this atlas. So the structures property will just print for us, like printing it will just print for us the full hierarchy of the brain regions, and we can query it as we would do with a dictionary to get information about, for example, visual cortex. And it will print that for us is a little dictionary that we can use. The very important part is the reference stacks. So reference and annotation stacks are just properties of the Brain Globe Atlas object. So we can very easily fetch them. And at this point, they're just NumPy arrays that we might want to plot. For example, here I'm just plotting a slice in the middle of the stack. And if I now look at the annotation, this is just providing me the same image with a number that corresponds to each brain region that is comprehended in this stack. There's also methods for already automatically querying the atlas, for example, asking which structure is located at some given coordinates, and there's also utilities for example, generating the mask for a specific region. So let's assume we want a mask that has ones where the cortex is and zeros where and zeros outside. We can just use the get structure mask method that create that stack for us. And if we now plot it at the same level, at the same slice, we can see that now we have an image of just the cortex of the in the atlas at that point. And we have been developing it, but also already implementing it in the tools that I started all of this for, for finally getting to use. And this actually produced now allow me to perform the same kind of visualization that Fede was able to do for mice, but now for fish, which I like much more. And with Adam, we have been also implementing, we have also adding Brain Globe multi-atlas support in his program SelfFinder, which is a great software for performing neural network-based detection of neurons labeled in whole brain datasets. So you can see here a very brief overview of the software where the image is analyzed in a way that finds all valid neurons that are in the image. It does it on in whole brain datasets and allows them to visualize them also using, in this case, brain render. And using Brain Globe Atlas API, SelfFinder user can, well, at the moment they can, for example, compare the results in a different annotation from the Allen mouse brain, which is the Kim-enhanced atlas, where some regions are annotated with finer resolution. But one could also think of using the software for performing the same analysis on even different species. So this is leading us to the concept of a full ecosystem of applications that are all interacting with the this core Brain Globe Atlas API package, and so they kind of use the same standards and formats and are compatible with each other. So apart from Brain Render and Filer, there is Brain Reg, which is a package for for whole brain image registration. There's also a Napari plugin.
Luigi.
There's also a Napari plugin that can be used for both exploring the atlases and having a look at the results of the registrations that has been done with BrainReg. So we just came out with version 1.0, but of course, like we are at an early stage of the project. So we are really looking for the feedback from potential users. In particular, if you would like to use some specific atlas in your Python analysis, or on the other side, if your lab has an atlas that might be useful to share, but you don't have the tools for developing a web application, or like you don't want to take care of the distributions yourself, just reach out. We are very happy to include more datasets in our project. And we are also looking forward for people interested in integrating it with their own software for whatever kind of analysis. But of course, also in just like the scripts that people use for their internal analysis, BrainGlobe can definitely be interesting things to check out. You can find us on Twitter, on GitHub, so reach out. With this, I would like to thank all the new contributors to the project and the API. So Federico and Adam, with whom we started the project, and also Nick Del Grosso, who recently joined and will sure help us improving it even more, our supervisors, our hosting institutions, and of course, you for your attention.
All right, thank you very much, Luigi. Very useful and very intuitive tool that you just described to us. And we have time for a few questions. So one question we have is from Edwin. Is it possible to generate cuts at arbitrary angles for the purposes of mapping experimental samples?
So there's actually an ongoing project that well, I'm not following, like Adam and Nick are involved to this for exactly looking, like going in the direction of generating an interface for registration of slices at different angles. At the moment, this is not supported. You would need to use your own tools for like cutting the stack. But in the future, hopefully, we will get there.
Okay, thank you. We actually have one more question from Thomas, but we are out of time. So if you have time, Luigi, please check it in Q&A box, and maybe you can answer to him in the chat. So, and we are going to move to our next speaker, who is Marcel Stimberg. He is a software research engineer in Sorbonne University in the Romain Bread Lab. And he is developing open source tools for neural simulations. You might be familiar with the Brain Simulator, and tools for data analysis and spike sorting, such as Spike in Circus. And today he's going to talk about automatic model description in neural network simulators developed to minimize the errors and typos in the model descriptions for publications. And also, Marcel encourages you to share codes with the community because sharing is caring. So with that, Marcel, please go ahead.
Okay, thank you very much. You can see my screen well, I hope.
Yes.
Okay. So yes, I'm happy to be here and talk today about our work on automatizing model descriptions in a neural simulator. And this work has been done mostly by a talented young student, Vinesh Varan, who did a Google Summer of Code project this summer. So what do I mean when I talk about model descriptions? So all of us who work on computational models, we are not only going to share the code, which I would encourage you to do, but in many situations, we also have to share information about our model with collaborators, colleagues, that don't know our simulation language, that don't know the programming language, maybe even.
And we might use prose or equations, etc., to share with them. And of course, in a more formal way, we are also going to describe our model in scientific publications, for example, in the material and method section, or in the supplementary information, again, using text, equations, and maybe a table for the values that we used. And this is a manual process, and manual processes are error prone. And I think all of us who work on these kind of models, who have tried to reproduce other people's model, already ran into some of these kind of problems. So here's one example. Imagine we have a code for the neuron simulator where we're setting the conductance, GKA conductance to the value 0.0031. And in our paper, we described this as being the value of 3.1 microsiemens per micrometer squared. And it turns out we did a conversion error because it's actually supposed to be 3.1 millisiemens over square centimeters. And these problems, of course, happens all the time. And it could be conversion errors for units, but of course it could also be a simple typo, moving the decimal point to wrong place, etc. And even in simulators like the Brine simulators, where we have a system for units, so where this kind of conversion error cannot happen, we could run into the following situation where we have the value of a parameter of our model in some place in the code, and we report this in our model, but if we forget about that later in the code, actually, we are going to normalize these weight by the number, a total number of neurons, a total number of synapses, so we actually make a mistake. And finally, there's a third kind of error that we can make is simply when we have a complex model, there might be several options, several code paths that we can take. We have flags, we have parameters that change the way simulation behaves. And we might just make an error thinking that our code took one of the paths where it actually took another path. So all these arise because we separate the simulation itself from the way we describe the simulation. It's a manual process mostly, of course, involving a lot of copy and paste in many cases. But the solution, or part of the solution that I want to propose here is to generate these kind of descriptions automatically or semi-automatically, and in particular interpret things in the same way as the simulator would. So have this be handled by the simulator, so there can be no ambiguity. The simulator knows how it interprets the code. And so just to mention quickly a few existing approaches that go a bit into this direction. So there's a website called opensource.brain that collects models for the NeurML model description languages, and it has a web interface that allows you to look a bit in the detail of models, mostly multi-compartmental models. There's also ModelDB that I guess many people of you know, which has a lot of models for various simulators, but in particular for the neuron simulator. And for models for the neuron simulator, there's an add-on called ModelView that also allows you on the website interactively have a look at the details of the model, see where ion channels are located in the neuron and so on. And finally, there's a simulator that I feel is a bit underappreciated. Not many people know about it, the Anarchy simulator. And it has a model generation framework, which is actually very similar to what I'm going to present today. But the simulator doesn't have a system for physical units, so this kind of problem can still occur. Okay, with that, I want to come to the Brine simulator, which is a simulator I've been working on in the last years, which has been originally created by Romain Brett and Dan Goodman. And for those of you that don't know that simulator, just a quick idea of what is a bit special, or what is the approach the simulator uses. So on the left, you see it uses to describe models, it uses mathematical language. You write down the equations of your models as a textual string. And these equations and also the setting of parameters and so on uses physical units. You express things in volt, millivolt, etc. And the brine simulator takes care of raising an error if something doesn't match up. You cannot add up two different quantities with different units, for example, etc. And from this like high-level mathematical model description, then the brine simulator generates low-level code, so for example, C++ code, to efficiently execute the model. And so for this simulator, what we set out over the summer was to create automatic model description framework. And as I said in the beginning, that was done by Aneshwaran, who is a student from Chennai, India, who has been working at this over the summer as a Google Summer of Code project with a mentoring organization, INCF. And this has now been integrated into the Brine 2 Tools package, where we hopefully do a release soon that includes all this work. If you are interested in like a first-hand report about his experience in the project, he wrote a little blog, reporting his progress. Okay, without further ado, I'm going to show a little example of what this can look like in practice. So this is a brine simulation. Without going into the details, there's some parameters, constants defined up here, which uses a system of physical units. Then we describe the neural model using equations, and because in this case it's a type of integrate and fire model, we also have a threshold condition and a reset and a refractory period. And then we have some initial value of the membrane potential that is also given in kind of equation form here with a random number. And finally, we define synapses, which in this case are current-based synapses, where for each incoming spikes, the post-synaptic current in the neuron equation jumps up, and we connect things up randomly with a certain probability. So this is the kind of like Brunel type network that people use to analyze the dynamic and randomly connected networks. You see like asynchronous or synchronous activity and so on. And so with this new model description framework, this simulation can be directly translated into a description, which you generate in Markdown, and Markdown then can be translated to HTML to be included on a web page, or it could be converted to a PDF, for example. And it looks like something like this. It tells there's a group of 4,000 neurons with a certain equations governing its dynamics with constants and their units, and the equation that we're using for the initial values of the membrane potential. And similar description also for the synapses, where it's at what it actually connects to each other, with what kind of connection rule, or in this case it's probabilistic, what happens for an incoming spike, and what are the parameters of the model. And of course, if this were a more complex synapse model, like for example, a synapse model with plasticity, SCDP or something like this, then this description would of course also look much more complex with equations and so on. Okay, so how does this work? First of all, how can you use this as a user? So this is again our example that we've seen earlier, just shortened a bit for all the constants that we defined in the beginning. And so this is the code you use to run your model. And now there's only a very small addition needed to, instead of running a model, create a description from the model. You import this Brine 2 Tools package, and you tell it, instead of to generate code to run the model, to generate a markdown description of the model, in this case to a certain file. And internally, how does it work? So it's actually a two-step procedure. So what happens when you run this is that from the simulation code, it first generates a kind of intermediate representation, a dictionary structure, or you can think of it as a like JSON description of the network, which is pretty straightforward and easy to understand, even if you don't know much about Brine. And it abstracts away all the details about in what kind of Brine object things are stored, what are the names of the attributes, how to extract all the information from the Brine code. So this is something that you can then quite easily use to convert the representation into something else. And what I was mentioning here is transforming it into Markdown. And maybe to briefly mention it here, we've written this in a way that it is easily customizable.
2 minutes.
Yes, thank you.
You have 2 minutes.
Customizable. So you can inherit from the class we created and override certain methods to change the details of how it looks like. But it can, the same intermediate representation can also be used to export to different formats. For example, we have a work in progress export to Neuron. So just quickly to mention some limitations of the current code. There's no support for multi-compartmental methods yet. We don't well handle multiple simulation runs where you run the same simulation, for example, say with an input current and without a current. Currently, the description would just repeat the model description over and over. And also now you have to decide whether to run the model or to describe it. Ideally, we would like to have the same at the same time to avoid some mismatches between the two. And what are our plans for the future? So it's, first of all, it's to give the user more options to annotate the model manually with comments, manual human readable descriptions that then can be included in the Markdown output. And also to annotate the model automatically, for example, to determine that some equations are stochastic, or that we have current-based synapses or conductance-based synapses, etc. With that, I want to conclude. So we now have this Brian to Tools package that will have a new release soon, which will allow you to give this automatic model description. And we think it's can be a useful tool to model sharing and to avoid errors in manual model descriptions. And with that, I thank everyone involved, in particular Vinesh, who has been really writing all of the code I was talking about. And as a final comment, as Katja mentioned in the beginning, even though this is nice for model sharing, please don't forget to share your code. That still remains the most important thing. Thank you.
Okay, thank you very much, Marcel. I think it's a wonderful idea and definitely should make life of computational neuroscientists much easier, because I myself ran into some issues while trying to reproduce the results of the model from the paper and couldn't because there was some typos. So I think it's fantastic. And maybe we have time for one question. So I don't see any questions yet in Q&A box, but please ask questions. So maybe I'll ask you a quick question then. So I was wondering if this tool is now only available for the Brian simulator, or it could be adapted for, you know, like in-home code or like for other simulators.
Yeah, at the moment it's only available for the Brian simulator, and there are some things that are very specific to it. I mean, in general, what would be an option for other simulators, and what some other simulators are using, is to use some like intermediate steps. So for example, converting things to NeuroML, for example, which is now supported by several simulators. And then you can build a similar framework on top of NeuroML, for example, that would then benefit other simulators as well. One reason why we didn't do this here is that from the point of view of Brian, if we convert to NeuroML, we lose some kind of information. So it's a bit hard to describe, but in a way, like information about the intent, what the user wanted to do. What NeuroML does well is to accurately represent the mathematics, really the details of the model, so you can simulate it exactly in the same way. But sometimes it's hard to go back to what the user asked to do. So for example, it's something like a absolute refractory period, which is a simple thing to say and state like this, actually translates in a number of equations and if statements. And that would be a bit of a pity if the model description then looked more complex than necessary. So that's why we didn't go this way.
Okay, I see. Well, thank you very much again, Marcel. And you can find information about the Brian on the Twitter and as well on the website. So some of the links are in the chat. And with that, we are going to move to our last speaker of this session, who is Johnny Sanders, and they are a graduate student in Mike Ware Lab at the University of Oregon. So they developed Autopilot, which is a software for behavioral experiments that allows to efficiently combine arbitrary number of hardware components to be coordinated in the experiment, and it will allow for more complex and reproducible experiments. And you can you'll be able to find information about it on the website, and the link is going to be in the chat and in the preprint and by archive. So with that, Johnny, please welcome and go ahead.
Thanks y'all for being here. Thank you, Kaya, from I apologize, I'm late. It turns out I completely misunderstood the structure of the conference.
You're late for your talk, so it's good.
Yes, at least there is that. So I'm also a Zoom novice, so allow me to share my screen real quick. Okay, so thank you all for being here, coming to you live from the illustrious basement lab, where I've been for the last 7 months, so going for the most uncultivated Zoom backdrop of the conference. So today we're going to give a little bit of less of a technical talk about how the system works and more of a sort of like design talk about what is the tool like, why is it like this, and why do we need something like this. So to do that, I'm going to tell a little bit of a story. And this is a story of how would we go about studying a behavior like this. We observe that mice bob their heads to judge a distance before making a jump. And so pretty straightforward behavior. What kind of tools would a neuroscientist need to study this? So one, straightforward, let's just study it open loop. We're just getting off the ground. We're just getting invested in this project. So we're just going to hook up 2 high-speed cameras, write some simple script for it that captures the images. And then later on, we're going to do some deep lab cut analysis just to like sort of quantify the behavior. First of all, simple enough. 2 components, 1 computer, great. Next thing we want to do is be able to control the platform that the mouse is jumping onto. So we go over to the machine shop, ask them to build something that's a motorized platform, and they come back to us with something with this beautiful behavior, but a very esoteric and complicated way of controlling it. So we need to write some custom firmware, but our computer doesn't have a serial port, so we need to use a little microcontroller anyway. And so we have to write some firmware for it in C, and then write some special serial protocol to communicate with that microcontroller. So okay, the complexity not too bad, 2 computers and 1 controller, but we're not quite at where we want to be at. Where we want to be, closing the loop. So in order to close the loop, we have this motorized platform. We're studying this bobbing behavior. So we want the platform to counteract the bob of the mouse. And so in order to do that, we have to get the motion data from the mouse really quickly. So we need to gather data from a motion sensor that we've implanted on the mouse. We also, while we're at it, want to gather video from the mouse's point of view and some pupillometry while we're at it. Neuroscientists, we just need to maximize the data streams. So, but our first computer is already maxed out. We have 2 high-speed cameras going on it. We're controlling another microcontroller, and just the computer is too sluggish to handle 2 more video streams and another motion sensor. So let's get another computer in here. So the second computer, but the problem is the second computer. We haven't thought about how to do the synchronization. So we come up with what neuroscientists usually do, is make some sort of synchronizing TTL pulse that flashes an LED in our camera so that we can see when that is happening in our video, so that we can synchronize it later. Unfortunately, that doesn't let us do real-time image processing as easily as we'd like to, because it needs to be aligned after the fact. So we had to compromise on some of our experiments. But at this point, we have a working closed loop experiment. And I'm going to skip a lot of the details here. And so on it goes, where we also want to gather electrophysiological data. We also want to control another set of solenoids to deliver rewards, etc. So the complexity only compounds when we get into the full experiment. And what about the data that comes from an experiment like this? So we have, 10 or so different types of data. They've been gathered at different times in the experiment as we developed it. They're all on different computers. They might be in different formats. We need to write code to synchronize our video that's been taken. God knows what the organization is like, if there is any metadata at all, and whether we've preserved information about the animal's training history as they've been going through the different iterations of the task, all up to question. So basically need to write another library just to clean the data. So, and then we haven't even talked about the entire infrastructure for writing the task in the first place. So is this some script that we wrote from scratch? If we were using some prepackaged behavioral library, how does that integrate with the rest of the tools? We have multiple computers. Are we walking around the lab to start all these scripts at the same time? Or if we're very fancy, we have some bash script that starts it all for us. And that also ignores sort of any training or shaping that needs to go into it. The mice don't just jump up on the platforms themselves. We have to sort of train and shape this. So did we just write multiple scripts? So I think that using this as an example, thinking about what the next 10 years of neuroscience experiments are going to be like, where this is just one example of a way that a neuroscientist would typically tackle a project that they want to do. But I think it's indicative of the larger structure of these type of experiments. So lots of hardware, lots of heterogeneous hardware that just like doesn't know necessarily how to talk to each other. We're a mix of custom and off-the-shelf components where we don't want to do everything from scratch. We want to be able to buy cameras, for example, but also we're going to have whatever weird stuff we have lying around the lab, and we need to be able to accommodate both. We need to have all-to-all control. So that's the nature of closed loop and real-time behavior. You need to be able to take some input data stream, really easily plug it into some output and be able to control that. You need to do the real-time data processing to make that possible. So taking an image, processing the image, converting that to some useful information. And synchronization, of course, needs to be baked into the core of the library. The tasks it needs to do need to accommodate sort of like more complex process through a task. So train the animal, shape the animal. And then when we get into the high density silicon probe recordings we all know and love, and the wide scale calcium imaging, we need to be able to accommodate high bandwidth and multimodal data collection. So what does that mean for the tools that we need? We need them to be flexible. So first of all, we need to be able to use any hardware that we have and write any task that we need. So what we need is a framework rather than a program. We need something that we have a set of tools that we can build experiments with rather than something that's sort of limited off the shelf intended to do X things. So we just need to separate the tools from their implementation as a software. They need to be distributed, and there really is no getting away from this. At the point when we need this many hardware components at a time, you have to use multiple computers. And if you ignore that component and sort of treat it as an afterthought, you're going to be doing a lot of cleanup later. So it just should be distributed from the start. And I'm glad to hear that we're talking about reproducibility in this panel, because that's the other thing that we need to fight. We have these better angels within us that we want to do good science, but we also have this beautiful instinct to just hack stuff together and make the one idiosyncratic rig in the world that can do this experiment, because only I know this one pattern of resistors that seems to work sometimes. And so we need to have data that's clean, complete, and annotated at the time of acquisition to lower the barriers to sharing that data, but also an experimental framework that allows replication. And so I want to talk about what Autopilot does and sort of some examples of how we might do this experiment differently with Autopilot. Unfortunately, we don't have enough time to give an actual good description of what the system is, but in short, it's a distributed system for behavior that uses a lot of Raspberry Pis that are networked. And the basis of its design is that it's a bunch of modular components that you can sort of split out, modify, change, and use as you want to. It's a Python library for behavioral neuroscience. And so one thing that we can do initially just with the hardware, instead of needing to write a custom firmware and sort of writing our own new communication protocol, we just take existing code in the library. For example, we already have code in the library for controlling the GPIO input-output pins, and just extend that to make them behave in the sort of custom way that we want to. And but we keep all the functionality that the rest of the hardware objects has. This object already knows how to network to other things. This object already knows what it's like to make a pulse train, etc. So it makes it easier to extend, and it keeps it integrated with the system. And recent little feature I made, I've been working on recently is, if you don't like Autopilot, don't like the rest of it, but just want to use some of its hardware components, now you can just import them and use them anywhere. A lot of the back-end stuff has been abstracted away. And then when we talk about a distributed topology, this experiment becomes a lot easier when you can sort of do it out of the box, where every object can be networked. every LED can have networked messages sent to it to control its value, for example. So real-time closed-loop control is the heart of the program. So in this case, we can, because it's relatively inexpensive to implement, and because it's possible to control a bunch of these things in parallel, we can scale our N and avoid a reproducibility crisis. And we can also scale the complexity. So an experiment like this becomes—
You have 2 minutes.
Okay, thank you. I wouldn't say relatively trivial, but easier than it was. And another thing we're excited about this distributed topology is we collaborated recently with the Deep Lab Cut folks and to work with Deep Lab Cut Live. And in the paper, we talk about an experiment where we capture images on a Pi, send them to another GPU to process, and send the information back. So we think this is a promising foundation for many-to-many real-time image processing, many cameras, many GPUs, complex experiments. And this is part of a general transformation module that's easy to use, but I'm going to skip a little bit of that because I'm running out of time. And the final thing I have here on reproducibility is that because all of the components are integrated into the same system, the data can be unified at the time of its collection. So Autopilot keeps all of the data from all of your tasks. If you ever change the task, reassign an animal to a different task, it keeps track of that. Any parameters that change in your task, any version changes that happen to the code, any local changes you make to the code base, etc. We want to make sure every part of the experiment is documented so it's possible to reconstruct. And in addition to that, it comes with a single file, a portable task description, so anyone can re-implement your task exactly. So in sum, we built this tool to do the extravagantly complex experiments we all want to do. So for the future, the couple of things we want to do is, one, we want to integrate OpenEFIS acquisition. I think it'd be really fun to be able to acquire from 20 different head stages at once really trivially. That to me is really useful for example, social experiments. We want to have a better agent framework. So agents are just like what we call sort of like the different behaviors of a Raspberry Pi versus a GPU versus a computer in the swarm. So we want to do multi-timescale experiments where you have some observation in the home cage or something like that over the whole animal's life, but then are also able to integrate that with the day-to-day task that they do. Want to move to a task library so that we don't all have to rewrite the basis of a true alternative forced choice task every time and can build off of each other's expertise. Like someone else has a really beautiful graduated reward structure. I want to be able to borrow that. We want to work on our hardware library. Currently, we have this beautiful under construction web page, but what the idea is to be able to, because we're not prescribing any hardware components, as people make different components.
We need to, I apologize, but we need to wrap up.
Oh, gotcha. So then the final thing is we just need to make the code better. And then thank you everyone that was here. And these folks for helping me out. But I won't take any more time, thanks.
Okay, thank you so much, Donnie. I think it's a wonderful tool, and I'm sure a lot of behavioral neuroscientists will want to try it out, considering especially the increasing complexity of behavioral experiments. And so I'll encourage you to reach out to Johnny if you have any questions or you want to try it out. Unfortunately, we don't have any time for questions, but Johnny, you can quickly check the Q&A box. Oh, I think it was already removed. So with that, I actually want to thank all the speakers. Oh, I think we were just removed. Yes. Okay, I want to thank all the speakers again and with us.
Yes, thanks y'all.
Okay, thank you.
Thank you.
Katajun Ganai from York University. And today she's going to be talking about terpsichore in isolation, a study on the correlation between online dancing and telehealth during COVID-19 pandemic on people with Parkinson's disease. And I'm your host, Alex. And if you have questions, put them in the Q&A or on the chat. And without further ado, Katajun.
Yes, hi everybody. I'm Katajun Ganai. I'm from Toronto, Canada, and as Alex said, my title is Terpsichore in Isolation. It's a study on the correlation between online dancing and telehealth during COVID-19 pandemic on people with Parkinson's disease. So who is Terpsichore? Zeus, king of gods in Greek mythology, has 9 daughters, which represent art and science. Terpsichore is the goddess of dance. Her instrument is lyre, and lyre has been used for music therapy and healing since ancient times. The healing properties of David's lyre, as presented in the Old Testament, has opened a new window to me as an artist, and the concept of art serving a function beyond mere enjoyment becomes as magical as the mythology itself. Dancing with Parkinson's expands the perception of music and dance within the studio. So what is Parkinson? Parkinson's disease is a degenerative neurological disorder with no known cure. Based on the statistics of Parkinson's Foundation, more than 10 million people have Parkinson's worldwide. Each year, about 60,000 Americans are diagnosed with the disease, with this number expected to be around 1 million this year in the US alone. In Canada, 100,000 people suffer with PD. PD develops when the nerve cells in the brain responsible for the production of dopamine are reduced to a critical level. The reason for this damage is not yet known, and the only way to decrease the pace of degeneration is to use medication. The bradykinesia appearing simultaneously together with gait problem in PD has led to Foundation of Movement Disorders Society, MDS, in 1985 by David Marston and Stanley Fawn to provide support for the latest research and diagnosis for people with Parkinson's through designing a unified scale rate, UPDRS, for evaluation non-motor and motor function of people with PD, and other standards questionnaires such as PANASEX in order to bring all studies in the same direction. So among all the researches that have been done, Dance for PD is a program which has been, oh, I can't find my mouse here, which has been running in the world for 10 years. Research has shown positive effects of dance on PD. In order to get familiar with what was going on in the live classes, I will describe the classes at NBS Canada. The format of the dance classes was an hour class once a week with a certified dance instructor and a pianist. Each class had a number of volunteers who would watch for the dancers and assist them whenever needed. The elements of the dance class were a combination of jazz, step, ballet, Argentinian tango, dance theater, and freestyle, and a choreographed movement. Based on Mark Murray's technique, the class had a routine in which half of the class was seated and half was standing. Closing ceremony always was standing in a big circle, holding hands and bowing to each other, saying thank you. And there was a special thank you to the piano player from everybody. Each part would be performed with a specific beat, and the music would be matched with the narrative that the choreographer would improvise. The piano player would either improvise or play a theme arranged to fit the storyline. The instructor would ask him to increase or decrease the tempo based on the mood or capability of the dancers in each session. And the pianist would continue playing as long as the instructor continued the narration. Dancing at NBS or Trinity was more a theatrical dance, which involved free imagination of the dancers at the same time. And participants in Dance with PD Canada have built a community during all these years. The testing process which our team was doing consisted of one fMRI at the beginning of the period of the research and one at the end of each period of research. It also consisted of 2 parts each time, filling out the forms of PANASEX for evaluation of the mood and UPDRS test for evaluation of motor control, working memory, attention, and EEG scanning before dancing. The second part was the same test immediately after dancing. The participants had to take a more detailed form to home and fill that out during the week and would bring it back to the class the week after. With the announcement of a pandemic situation, studio classes for Dance with Parkinson's all over Canada was officially closed. The people with PD are considered as one of the most vulnerable population of the society in the world. Thus, even with opening all other activities in gyms and schools, people with Parkinson's in the world, thus, even with opening all these activities, should take precautions not to contract the virus. Sarah Robichaud announced starting her online class in April 2020. This class has been running since then, and all seniors from all over the country can join every day via Zoom and free of charge. It's all seated with the same routine as live classes while they were seated. The only difference is that the music is recorded and normally with lyrics. As an example, every day 60 people on average will attend in Sarah's class. Since the class is online and everybody can easily access from their homes, elderly without Parkinson's have also the chance to participate. Only a few cameras are on and most of them are off. And the cameras which are on don't give the possibility for the dancers to watch each other as they dance. They have to set it at speaker's view to be able to see the instructor. Many people don't know each other, and although sometimes they talk at the end of the class, they don't have the opportunity to connect to each other. Although we should pay attention that this may be the only social interaction for some people during the day. The instructor, from the other hand, doesn't have a proper connection to the dancers to feel their mood and choose the songs based on that. Time is very limited, and she should have already chosen the order of songs, which can sometimes accommodate her own mood on that day. Since the instructor should talk at the same time that music is played, the quality of sound is not as good as it should be. The same decision was done for going online in other countries such as the United States, Malta, England, Brazil, Germany, Italy, France, Australia, and South Africa. Dance for PD organization based on the Mark Morris Foundation in New York has 26 affiliates in different cities throughout the United States who are running online classes. Although each class may have its own content, but almost all of them are free of charge. And Dance for PD organization, founded by Brooklyn Parkinson's Group in collaboration with Mark Morris Dance Group in 2001, has 300 communities all around the world. Some classes in the world never went online, such as in Germany and France. Some became online, but the demand for it dropped in time, such as Australia. Online dance classes are held differently in each country, from once a week to every day in 20 minutes to 1 hour length. With all these differences, it's been 6 months that 30 people with Parkinson's on average are attending the classes of Sarah Robichaud every day. So since the effects of dance on the brain, motor function, cognition, and mood of people with PD have been studied, the focus of this study is to compare the effects of music on dance with PD. With the need of the new world for being more virtual, live music will not be accessible anymore. The studying on the effects of the recorded music on dance with PD can help the administrators to plan and design better for the future of the online dance programs. Terpsichore shall dance. So this study's primary focus is to compare the post-pandemic virtual dance courses with the pre-pandemic live classes with respect to two essential aspects of the classes that have been influenced by the pandemic: the presence of live music and social gatherings. And we have two variables: a dependent, which is the mood of the participants measured by PANASEX, and an independent variable, the role of music measured by the hours of participation per week. There are two research questions. The first one is, what is the role of music in virtual dancing classes in helping people with PD's well-being and their quality of life? And my hypothesis is that recorded music has an advantage to live music for people with PD in their transition from live classes to the virtual classes. And the second question can be, does the number of hours that one participates in Dance with PD's program virtual classes correlate with the participant's mood? And my hypothesis is that participating in Dance with PD virtually is going to positively correlate with participants' mood. In order to study this, we have two groups of individuals: the ones who were dancing before in live classes and the ones who are dancing now online classes. And we are sending out online surveys to the participants in the virtual classes. And our data analysis is going to be a correlation study on patients' moods obtained through PANASEX before and after class and hours of participation during the week. We will create a matched cohort designed to compare the influence of participation in live versus virtual classes. We will measure the influence of music on the correlations through analyzing the questions from COVID-19 music survey that we have designed. And we have contacted already 28 locations with online dance for PD. We have received responses from 14 locations, and Qualtrics links are being filled by different participants throughout the world. There are, of course, limitations in our study. There is not enough data from the same individuals who continued with the Institute's dancing classes after the pandemic. And there is a limitation regarding the language barriers for collecting international data. And there are future directions for this study, which can be a longitudinal study of online dancing and the progression of diseases in PD patients. That's it. And special thank you to my head lab, Dr. Joseph D'Souza, and Dr. Michael Coglan as my supervisor, music department. Thank you.
All right, so we have a few minutes for questions. If anyone wants to ask questions in the Q&A right now, would be a great time. And thank you, Katajin, for a great talk. So we have the first question on the chat. Is what type of dancing do the participants engage in?
So it's almost unified in all countries. Mark Morris group has designed this dance. I had the details of it. So it's part seated, part standing, and in different countries, they use different kinds of dance, like they use elements of ballet, jazz, Argentinian tango, and they start with warming up the body, first seated, and then little by little, they stand up and do pliés, ballet movements, and they move all the parts of the body. Actually, I talked to India last night. They have also these online classes. They use Bollywood music and they use Indian elements of dance. So it's different in each culture, but the purpose is to move every part of the body possible for these people.
All right, that is great. I have a question. So, have you been able to measure on your assays the mood of participants after the online classes?
Yes, so now the Qualtrics link that we have sent to them includes the PANASEX questionnaire for mood. So they have to fill it out before they start the class. And then immediately after the class, they have to fill the same form again, and we compare it together to measure how the mood has changed after dancing.
What are the results so far?
I'm still collecting data, but before COVID-19, because we have all those data, the results were very positive.
Fantastic. So we have another question in the audience from the chat. How is the therapeutic value in comparison with other more traditional measures?
So there have been many other studies out there comparing dance with exercise, comparing dance with Tai Chi, comparing dance with ballet. And dance has lots of promising results. Dance has been so far worked better on people with Parkinson's.
All right, well, thank you, Katajin. If there are no more questions, or we can leave it, we can allow more questions, but if you can stop sharing your screen so Eva can start setting up.
Okay, thank you.
All right, so we're switching gears a little bit and talking more about data analysis here. Eva Limberopoulos is coming to us from University College London, and today she's going to be talking about using topological data analysis to investigate the relationship between population gut microbiome and Parkinson's disease. So thank you, Eva, and go ahead.
Thanks, Alex, and thanks everyone for coming. In case you're wondering, this is my contribution to the Halloween costume contest. I'm Rowena Ravenclaw, if you're wondering. So yeah, let's get started. Does this work? Okay, so Parkinson's disease is, as you probably know, a debilitating degenerative movement disorder marked by tremor, rigidity, and bradykinesia, causing great disability for all patients. Recent research demonstrates that it is the fastest growing neurological disorder, estimating that patient numbers will more than double within the next 20 years. There is thus an urgent unmet need for disease-modifying treatment, and there are many avenues to this. Some research focuses on preventing alpha-synuclein aggregation or resolving the existing Lewy bodies. Others focus on mitochondrial dysfunction or oxidative stress. However, these avenues have not been very fruitful so far, and treatment is still limited to symptomatic through dopaminergic drugs or deep brain stimulation, or other avenues like the dance interventions we just heard about. The microbiome is an emerging candidate for treatment development and has received a lot of attention over the past couple of years for a variety of topics. It is an attractive candidate because it is highly accessible and any potential treatment would be easily scalable to a large number of people. Moreover, it introduces the option of preventative treatment using the preclinical window of up to over 20 years. And indeed, many animal studies and increasingly human studies as well find support for the role of the microbiome in Parkinson's. It is hypothesized that patients have a more pro-inflammatory profile of gut bacteria, which can damage the epithelium, which is the lining of the gut, which can then cause pro-inflammatory microbiome products to leak out of the gut, such as LPS, and then cause local inflammation, triggering an immune and inflammation cascade. The pro-inflammatory bacteria also encourage alpha-synuclein aggregation locally in the gut. The gut is connected to the brain via the vagal nerve, and that enables bidirectional communication between the gut and the brain, but it also enables the spread of alpha-synuclein into the brain, where it then spreads according to the Braak stages into the cortex, as well as spread of these pro-inflammatory cytokines and other inflammatory markers, which can then cause inflammation and oxidative stress in the brain through activation of microglia. That's the hypothesis so far, and evidence for this comes from animal studies mostly, and also some human studies. One problem with the literature so far, however, is that while the importance of the gut in Parkinson's is quite robustly established now, there's considerable heterogeneity with regards to which specific taxa contribute to this dysbiosis. That is, for one, due to the sheer scale and complexity of the microbiome, which includes 100 trillion individual microbes and over 1,000 species interacting in nonlinear ways. Most studies are too small, underpowered, and using analysis methods that can only consider linear relationships. And because of this, we wanted to use a population-based approach and combine this with topological data analysis, which can account for non-linear relationships of the data. This is the first study to use these methods in Parkinson's disease, and we hope to find specific associations of putatively beneficial and detrimental microbes associated with Parkinson's disease. This is a quick overview of the method. So we have the population-based data set, which is then fed into the topological data analysis through the mapper algorithm, which then yields a network. And then in the second step, the network enrichment analysis, it's analyzed with the so-called SAFE algorithm, which stands for spatial analysis of functional enrichment, which can then map variables onto this network. I will go into more detail now, don't worry. So the data set is a big open population microbiome data set called Atlas 1006. There's over 1,000 individual samples, and participants had an average age of 45. Crucially, they were all healthy, and we established association with Parkinson's disease through the countries or regions that they belong to. This means that we base indirect association with Parkinson's disease on geographic variation in Parkinson's disease burden. So different countries have different burden of disease, and this measure years lived with disease as well. So based on that, we divide countries into low or high Parkinson's disease burden. It is already established that there's not only a difference in this prevalence or the burden between countries, but there's also considerable variation of the population microbiome between countries and regions. And we think that these variations are connected and that we can therefore look at the microbiome of the whole population to infer something about the population's health. So any bacterial taxa associated with the low burden group would be deemed potentially beneficial or protective, while those associated with the high burden group would be deemed potentially detrimental. This high dimensional data is then represented as a point cloud. As the input for the first algorithm, the topological method analyzes the shape of this data without sacrificing its complexity, and through several steps of filtering, binning, and clustering, it yields the data is represented in a network with nodes and links between the nodes. The network that we get looks like this. Again, the nodes represent groups of samples, so groups of individuals that have overlapping microbiomes, and the links between the nodes represent shared samples between these groups. The network that we get for this, for our data set, has 3 distinct clusters, here and here and here, which means that these represent distinct microbiome signatures. Interestingly, cluster 2 and 3 are relatively more interconnected with each other than either are with cluster 1. So it seems that these are a bit more similar to each other than to cluster 1. We can then, in the next step, we can perform enrichment analysis, where we map a variable onto the network. So looking at the average value for each node, then we permutate over the network, it's ranked, and then we basically get the significance of this mapping, which is then called enrichment. Each node gets a score of enrichment, which gets mapped back onto the network, which yields this kind of network here. We can also see that in the next slide. You can get some heat maps. These are the absolute values of this bacterium across the network, and this is the enrichment. We can see that it's highly enriched in cluster 1. This bacterium is called Caenorhabditis. Just as an example of what that looks like, we performed this for all the different taxa and also ranked them, because you can average these scores of the whole network and get a score that's basically like an effect size for how significantly this variable explains the shape of the network. We can also compare groups in this way. Here we compare the Parkinson's burden groups. They had a very high total enrichment score and were notably the only host metadata which mapped onto these clusters. So you can see that the high Parkinson's group maps quite neatly onto cluster 1, whereas the low Parkinson's group is represented in cluster 2 and 3. This can potentially explain the shape of this network, and indeed when we look at co-enrichment between the most significant taxa with the enrichment of the Parkinson's burdens group, we can see that there is striking overlap. The higher Parkinson's disease burden group is associated with Bacteroidetes, and the low group is associated with Actina and Proteobacteria in cluster 3 and Firmicutes in cluster 2. Interestingly, this maps really well onto the current literature. We find support for the hypothesis that Bacteroidetes seem detrimental because they have high pathogenic potential, which means that if they're really abundant, they can change phenotype and become more pathogenic towards the host. We also find support that Firmicutes are beneficial, specifically because they produce short-chain fatty acids such as butyrate, we found a lot of butyrate producers, which is really interesting. Butyrate stabilizes the gut epithelium, is important for gut health and inflammation, etc. We found it weird that actina and proteobacteria were associated with the low Parkinson's disease group, Parkinson's burden group, because they have characteristics that would make them, in theory, more detrimental, but their high pathogenic potential and the increase in hydrogen sulfide is also connected to butyrate. So butyrate producing bacteria exist less when these are more prevalent in the gut and vice versa. That means that their association with the low Parkinson's disease group would be through an inverse relationship with the protective firmicutes. We conclude that the microbiome is crucial in Parkinson's disease, and it warrants further study, and it's particularly interesting due to its potential for treatment, which could look like dietary interventions, pre, pro, and antibiotics as drugs, as well as fecal microbiota, where healthy fecal matter is transplanted into patients or those in prodromal stages of the disease. So I'm very happy to answer any questions, and feel free to reach out to me via Twitter or email.
Well, thank you, Eva, for a fantastic talk. And let me see if there's any questions at the moment. If not, I have a question. Have you looked at any kind of race effects within the populations of what you consider low and high risk for Parkinson's?
That's really interesting. We haven't looked at that because we don't get, that's not collected as part of the data set, and because it's an open data set, we didn't have any influence on that. We will, we're currently trying to replicate this finding in bigger microbiome data sets, and they spread across more countries, because obviously this was a study in mostly Western countries and European and American countries, and we're looking to look at other continents and then look at that, because the microbiome is definitely associated with obviously with genetic factors, but also with differing diets, etc. So there definitely is potential for that, yeah.
Like a question in the chat, are you interested in collaborations with other groups who study Parkinson's disease?
Yeah, of course. This is a really new kind of area. There's not a lot of people looking at the microbiome in degenerative diseases. Well, there is research, but it's still kind of niche. So we're always really happy to, yeah, collaborate with other people.
I personally found it very striking, the segregation of the low and high clusters. Besides you try to infer what kind of bacteria or gut bacteria were in those subpopulations or clusters. Can you say anything more about the specific diets that contribute to those bacteria?
Yeah, so we do, it's quite complex, because what I showed was basically on the highest hierarchical level, so the phylum level, and then you have families, and then you have species, and then you have strains. So it's like each strain can have a slightly different, each species can have a slightly different function. So it's not very clear, but firmicutes, for example, which we found to be quite beneficial, which other studies have also generally found to be beneficial for gut health. These really like resistant starches and carbohydrates that are found in vegetables. So a lot of root vegetables and like raw onions and stuff are really helpful for that. So there is actual like foods that are really high in prebiotic fibers, which is what it's called. It's basically food for the bacteria.
Awesome. Well, thank you so much, Eva. And like, if you have any questions, there's the information for Eva and her email. And now we're going to be transitioning to Carolina Bears, that is coming to us from York University in Toronto. And she's going to be talking to us about reductions in Parkinson's disease symptoms and alpha rhythm modulation as a result of multi-network learning to music over 3 years. So thank you, Eva, and welcome, Carolina. You can go ahead. We cannot hear you at the moment. Maybe our host can help a little bit with this.
Hey, Carolina, try to unmute yourself. Okay, try now. And we still cannot hear you. Maybe try to check your audio settings on your computer. Unfortunately, we still cannot hear you.
Not yet, Carolina. For our attendees, thank you for your patience while we figure technical glitches.
Still nothing, unfortunately. No, still nothing. Maybe try to close Zoom, set audio settings such that your computer microphone is the main input, and then try to log in into Zoom session again. Because you're unmuted in Zoom, so this is definitely not a Zoom issue.
If there are any additional questions for Eva or Katajun while we wait for the for Carolina to come back, I think it's a good way to spend our time. All right. You're muted right now, Carolina. No.
Now you're unmuted, but we still cannot hear you. I'm so sorry, I have no idea what to do right now.
Yeah, I believe there's an option to switch to phone audio if you want to try that, Carolina. And while we wait, Eva, have you tried, is it planned to actually do this analysis on Parkinson patients?
Right now, we're only focusing on the population scale because especially right now with COVID, collecting stool samples is kind of difficult and we can't really be in the lab with the lockdowns, etc. So, and also I'm not trained for that yet, but I would find it really cool. My lab actually has a fecal microbiota transplant trial, not for Parkinson's, but for motor neuron disease. So we do also work with patients like in the lab. This specific project doesn't do that yet, but in the future, there's definitely potential for that. If this other trial works well, then we might do another one with Parkinson's. I would really love that. That would be really interesting for me.
And for our attendees, at the moment, we're trying to figure out a technical glitch. Carolina Burrs, our speaker, her audio is not working. Oh, but maybe right now it is.
Oh, hello.
Hello, we can hear you.
Okay, great. Now let me just go back to the Zoom meeting and share my screen. Sorry about that. I'm not sure why it's not working. I can hear everyone, but I guess you can't hear me. All right. Are you able to see my screen?
We can, yes.
Okay. Let's get right into it. So thank you, Alex, and sorry everyone for waiting. My name is Carolina, and I'll be discussing Parkinson's disease symptoms and changes to multi-network learning to music over 3 years. And all the data that I'll be discussing comes from Dr. Joseph D'Souza lab at York University. So let's just get right into it. Parkinson's disease is a neurodegenerative disorder, I'm sure as you all know, and it essentially stems from a depletion of dopamine neurons within the basal ganglia. So within healthy basal ganglia, we have this dopamine secretion that innervates the striatum, which is responsible for the direct and indirect pathway, where it increases the direct pathway that's responsible for stimulating motor movement and reduces the indirect pathway, which is responsible for preventing any unwanted movements from occurring. And essentially what happens is you get these voluntary, smooth, controlled motor movements that come out. Whereas in Parkinson's disease, it's quite the opposite. You have these depletion of dopamine neurons in substantia nigra, and there's the switch that happens within the direct and indirect. You have a reduced direct and increased indirect pathway, and this leads to the motor symptoms that are uncontrolled, unwanted motor movements that you see in Parkinson's disease, like bradykinesia, rigidity, resting tremor, and whatnot. You also show, some individuals also show non-motor symptoms that can arise from motor symptoms like depression and positive and negative affect and sleep problems. And there's also research that shows that there's differences in people with Parkinson's disease in alpha brain rhythms, where it's a little bit of contradictory research. Some show that there's increases in alpha rhythms, some show that there's decreases. And then there's also research that says that these alpha rhythms can originate from these deep brain thalamic cortical structures. And so essentially, if this is the case, we would see that there would be some changes in alpha rhythms that we can see in something like EEGs. There's also a lot of research that shows exercise tends to influence these alpha rhythms where, by increasing them after participating in exercise. And one form of exercise that's sort of important in our Parkinson's disease research currently is a form of dance therapy. There's a lot of research that has shown that dance does improve motor symptoms as well as quality of life, but this research looks at like the longitudinal, long-term effects of dance, and particularly at 8 months or 17 months. We wanted to see these effects at a shorter duration at 12 weeks. And so we looked at balance and gait speed. And in fact, we did see that balance improved in people with Parkinson's disease that dance, and also gait speed decreased in individuals that danced as well. So we basically replicated these long-term findings at 12 weeks. So knowing that, we wanted to do the same thing, but we wanted to see the single dance class effects after dancing in a single dance class. And we polled people from Canada's National Ballet with a project called Dancing with Parkinson's Disease. And what we did is we ran pre and post class testing. We added some additional measures to our testing. We did the PANAS, which is a positive and negative affect score, non-motor symptom score, something called a UPDRS, MDS UPDRS, part 3, which looks at motor symptoms. And then we also added this resting state EEG, looking at alpha rhythms precisely, where they did 3 minutes of eyes open, and then they did 3 minutes of eyes closed. Then they danced for 75 minutes, and then we did the same testing after the dance class. And we did this with 17 people with Parkinson's disease and 19 healthy controls. What we showed that there was a significant decreases in negative affect after a single dance class. We showed positive increases in positive affect after a dance class, and there was a significant condition by group interaction, where this was mostly shown in our healthy control sample. Then we looked at our motor movements, and as you can see here, this is our PD or PD dancer group and our healthy controls before and after dance class, and we showed that there was improvements in motor symptoms after the dance class. Greater scores means more motor impairment on the scale. Then we looked at global alpha power, and we showed that overall alpha power increased after the dance class, and another graduate student from our lab, Abby Labkoff, showed that within just our PD cohort before and after dance class, these alpha powers increased. So we were able to show that at 12 weeks, and we were able to show that after a single dance class. Now we wanted to see these effects over time. What happens when these people dance for a longer period of time to these motor, non-motor, and resting state EEG? So we dug into the literature, and we pulled out some studies. One particular study, Yankovic et al., showed that part 3 motor symptom, regular progression of people with Parkinson's disease and their motor symptoms was at about a 1.4 score on that UPDRS annually. They also showed that those who had Parkinson's disease that were greater than 57 years of age had an even greater rapid motor impairment of 2.7 almost annually. And then there was also another Parkinson's study group that showed that within 5 years of being diagnosed with Parkinson's disease, these UPDRS scores increased from 5 to about 9 points annually. So we wanted to see whether our data showed something similar or whether it was different than the research that's out there. So what we did is we pulled 16 individuals with Parkinson's disease that danced from the same Dancing with Parkinson's Disease cohort in Toronto. We matched them to the data set that's available from the Michael J. Fox Foundation as our controls, and we matched them on age and Parkinson's disease severity, gender, and disease duration. And we had a total of 60 dance sessions at about 75 hours of dancing with our cohort, and the methodology was the exact same that has been in our 2 previous studies. Except here we added the other parts of the MDS UPDRS, which parts 1, 2, and 4 that basically add some non-motor aspects of Parkinson's disease to look at as well. And these are our results. So just to quickly go over, all of the data that is shown in blue or purple is our PD dancers. Our PD MJFF controls from the Michael J. Fox Foundation are all shown in solid black. And then down here, we have all of the different sections of the UPDRS shown in solid black for those that would be controls, and in blue that would be our PD dancers. And we added the Yankovic and Kaphadia study that I just showed a few seconds ago. So overall, what we can see here is up top here in the graph A, we plotted all 16 of our PD dancers, and we looked at their progression in the motor symptoms over time, and they're showing that there is no difference of motor progression over time. we compared them to the PD-MJFF controls, and we essentially saw that those individuals who do not dance had a greater motor impairment in comparison to ours that do dance. Down here in graph C, we showed baseline measures on the UPDRS-3 again, so the motor symptoms of our PD dancers, of our 16 PD dancers and our 60 Michael J. Fox controlled, as well as their overall means, and we said that there was a significant difference. So again, essentially showing what's up here in this graph. Down here in graph D, we showed all of the UPDRS sections, and essentially what we're showing is that our cohort, our PD dancers, are showing no symptom progression within almost 4 years of dancing, whereas our PD-MJF controls showed progression in all of the variables except for the last one, which is the non-motor complications. It was the only one that was not significant over time. Just to quickly show this graph, we wanted to expand our findings and do a projected 5-year motor symptom because of that one study that showed, this huge impact within the 5 years of having Parkinson's disease and this motor progression. And you can see our PD dancers are controlled, the Yankovic study and the Parkinson's study group. And over 5 years, you can see that our PD dance cohort is actually not progressing as rapidly as all of the other studies and our controls are in terms of our group. And recently, we just started looking and doing the pre-processing of our resting state EEG, and we have a cohort of 3 here. And essentially, we just started looking at this, but we're looking at the eyes open and eyes closed, but I'm more interested in the eyes closed condition, as you can see more of the alpha rhythm being produced during this stage. And essentially what we want to see is whether there's any changes in this alpha rhythm from the first time that they dance to 3, almost 4 years later after participating in dance. And so our next step would be to pull this data out and run some statistical analysis to see whether there are any alpha changes and what do they mean. Maybe they even correlate to some of our motor changes and improvements that we are seeing. So that's sort of our next step that we are doing. And quickly, our conclusions is that we can see that dance can show these positive effects in both motor and non-motor symptoms of Parkinson's disease, short-term and long-term over time. Like I said, our next steps would be to analyze these, the numbers from this resting state EEG to see whether our data matches this hypothesis that the alpha rhythms originate from these deep thalamic structures that are affected in Parkinson's disease. And this is just an acknowledgement of all the Dancing with Parkinson's group, York University, our lab, Center for Vision Research is part of York, and all of our grants. And that would be all.
Well, thank you, Carolina. And we're running out of time. We're going to get kicked out. So if there's any urgent questions, please ask them right now. Or if not, Carolina, if you can put in your email in the chat so people can reach out to you if they have questions that they were not able to answer at the moment, that'd be great.
Absolutely. Thank you so much.
And the thing is, we have a hard out at 50, so we might get kicked out. So because there are no questions, I'll ask you a quick one. I'm more familiar with like tone exchanges in alpha being related to like attention and things like that. And as in like increased alpha, it means less attention. Why do you think, why in Parkinson's an increase in alpha is a good thing? I guess I'm confused about that.
I'm thinking it's just what the literature shows is that there could be this possibility. It's contradictory in people with Parkinson's disease, that some do show increases in alpha, some show decreases in alpha. And actually, we did see, like in my very first study, we showed that there is these positive and negative changes after participating in a single dance class. And we actually correlated these changes to the alpha rhythm changes that we saw. and there was no correlation. So there was no, show that there was no effective link between alpha changes and our positive and negative affect changes in terms of that. So we're trying to see if maybe these alpha changes have something to do with the motor part of it, because we didn't see that in terms of like our positive and negative affect changes from our very first study.
All right, well, thank you, Carolina, and thank you all for coming. And we'll have to close now, but thank you to all the panelists for great talks.
Perfect, thank you.
Thank you.
Thank you, bye guys.
Thank you.
To anyone, I'm just filling the void and showing off my costume because at NMC today, it's Halloween day and we're actually voting on costumes. So if our other panelists have a costume somewhere flying around and have like a minute between talks or during talks to pick it up, you can still do so and we'll take a screenshot.
And we're good to go.
Okay, with that, we should get started. So welcome everyone to this session about imaging methods and how they relate to the brain. Our backend host is Nicola Jaikai. So he'll make sure that everything runs smoothly and I will be emceeing. And so it's my great honor to introduce our first speaker, Josh Siegle. He is an assistant investigator at the Allen Institute and is actually fairly well known from what I understand. Well, from what I have known him before, as the co-founder of OpenEFIS, which is a very popular package. It's a non-for-profit that is dedicated to the development and distribution of open source tools for neuroscience research. For those of you who don't know it, I really encourage you to check that out. And so today, Josh will talk to us about reconciling functional differences in populations of neurons recorded with 2-photon imaging and electrophysiology. Josh, please take it away.
Okay, thanks a lot for the introduction. So yeah, I'll jump right into it. Yeah, so at the Allen Institute, we are interested in doing experiments on a scale that is larger and more standardized than what can typically be accomplished in academic labs. So to that effect, we have set up these Allen Brain Observatory pipelines, which allow us to do high throughput data collection in a highly standardized way from mice using either 2-photon imaging or electrophysiology. And to the greatest extent possible, we try to keep the steps in the pipeline as similar as possible for the two experimental modalities. So we start with transgenic mice, perform surgery on them to remove a piece of the skull so we can have access to the brain. We use retinotopic mapping to map out the visual areas of the mouse on the dorsal cortical surface. We habituate the mice to the recording rig, and at that point, the two pipelines bifurcate, and some of the mice go to 2-photon imaging experiments, and some go to electrophysiology experiments. And then at the end, we either do tissue site or optical projection tomography to figure out exactly where we were recording from. And so each of these pipelines kind of stands on their own in terms of generating very high quality reusable data that we have made freely available to the neuroscience community. But in this talk, I'm going to discuss our efforts to compare the results of these two pipelines and try and see, like, given that we have these two data sets that are so similar, what happens when we try to directly compare the results that we get from imaging experiments with those that we get from electrophysiology experiments. So we try to keep the rigs as similar as possible. So not shown in these images is the visual stimulus display, which is right in front of the mouse's right eye. But these are on the left is our electrophysiology rig. You can see some of the manipulators for the 6 neuropixels probes that we insert in each experiment. And on the right is rendering of the imaging rig. You can see the objective coming in towards the mouse's brain. And as you can see, from the mouse's perspective, more or less the experiment is basically identical. And so we also show very similar stimuli across the imaging and electrophysiology experiments. So yeah, here you can see we have a little green icon representing the imaging experiments and the gray icon representing the EFIS experiments. For the EFIS, we're doing acute insertion. So we insert the probes, record for about 2.5 hours, and then retract them. And so we can only access each cell that we record on one day. And so we try to cram all of the stimuli that we want to show into a single session. Whereas with imaging, we can go back to the same cells over multiple days. And then we can basically like record each cell's response to a variety of visual stimuli. So we tried to include both naturalistic stimuli as well as artificial stimuli, and be able to characterize the responses to this broad stimulus set. So as you can see, we basically, for the EFIS stimulus set, we basically concatenated 2 of the sessions from the imaging experiments together. And then one difference is that we use a different receptive field mapping stimulus in EFIS. So today I'm just going to talk about the stimuli that were overlapping between those 2 experiments, which include drifting gratings, static gratings, natural scenes, and natural movies. And so we've done each of these experiments across many mice. In the EFIS experiments, we try to target all 5 of these cortical visual areas simultaneously using separate neuropixels probes. In OFIS, or the imaging experiments, we have to target 1 at a time, because these experiments are not using some of the larger field of view microscopes that are now available. These are using kind of standard field of view, target 1 area at a time experiments. So we've done hundreds of these experiments and built up a broad database of activity from all different layers of these different cortical visual areas. In the EVIS experiments, of course, we also have data from thalamus and hippocampus and kind of all the regions that the probe passes through after it passes through the cortical visual areas. But I'm not going to be discussing that today. So just looking at the data that is directly comparable between the imaging and electrophysiology data sets. So some of the key questions we want to try to answer, I guess the most important one is how might scientific conclusions differ when recording with one modality or the other? We're really interested in like how do the different biases of these methods potentially change our understanding of the brain when we use one or the other. And I think this is a really important question that the field hasn't really addressed in detail. And our focus here is going to be on visual tuning properties. We're recording in visual cortex, so that makes sense. And kind of looking at the distribution of these properties across populations of neurons and seeing how those distributions shift depending on which modality we use. So there are 3 specific types of properties that we're going to focus on. One is responsiveness. Just a really general question is, what fraction of neurons in an area respond to a stimulus? Like what fraction of neurons are responding to drifting gratings versus static gratings? How selective are these neurons for individual stimulus categories? Do they respond broadly to all different categories of stimuli, or do they have very specific responses to, say, a specific orientation? And then what types of stimuli are preferred by different brain regions? Do we see the same kind of distributions of temporal frequency tuning across different brain areas? And so if there are differences, we want to know how we can reconcile them. And some of the things that we considered, which I won't have time to talk about all of these today, but the things we've looked at. So whether or not GCAMP6 expression alone has an effect on tuning properties by recording in mice that express GCAMP using electrophysiology. Look for potential behavioral differences that could influence our results, and look at the effects of measuring calcium versus spikes. We think this is really important. You kind of read out different information from the cells, whether you're looking at whether you're imaging their internal calcium concentration or looking at their spiking outputs. And finally, there's a well-known sampling bias in electrophysiology, which we think will need to be accounted for. So there are spatial temporal differences. Obviously, in electrophysiology, we're recording along a line, and in imaging, we're recording in a plane. Also, if you look kind of at a high level at populations of cells recorded in the same area, there's a very striking difference between imaging and electrophysiology. In the plot on the right, you can see 100 cells from primary visual cortex showing kind of a heat map of their calcium responses. compared to 100 cells from visual cortex recorded electrophysiologically, showing their spiking patterns. And these look a lot different, primarily because we have much higher temporal resolution in electrophysiology. So the tuning properties that we're looking at, as I said before, we're looking at kind of tuning preference, which stimulus category evokes the highest response, whether the cells are responsive at all to different stimulus types, and also how selective those responses are. And so at a high level, we see similar stimulus preferences for imaging in electrophysiology. So kind of the distribution of preferred temporal frequencies, preferred spatial frequencies are really similar. So that's reassuring. We do see that in ePhys, cells tend to appear more responsive. So across all the areas and all the different stimuli that we tested, we get more responsive neurons in ePhys than in imaging. And I'll talk a bit later why we think that might be. And then in imaging or OPhys, neurons appear more selective. So here we're looking at lifetime sparseness, which is a measure of selectivity. For the drifting gratings, you can see that these distributions are shifted far to the right for imaging and far to the left for electrophysiology. So the reason why we think that cells appear more selective in imaging is because of this transformation from spiking activity to calcium. And so in order to look at this in our data, we simulated this transformation using a forward model called ML spike. And so we can take the spike train as an input and then generate these simulated delta F over F traces and then extract these L0 events, which are what we actually analyze in the imaging data, and see how our metrics change. And so here you can see in this example for these different directions of a drifting grating. In the original data, you can see what the original spikes looked like, and then after the forward model, when it's transformed into these calcium events, you can see an increasing sparsification. So the preferred stimulus, the response to the preferred stimulus is much higher relatively than the response to the non-preferred stimulus. And we see this consistently across cells. And so what you get is that once you apply this forward model, you see a more selectivity in ePhys. However, you don't see decreased responsiveness, which is something that we actually predicted originally. So the forward model does not change responsiveness, which made us believe that actually responsiveness differences are due to the sampling bias of ePhys. So you're recording both higher firing rate cells in ePhys, which tend to be more responsive, and you're also sometimes incorrectly merging units in electrophysiology, which leads to kind of contamination of responses and the appearance of higher responsiveness. So I urge everyone to check out the preprint that we've released on this. This is just a summary figure showing kind of all the different manipulations that we tried and what happens to the comparison between imaging and electrophysiology before and after the forward model. In conclusion, so we do think that scientific conclusions can differ between the two modalities. ePhys makes neurons appear more responsive. OPhys makes neurons appear more selective. Preferences are largely the same. I'm not going to go into all of the different manipulations that we tried, but basically we think that the biggest differences are due to this transformation of spikes into calcium responses, which we can simulate with the forward model, as well as the sampling bias in electrophysiology. So yeah, again, check out the bioRxiv preprint. I want to thank Peter and Saskia, who really put in a nice effort to move this project forward. And also we'll mention that all of this data is publicly available. So learn more on our documentation site. And if you have any questions, check out the Allen Community Forum.
Yay for open science. Thank you so much, Josh.
No problem.
Okay, are there any questions? Yeah, Peter, do you want to ask your question?
Yes, I have a question about what would be your prediction about, so you mentioned that the sampling, the ePhys are biased toward predicting higher firing rates or responsiveness to rates.
Yeah.
What would be your predictions for other brain regions like in the hippocampus, where the neurons are much more dense than in the cortex?
I think in regions with denser cells, you're probably going to have even more cells that are incorrectly merged together after spike sorting. And so you're going to have to be careful and kind of set more rigorous thresholds on what you call a single unit. I think this is one thing that we're really trying to push is for rigorous methods of performing quality control on electrophysiology data, rather than just kind of saying qualitatively whether you think something is a well-isolated single unit, to actually apply more rigorous metrics to determine what the probability of contamination is. And so I think in places where you have more dense cells, you're more likely to get some contamination and potentially see even more bias in the EFIS data.
Yeah, and there's somewhat a related question, I guess, in the Q&A by Jeremy, who asks, is more activity in EFIS due to more temporal resolution, meaning you miss spikes in calcium imaging because of the poor temporal resolution?
Yeah, so that's definitely one factor, but through our application of the forward model, we tried to simulate that and say, like, if for the exact same cells, if we had imaged them rather than record them electrophysiologically, what would their responses look like? And the fact that we didn't actually see a decrease in responsiveness after applying the forward model suggests that these cells are just inherently more responsive, and it's not just this transformation between spikes and calcium that's accounting for this responsiveness difference.
Cool. There's another question by Domokos. Is it possible, for example, using filtering algorithms for photoelectric artifact or transparent material to obtain co-localized and simultaneous, so same time or same field of view, EFIS or OFIS data to validate your reconciliation?
Yeah, I mean, we would love to do that. It's technically challenging because of the light artifact or the need to get electrodes that don't have this light artifact. And it's also very low yield because the sampling is somewhat orthogonal between the two modalities. So you're only going to get a few cells that actually intersect between your electrode and your imaging plane. But I think, yeah, that would be the best way to do this validation.
Yeah. Cool. We should move on. There's another question in the Q&A. Josh, I would encourage you to take that offline and answer it in writing. Hopefully that'll be okay. There's more than one question actually. So if you can take a second to answer those, that would be awesome. And so with that, we're moving to our next speaker, Peter Ruprecht. Peter is, like many people in the field of computational neuroscience, is a physicist by training. And then he did his PhD in experimental neuroscience and is now a postdoc in the lab of Friedhof Helmchen in Zurich. And so he'll be talking about a deep learning toolbox for noise optimized generalized spike inference from calcium imaging data. Peter, you're muted.
Oh, better now?
Much better.
Okay. Okay. So I think we all agree that calcium imaging is a really powerful method, also because it's visually stunning. However, I think the readout of neural activity is quite indirect, as Josh already mentioned before in his talk, and therefore hard to interpret. So for example, what do these transients really mean? So what does it mean in terms of spiking activity? So many algorithms have been developed, which basically infer the firing rate based on these slow and indirect signals like sweet-to-P from Kaiman, ML spike, as mentioned by Josh, from peeling, MCMC, and so on. It's a huge range of algorithms. However, when I first entered the field, I was really a bit depressed by these algorithms, because in the beginning, when I tried to apply those algorithms on my own data, I was not so confident that I got really usable results. First of all, I didn't know how to tune the parameters of the model. Second, the results seem to be often very arbitrarily scaled. Sometimes I got results like 100 Hertz firing rates when it was obviously not the case. just a bad fitting. And the key question I was asking myself was, how do these algorithms work on my own data? Because these have been designed on 1 or 2 data sets which had been recorded by somebody else. But how do these algorithms translate and transfer their power to my data? And obviously, so can I believe these results? And the way to address this question is to look how these algorithms generalize to new unseen data sets. And this was basically not possible until recently because there were not enough data sets available. So that's basically a key result of my approach, to basically assemble a lot of data sets. Some of them had been acquired already before by the Genie project, by the SpikeFNet project, and also by the Allen Institute. Others I have re-extracted from existing publications, and yet again others I have recorded myself or in collaboration with other people. Together this makes up a data set of database of 27 single data sets with different conditions, totaling more than 300 neurons and many brain regions, many different calcium indicators, spanning a huge variety of conditions. Of course, now I could have used one of the previously mentioned model-based methods, basically fit the parameters of these models. However, I think that's really not the way to go. If you have a huge data set with millions of data points, therefore I used a deep network and basically used a local environment of the calcium trace to predict the spiking probability or the spike rate for this time point using this deep network. This is actually a very simple and small network, so it's so small that I can run it on my laptop without any issues, which is really rare for real-world applications and very instructive if you want to go into deep learning without huge GPUs. So I want to highlight only 2 aspects of this algorithm. First is how it generalizes to different data sets. So basically each row is a model trained on one data set. And the columns indicate how well does this model for data set 2 predict the spiking activity for the data sets which are different from this data set. And as you can clearly see, there's not a lot of structure. And this is surprising because we organized these data sets according to calcium indicators and species. And the fact that we don't see a lot of structure in this predictive matrix indicates that it is not possible to predict from the calcium indicator or from the indicator species, from the species, how well the algorithm will perform on unseen data. So to circumvent this limitation, which is basically simply a fact which we found, is that we basically trained a single deep network on all of the data sets. And we found that this network, which is basically using all information available in all data sets, performs much better than any of the single models. The second, this is an interesting observation. The second aspect I want to highlight is that for population calcium imaging, the delta F over F calcium signals are typically quite noisy. And whereas for the ground truth that is underlying all these data sets is really assumed in single neuron, which is recorded at very high signal to noise ratio and temporal resolution. So, however, if you train the model on these very high signal to noise ratio data, you basically get only crap out of it because the network never has seen these noisy signals and therefore assumes, oh, okay, all of those are signals. However, if you add some noise on top of the ground truth and train the network again on this ground truth, then the predictions are much better. So it's basically very important to noise match basically the ground truth to your target recordings. And For the rest, I will just summarize some key aspects of our algorithm. So first of all, the network is able to retrain itself based on noise levels, which it detects automatically. There are no parameters which have to be tuned because it's a universal model which works for all of the investigated data sets, with some caveats, of course. We found that the performance much better than any of the model-based algorithms which we tune to all of our data sets as well. In addition, the result is properly scaled in terms of spike rates. And of course, the most important aspect is that we have tested the generalization to unseen data. And we have not, which allows us to estimate the performance on unseen data, which are basically data that you and I have recorded by ourselves. And then we can predict how well it will perform on those unseen data sets. Yet another reason why you should give it a try to use Cascade, which is the name of the algorithm, is basically that you don't have to tune parameters and you don't have to install it. So basically, you can just click on the link in a GitHub repository, and you will basically be able to open a Colab notebook and just let it run by clicking on the play buttons here and there. And so it's quite easy to use, I would say. With this, I'd like to thank the people involved, for instance, Rainer Friedrich and Friedrich Hempien, who supervised the project, and then Edwin Hoffman, who contributed to the toolbox, and many other people from Tokyo, Yale, Berkeley, London, and the Allen Institute, who contributed these very rare and important ground truth recordings. Of course, I'd like to also mention some open data sets which I used for this project and my funding sources. Thank you.
Awesome. Thank you so much, Peter. Are there any questions? Please submit your questions. Do any of the other two panelists have a question?
Yeah, I have a question. So if you compare the spike rates that you read out using Cascade from the Allen 2-photon brain observatory, the rates are over an order of magnitude lower than what we get when we record electrophysiologically. And of course, some of this, like I said, is probably due to the sampling bias of EFIS towards higher firing rate cells, but there's still a pretty big discrepancy there. So how do you account for that?
I'm quite confident that the firing rates are quite realistic. That's what I would say. Just to give some context, so I applied this algorithm also on a data set which was used by Josh Siegel in his talk as well. And based on that, I inferred typical firing rates for all of those neurons. And typical firing rates were around 0.1 or 0.2 hertz. And this is based on calibration experiments, which were also recorded in the Allen Institute. So I think it's as good as it can get, because it's even using the same transgenic lines in some cases. So I'm pretty confident that it is quite precise. Or what do you think about this, Josh?
Well, I mean, we almost never record cells with firing rates of 0.1 hertz. It's almost always 0.1 hertz and above, up to 10, 20 hertz in our EFIS experiments. And so somehow either it's missing spikes or the training was biased in some way. Like I'm not really sure how to account for such a large order of magnitude difference in firing rate.
Yes, I think that's an open question. But for the bias that you mentioned, I think we measured this bias based on our ground truth, and I think it was quite small. So there's not an order of magnitude bias, but maybe at most 50%.
Yeah.
Or I think one reason could be that there are many. When I looked at the Allen data set, many neurons seem to be simply completely silent. There was nothing happening at all. I think maybe these are simply not recorded at all in the EFIS data.
Yeah,
it's a lot of sources.
That's the sampling bias that we're trying to quantify.
Great. Are there other questions? I have a question. So did you look at or inside your network to figure out how this how the network manages to do the inference?
Yes, actually, since the network is so small, it was quite easy to do. Basically, and here is the look inside end of the network. So here's basically the input, the delta F of F trace. Here is basically the inferred firing rate. And here you can look at the intermediate layers at the activation. And that's what you see immediately. So it's basically, I think it's interesting because you can visualize the entire network just on the half slide of a PowerPoint. So it's relatively transparent. I think maybe you're thinking more about understanding the receptive fields. And this is something I haven't done systematically, but maybe it's interesting. Would be an interesting idea.
Yeah, I was thinking more. Yeah, you call it receptive field. I would probably call it more like a transfer function or something like that. But or like essentially the relationship that allows you to transition from your input to the spike rate, right? Yes, you should be able to analyze that and get like an approximate closed form or some relationship that's approximate that you can get out of the network.
You mean from end to end or from single events?
Yeah, end to end.
I think it's, of course, it's not a linear function because we tried, if we apply the linear network to the same application, it doesn't work at all, or not by far not as good. I think it's quite tough.
So, but don't you expect to get the generative function that Josh was talking about out of this?
This generative function, which you mentioned, this is from the ML Spike toolbox. It's quite, I would say, it's stereotypic, and it's basically can be optimized by using a mean approach. However, the calcium signals are per spike per neuron are quite variable across neurons. I think to account for this variability, I think it's not enough to consider the mean transfer function. I think it's missing a lot of important aspects of the network. But this could be done, of course, yeah.
Cool. Okay, thank you so much. I think we should move on. Next speaker is Gilles van Wallegem. Gilles has a very interesting background in parasitology from Belgium. And then he did his PhD in host pathogen interactions with Trypanosoma brucei. I have no idea what that is, but that sounds very, very fancy. And apparently they can affect the brain and cause all sorts of mayhem. This is part of why he got interested into neuroscience. And so now he's working on zebrafish sensory processing and is slowly transitioning into the enteric nervous system. And it sounds like he's also on the job market. So if you know of any positions he might fit in, please let him know. Okay, and today he'll be talking about calcium imaging and the curse of negativity. All yours.
Thank you, Gunnar, for the introduction. And yeah, the title is probably appropriate for Halloween. So yes, as he said, I'm going to talk a bit about this so-called curse of negativity, which is a bit of a tongue-in-cheek name and title. So we have a preprint that you can check out if you want to know more of the details. But this, I'll try to keep brief because I'm interested in feedback and what people think of it. So as we've seen with the previous speakers, calcium imaging is amazing. So this is a whole brain maximum intensity projection of a fish to whom we're playing sound. So this one is not the one that we also shared on Twitter, which is the zebrafish responding to hammer time. So we have these beautiful data sets, and as Peter and Josh have talked about, obviously this is quite different from EFIs and from the spikes that we get. And ultimately that's one of the things that we would like to infer and to get. And the thing is that in a previous data set that we published that was spearheaded by Itia Favrbull that you can see here in the top left corner. So what in that case, what she's done is she's basically invented tractor beams for zebrafish. And so she can move the otolith, so these are small stone in the ear of the fish, and she can move them using optical tweezers to generate sound. So that's a paper that's going to be published soon. But in the paper in which I helped her analyze the data, it was to simulate vestibular stimulation, so movement and acceleration. And in that data set, that's the first time that while we were doing the analysis, what we identified were basically positive and negative responses. So if you see here on the right, you'll see that the trace of the neuron that's outlined in green, you see the classical positive transients of calcium that Josh and Peter have shown, which of course, when the spike rate increases, then you get these calcium transient that represent the spikes. However, what we also saw, and that was located in very specific parts of the brain, so in purple there and in the red outline, that's the cerebellum of the zebrafish. And so in the dorsal part where we think it's probably Purkinje cells, but we are not 100% sure, we see those negative deviations from the baseline. And these were very consistent. We have many neurons that do it across multiple fish. It's not a movement artifact, so we made sure of all of these things. But these were negative deviations. So there are a couple of issues that come with that, is that a lot of the tools that have been developed to actually analyze calcium imaging data have this assumption that those calcium transients are always positive, so that you'll get these spikes of these increases in fluorescence, and these are where the focus is. And for example, if you look in the middle row at the right, you see that's the delta F over F, which is the classical way that these data are processed, where you have a moving window that goes along and you see the deviation from the moving average. Well, if you do that and there is a negative deviation, depending on the size of the window, if you use a classical window as recommended in some of the papers, you actually create positive artifacts, as you can see with the arrows. So you generate false increases in the calcium. So that's why we now use only z-scoring of the fluorescence, because that does not create those weird artifacts because of the moving average. There are other ways that you can deal with it, but that's the first point that I want to talk about, is the way that you actually normalize your data can generate those artifacts or hide those negative transients that we've observed. So this is all in real data that was published in the paper that's at the bottom. So that's the first thing with this curse of negativity. The second thing that we've also seen is that there are many people in our field that use non-negative assumptions in the way that the data is actually analyzed. So for example, here in the paper, again, we identified 3 clusters of responses. I won't go into the details. I'm happy to answer questions about them, but you can also check the paper. But basically, we observed 2 classes of positive responses, which were responding to different powers of the trap of the optical tweezers. But we also observed this inhibited cluster of responses. And this one we could only identify using a k-means approach to the clustering. Obviously, if you use a non-negative matrix factorization approach, which has been used by other groups to cluster the responses, you would wipe those responses completely. There are other approaches where people binarize their data depending on a threshold of response, where if you need to reach a certain delta F over F to be counted as active, obviously these would also wipe all of the negative responses. So that's the second part of this, which is that some approaches for data analysis will basically hide those responses. And finally, to try to a bit quantify those things and those issues, we turn to a simulation of basically responses. And so we used Naomi, that's been developed by Adam Charles, which is a simulation toolbox that allows you to generate fluorescent movie based on simulated spikes. And so you generate the spikes, and then you get the predicted neural activity with a GCamp kernel, and then from that you can generate the movies. And what I've done is I've then taken those movies and then I've put them through the classical, most cited approaches to extract the fluorescence of calcium imaging data. So Cayman from, oh God, I'm blanking on where it's from, Flatiron Institute, sorry, CellSort, which is a PCA-IC approach, and Sweet2P from Carlson and Janelia. And so we've taken those ideal movies and we've put them through all of these approaches. And again, I can discuss the details of how we've done it, but the advantage is that we know all of the parameters of our data. So we don't have to like fine tune and other parameters of all of these models. And we can extract the responses. We can look at the neurons. So it looks fine. They detect the neurons easily. However, if you look at the correlation between the fluorescent traces, so the GCAM signal, and the ideal responses over time, what you see is that in green on the left, you see that all of these tools do a very good job at finding all of these activated neurons. However, if you look at the inhibited ones, so on the right with the purple rectangle, you see that, for example, Cayman really struggles to find the inhibited neurons. CellSort and Sweet2P deal with it fine. And so if you look in fraction of ideal responses, you see that Cayman, CellSort, and Sweet2P find most of the neurons. And for the inhibited neurons, though, you see that Cayman really struggles again. So this is the third point, which is depending on the algorithm that you use, you may miss more or less of those inhibited neurons. And finally, spike inference. So I won't have to explain to you this because Josh and Peter have done so. And to the right, you see the quantification that we've done. Basically, on the activated neurons, most of the tools do fairly well, except CellSort, but it's a fairly old algorithm. Cascade with the universal model does very well on this, better than the other ones. However, if you look at the inhibited neurons, you see that the performance of Cascade, for example, collapses. Sweet2P does not do as well. Cayman apparently with the FOOPC implementation does fairly well. And that's something that we discussed with Peter quite a bit. And I'm thinking of training some models with the inhibited neurons, but we're still ironing that out with Peter. And a couple of problems when we try to make a universal model that includes the inhibited neurons. So, in summary, what my recommendations are, and we can discuss that, is that Z-scoring apparently generates way less artifacts than the DF1F with a moving baseline. That if you use a non-negative assumption in the way you analyze your data, such as NMF or binarizing your data, you may miss an entire class of responses, which is what would have happened to us if we had done so. Sweet2P seems to be the most robust amongst all of these for finding the fluorescent traces, and FOOPC for the spike inference gave the best results. And yeah, so I want to thank the lab. We're going to hire a lot of people next year. So if you're on the job market for neuroscience or computational neuroscience, keep an eye on the Scott Lab at the University of Queensland. It's very sunny here. It's very nice. And so, yeah, that's it. And I welcome all questions.
Great. Thank you so much, Jill. Questions? Do any of the other panelists have a question? Yeah, Peter.
Yes, I'm wondering, because you mentioned that you think that you have been recording from Purkinje cells. Have you? Are there any?
Yeah.
Are there? Sorry.
Go ahead. Sorry, finish your question.
Sorry. Are there any EFS recordings from Purkinje cells, or are there any calcium recordings in mice from Purkinje cells?
I don't know about mice. We did try to record from Purkinje cell with EFS. However, it was not successful, so we didn't manage to both have the electrophysiology recordings and the calcium fluorescence working perfectly. So that's something that we kind of dropped because we're not an EFS lab, and developing the whole thing was a bit tricky. We would be happy to verify that these are Purkinje cells, but it's just one of our hypotheses.
Cool. We have a question in the Q&A by Priscilla. who asks, which GCamp did you use? Do you think picking a different GCamp would change which algorithm works better?
So to answer the first question, for the real data that was done all with GCAM6S, for Naomi, the model uses GCAM6F. I don't think it would change anything because the issue is basically those negative deviations happen when you have a tonic firing of a cell that basically is inhibited during a stimulus, and that's something that Steinmetz has observed in his dataset with NeuroPixel. 20% of the neurons were actually inhibited in response to the stimulus. And so as long as the rising and decay time of your GCamp is higher than time in between each spike, you will have that kind of baseline high level of fluorescence that then is going to be dropped if the firing rate goes to 0 or close to 0.
Cool. Another question by Jeremy. So you'd need two models used sequentially, one for positive variations and one for negative, then merge the two. Downside is it would take more than twice the compute time.
So that's probably the ideal thing if you want to get the. It really depends what you're looking at, right? If you're talking about the fluorescence extraction, I think SWIT2P works fairly decently for both inhibited and positive neurons. If you're talking about spike inference, I think that's not that expensive because you could identify the negative deviations with many like computationally trivial tools to identify neurons that have these negative deviations. And then you could just pass these into, like, for example, a train cascade on an inhibited neurons dataset and use that for your spike inference, or use FOOPSI just for that subset if you're really worried. So I think it wouldn't be that computationally intense.
Cool. Other questions from the panelists? Yeah, Peter.
Just to confirm, did I get this right? So neurons are either have negative deflections or positive deflections, but not both.
I suspect they could be both. It's like I don't, from the top of my head, know of any example of neurons that have a tonic firing in the like 1 Hertz range that you can see in vestibular neurons, for example, vestibular responsive neurons. That would then change to a sparser firing rate. But I'm not sure that doesn't exist somewhere in the brain. And if you have something like, if you imagine that during a different arousal, you could have neurons that would have a high tonic rate, and then at a lower arousal state, they could have a lower high rate. So neurons could have a mix of both of these, yeah.
Great. Jill, there's a comment for you in the chat by Priscilla. Just don't miss that before we end the webinar. We have a couple minutes left, so I might use that to ask a question myself. So how common are these inhibited neurons, and does that change depending on where you look in the brain?
So in short, so the best quantification that I would say, and it comes with all the bias of course, this is analyzed data, right? So you ditch a lot of things. But Steinmetz in his paper with all the NeuroPixel data that he generated, there was about, I think, 20% of the cells that were inhibited. So that's a decent chunk of the responsive neurons. In our vestibular responses, it was a fraction, but I cannot remember exactly, but it's I think around 10% or something. So it's not like it's not insignificant percentage of the neurons.
Cool. Okay, that ends this session. Thank you so much to all three of you, Josh, Peter, and Jill, for the great presentations. I learned a lot here. This is a bit outside of my field, as you might have noticed for the questions. I hope that the audience enjoyed this as well. Thank you for attending. Thank you for asking great questions to our panelists. And I hope you enjoy the last few hours of the conference. as well. Thank you for attending. Thank you for asking great questions too.
All right, we are live on YouTube. We'll start in 2 minutes. Okay.
I'm back.
Cool.
With tea.
Wow, fancy cup.
I like the mug.
Yeah.
How is everyone enjoying the conference?
Cool, very exhausting, but yeah, I think I can't find a word to describe it, but very intense, but very social. I like the atmosphere. Like it's not intense in a stressful way. It's intense in a fun, social way. I like it.
Yeah, that's good.
Mm-hmm.
I feel like that's like the best of it, you know. I really like the atmosphere of NeuroMatch.
Mm-hmm.
I was a back end for Stephen actually back on Tuesday.
Yeah.
Yeah, I was back end for 5 hours Monday, Tuesday, and
cool.
Yeah, it was and I really liked it. I liked being like helping and contributing. I hope that the next NeuroMatch, everyone, like the volunteers, feel like, oh, I want to help more, you know, for the next conference.
Yeah, the pairing has worked out really nicely too.
Mm-hmm.
Do back end and front end stuff separately.
Yeah, I think that the back end, front end is very smooth.
Mm-hmm. Great. So shall we start?
Yeah.
Great.
Have you started the webinar yet?
No, I'll start now. Okay. All right, then have fun. See you.
All right, I'll give it just a second or two to see if some people come in. All right, well, I'll start with the intros then. So welcome everyone to session 30-22, room 3. My name's Steve, and I'll be running this session along with Arash on the back end. Today we have 3 fantastic talks, really at the intersection of neuroscience and AI, which should be really exciting. It is a traditional talk, so each presenter will have 12 minutes to present, 3 minutes for questions after. And you can ask questions in the Q&A, and the most upvoted questions will be asked first, so keep that in mind. So without any further ado, our first speaker is Haroon. He's a research scientist at the Nathan Klein Institute for Psychiatric Research in the lab of Sam Nemoitin. His talk is on using reinforcement learning to train biophysically detailed models of visuomotor cortex to play Atari games. So whenever you're ready, Haroon.
Yeah, sure. Thank you so much for your introduction. Okay. Okay, so you can see the slides?
Yes.
Okay, perfect. Hi, everyone. So the title of my talk is using reinforcement learning to train biophysically detailed models of visual motor cortex to play Atari games. Okay. So one of the major challenges in neuroscience is understanding neural mechanisms of complex behaviors like decision-making playing games, hunting, fighting, or there could be a lot of other such kind of behaviors. Most of these behaviors are learned over different time durations and require many repetitions of the actions, decisions for given scenarios. But if we try to break down these behaviors, we could think about that we need some modalities for the sensory information processing. That is to understand the situation. Then we would like to have motor actions that would cover how to respond in a particular situation. And then for both these things, we need some motivation or goals, why we need to learn any particular behavior. And that would be required for survival or winning, or there could be any other motivation. So right now, if we look at the neuroscience, we are at the very exciting time that system neuroscience is growing rapidly with the advent of calcium imaging and large-scale data recording techniques. However, we still are unable to actually dissect the full circuits of these kind of complex behaviors. On the other end, if we talk about the computational neuroscience, we really depend on experimental neuroscientists to provide us with the data that we can use to constrain our models. Otherwise, we have enormous space to explore in our models. So at the same time, we, for over a couple of years, we see that these artificial neural networks and deep learning techniques and algorithms, they are becoming very popular among the community. It's very helpful to understand the large-scale data recorded from the system neuroscientists. However, I want to remind that it not always meet the purpose of computational neuroscientists, where we are actually interested in understanding the mechanisms giving rise, basically mechanisms under the behavior. So our goal and motivation, if we talk about this framework, is that we want to achieve the performance of artificial neural networks and deep reinforcement learning. So if we just briefly look at the state-of-the-art deep reinforcement learning system, which was brought up by, I think it's DeepMind. So what is missing here? Why can't we use this system to understand the mechanisms of the behavior production in the brain? First of all, neurons are very simple. The circuits are simple. It's mostly feed-forward architecture. There is no recurrent connectivity. Feedback connectivity is missing. And then learning is more mathematical in terms of that they use back propagation and stochastic gradient descent. So they actually compute the error, and then based on that error, the weights and the whole connectivity is evolved to produce a particular behavior. So if we take one step ahead, then we have these kind of models where actually they train the models and then they try to use the learned connectivity with the spiking neurons, and then you can actually perform the behavior. However, still the use of these kind of hybrid training models is also limited. It can only tell us very limited about how the behavior is computed in the brain. So another paper which came out recently, so they talk about a couple of issues with the deep learning, a couple of difficulties with the deep learning models. So I would not say that we are actually far behind these deep learning models. We need much more computation if we want to achieve that level of success. However, the other problem is, as we mentioned before, that they are based on very mathematical descriptions and not help us understand the biological mechanisms behind that. So our motivation for this project is that we want to use more realistic microcircuits and neuron to provide insights into biological dynamics and their contribution to the function. And we are hoping that this will allow us to use more biological models in engineering. And then we have this platform of the games that can provide us a control setting to study multi-scale circuits dynamics and their role in learning and behavior. So if we look at the framework that we are going to use for this project, so it's basically we have two elements in this. One is actor, which would be our model, which will be basically linked with the environment. And in this case, we are linking it with the game environment of a Pong. And then it's basically the model will be receiving the visual input from the environment, as well as the reward signal or the punishment signal from the environment. And based on that, it will learn how to produce a right action, which would lead the model to score more and more points and win the game. So then I'm going to briefly talk about our first stage of the project, because this is not exactly what we would like to see the model as. But to begin with, what we have decided is to just represent the different areas in the visual hierarchy as a single layer of neurons. For example, if you see in this architecture that we have a retina neurons, which is basically taking inputs from the screen pixel by pixel, and it's driving those Poisson processes with certain firing rate depending on the intensity of the pixels. And then based on that activity, it excites in feed-forward manner the thalamus. So thalamus, when I call thalamus, it doesn't mean that it has anatomy or function of thalamus. These are mostly acting as a relay neurons, which are just passing information to the visual cortex. So in the visual cortex also, we have decided to represent visual cortex in to have two functions. One is to have a representation of the location, and the other one is the representation of the direction. So we have all these neurons, as you can see here, representing the center one is for the location, and all the other neurons are representing the direction of the motion of the object at that particular location. And then after the visual cortex, we have the motor cortex. Again, here we have a very simplified version of motor area rather, I should say. And then each of those, all of those neurons are basically receiving inputs from the visual cortex. And basically these synaptic connections are posed to the reinforcement learning method that we have applied in this paradigm. So if we look at the reinforcement learning paradigm that we are using, this is nothing new. This was proposed by Izekevich, and many groups have used this framework to teach their models to perform several behaviors. So we are using the similar kind of network, where if a presynaptic neuron and a postsynaptic neuron fires together, then actually they produce an eligibility trace, which is kind of a dopamine signal. And if during that eligibility trace, if a reward or punishment happens, then the synaptic strength will change based on if it will increase if there is a reward, and it will decrease if there is a punishment. So then we have a lot of image processing. So brain is basically doing all these things intrinsically while applying different circuits. But we have, again, because it's the start, we cannot capture everything. So we are using different image processing methods to compute the object motion direction. And then these motion directions are basically used to drive the direction neurons that you saw in the previous slide. So similarly, we divided the tasks or the reward into different regimes, and we decided to take on extra type of rewards. For example, it's not only about the winning or losing, but actually the player also needs to learn how to move in the right direction of the ball. So what we are doing here is, this is one example of the intermediate reward, that we are actually computing the projected location of where the ball would land. And then based on that, we are actually using, if the racket moves in the direction of the projected ball location, then it will receive a reward. Otherwise, it will receive a punishment. So similarly, Similarly, we have another reward, which is basically if the racket hits the ball, in game, you don't get a point for that, but in real life, that you need, a player must learn how to hit the ball before it can start achieving the games. So these are additional rewards signals that we implemented in the model. So then there could be different trade-offs for using different reward paradigms. For example, the reward punishment only for score point, lose point, infrequent reward signal mean training takes longer, however, less instrumentation required, and the model may discover novel strategies. Combination of the two approaches could yield better performance, and that's what we learned from our modeling also. So now, once we have a setup for our model, what we do is that before we start training our model, we have to set the initial parameters. And we set the initial parameters in a way so that the firing rate of these neurons is within some physiological range. Of course, here you will see some very large numbers, which are in the motor neurons, but because they are learning over time, so they are becoming quite active. However, that's how we actually control the firing rate. So on the left side, you can see the spiking activity of some neurons, and on right, you see all the roster of activity of the whole network.
Hey, Arun?
Yeah.
Don't mean to cut you off too much, but we're at 12 minutes and we want to leave a little bit of time for questions. So if you're able to wrap up in the next minute or so, that'd be great.
Oh, okay. Yeah, sure. Okay. So here is basically let me move through the basic results. But here is basically what this gives us ability to look at the activity of the whole network at different levels. So we are going to use at later stage to see and compare this kind of activity patterns to electrophysiological data. However, if we talk about the performance that we achieved in playing game, so you can see here that we follow the ball, that the racket learn how to follow the ball so that it can hit the ball. There actually we see quite learning over repetition of the games. However, we see that the score and miss, which requires quite a more complex behavior than simply following the ball or hit the ball, there we could not achieve more than this one. So this is an important point because I want to why we could not go beyond this one. So this is another thing that we learned from the model, that look at the activity, the weight matrices that are basically driving the motor cortex, producing up behavior and down behavior. So yeah, so the potential challenges and work in progress, that we need to learn spatial associations between pixels objects. Neurons activated by spatially close pixels objects may not converge onto higher cortical areas. Instead, neurons activated by distal pixels objects may converge onto higher cortical areas, forming associations. We are coming to this conclusion because we use this kind of convergence in our model. However, we saw that we cannot increase the performance over 0.4 using this kind of hierarchy. So we are changing our model so that we can have more different type of associations in the model. Then the other issue is credit assignment problem, which many people are recently looking into this problem, that there are different reward signals which have association to very different time point actions. So we also need to take care of that. The other issue that people are working on is forgetting. And for that, we are desensitizing synapses once have formed association with a particular action. And then we also need homeostatic synaptic plasticity. So these things we are currently implementing, and hopefully we will have results soon to present here. So the conclusions are that we can use these kind of detailed models, spiking neural network models in the framework of reinforcement learning. Then these kind of rewards are effective in teaching. And we are hoping that our models will eventually provide potential alternatives to deep learning models. And once we extend our model to incorporate more biological details, such model could be used for diagnostic purposes. That's our real aim, that we want to go there in that direction. So I just want to acknowledge the funding resources. Thank you.
Awesome. Thank you, Haran. That was really interesting. Unfortunately, I won't be able to ask you any questions. I'll put them in the chat and you can answer them. But I think we should move on to our second speaker, which is Elena Jones. And she is a PhD student from the University of Pennsylvania in Conrad Cording's lab. And she'll be talking about if single neurons can solve MNIST.
Hey, thank you so much, Stephen. I'll share my screen. All right. Can everyone see that? All right, cool. So my name is Elena Jones. I'm a PhD candidate in Professor Conrad Cording's lab, and my talk will be about, can single neurons solve MNIST, the computational power of biological dendritic trees. Oop. Since the neuron doctrine, we've asked, what can a neuron do? Do is a rather vague term, but we can define it. We can define it in terms of role. So what do neurons do to mediate behaviors? If you remove the neuron and the behavior stops, does that mean that that neuron is a neuron of that behavior, and so on? We also could define do in terms of computational capability, wherein we would consider a neuron as an input-output device, where the input is received in its dendritic tree, integrated into the soma, and producing a binary, or at least how I'll be talking about it, a binary output of the presence or absence of an action potential. I'm not the first to approach a neuron as an input-output device. In 1943, McCulloch and Pitts made the McCulloch and Pitts neuron, which was a linear weighted sum of the neuron's inputs put through a thresholding nonlinearity, producing a binary output. And if you hooked a bunch of those together, you could do logic functions. And if you fast forward about 70 years, you get you reach a very key artificial neural network, ImageNet, by Khrushchevsky et al. 2012, where they were able to hook up a whole bunch of neurons, thousands of neurons, and these neurons, which are actually quite familiar, they're linear weighted sums of their inputs put through some form of output nonlinearity. And if you hook them all up, you can do a high dimensional image classification, where you're able to identify the objects within the images that are present in these high dimensional, high resolution pictures. Both McCulloch and Pitts neurons and artificial neural network neurons are linear weighted sums of inputs mapped through an output nonlinearity. This follows the point neuron assumption, wherein that assumes the dendrites are linear. However, dendrites are nonlinear. They can do logical operations, coincidence detection, low-pass filtering, and other things. Point neuron models actually might be grossly underestimating what neurons could do. So how do we ask what neurons can do? A traditional way is the empirical neuron modeling procedure. It's a very common procedure, very typical, where you would take an input current that you know, and you input that current to a real neuron and record its empirical output. And then you take that same input current that you know, put it into a mathematical model, neuron model of your choice, and then observe what the output is. And what you want is for the output to match the original empirical recording. What you do in order to make it match is you do some form of optimization or training of the neuron model. And when that neuron model, given some input, produces an output that matches the empirical recording, you have a good neuron model. And this can be used to test counterfactuals and other conditions that you may not have tested in the original neuron, and so on. It's very useful. However, the other method of how do we ask what neurons can do that I'm proposing is led by this question. Like with artificial neural networks, can we test a neuron's ability to perform tasks? In this task-based neuron modeling procedure, I put them side by side. It's the input and output that are different. And I will say clearly now that the task that I have in mind is a binary classification task. Binary, and because neurons, as far as I'm considering them, produce a binary output. And I also have in mind for my neuron model that I'm going to ask questions about the dendrites. And to be able to ask questions about the dendrites, I need to have higher dimensional, multidimensional input. So I'd have this dendritic task input, which I'll define soon. And then the output is a correct binary classification. And the reasons why I do this is to take a principled approach as to setting what the dendritic inputs are, and so that I can quantify the neuron model's performance via an accuracy measure, either it is correct or incorrect. So what did I mean by binary classification? I turned to machine learning benchmark tasks. The first of which I will talk about is MNIST, which is a handwritten number data set, pictures of numbers that are handwritten that are 0 through 9, so there are 10 classes. I have 2 classes here that I choose for my binary classification task, and I've chosen them by taking a very simple linear classifier and seeing which ones are the least linearly separable. And another way of saying this is which 2 classes are the most confused when you put them side by side. And 3 and 5 were the most confused pair. So I have I have an input and output function. The input being an image of a number that is, when padded, a 1024 dimensional, each pixel counts as a dimension, image. And the output, which is the target label, 3 and 5, or what I'll reassign them to is 0 and 1. So what about my neuron model? I have my input, I have my output. What about my neuron model? And what I suggest for my neuron model is the one tree. And I'll make an aside here. Choosing your neuron model basically allows you to ask particular kinds of questions and hypotheses. I directly choose this neuron model in order to ask questions about a property of dendritic morphology. And that property I'll talk about now. So if you look at this cartoon of a neuron, I have these inputs, 1, 2, 3, 4, assumed to be only on the terminal ends of the dendritic tree. You'll see that in order for those inputs to reach the soma to be integrated, it needs to travel through this very strict binary tree structure. And so I wanted to ask, what about, does this binary tree structure have some sort of impact on computation? So I turned to deep neural networks, and I made a network that has this dendritic sparsity constraint and called it a one tree. And it's comprised of nonlinear nodes representing compartments of a dendritic tree. And so I want to compare the performance of this one tree on my binary classification task to a linear classifier that is a proxy for a linear point neuron model, and a densely connected, very much the opposite of a sparsely connected, but a densely connected, fully connected neural network that has the same number of parameters. So it's the same exact, well, not exact, but the same size of the one tree. So this is a very busy slide, but I'll direct you to A. And so what we have is the one tree, we have accuracy on the y-axis, and we have the one tree, the linear classifier, and the fully connected network. And so the one tree performs better than the linear classifier. The linear classifier can be kind of seen as a lower bound, but it performs worse than the densely connected, the densely connected network, or the fully connected network. And what you might conclude from this is that perhaps the sparsely connected nature of a neuron might limit the computations that a neuron is capable of compared to that of a fully connected network, theoretically. So this is all on deep networks. So of course, I'll have a slide talking about the limitations and assumptions of this later on, but this is the conclusion that I'll make. Oh, and we repeated this in other data sets, and most of which repeated what we found. And I'm not going to go too much further because I want to get on with time. So what else could impact single neuron computation? Neurons have thousands of synapses, and presynaptic axons synapse on average 4 times per postsynaptic neuron. This means that if you have a postsynaptic neuron, on average, you'll have 4 repeated inputs from the same presynaptic neuron onto that postsynaptic neuron. In vitro multisynaptic batons onto the same dendrite increases approximately 6-fold after LTP induction protocol. What this basically means, or the takeaway from this, is that learning may impact the replication of synapses. And so this leads us to the hypothesis: repeated synapses may also impact single neuron computation. So we try to test this hypothesis using the one tree. So if you look at this cartoon of a neuron here, I have these terminal inputs, 1, 2, 3, 4 again. And what if I repeated those inputs to other subtrees of this dendritic tree? That leads me to this structure here, the K tree, where you have these subtrees with independent weights receiving the same exact inputs across the whole model. So in this way, I'm repeating the number of inputs, and I'm also increasing the size of the tree. So how do I deal with the fact that I'm increasing the size of the tree in my apparatus? I try to modify the fully connected network to match the K-tree parameter size. So using these expressions where n is equal to the number of inputs to either the K-tree or the fully connected network, and h is the number of hidden layer nodes, and k is equal to the number of subtrees. If h is equal to 2k, then the amount of parameters is approximately equal. So another busy slide, I'll direct you to 8 again. So the y-axis is accuracy. The x-axis is the value k, so 1, 2, 4, 8, 16, and 32 determines the size of each of the respective models. And here I have, no, I lost my mouse. Here I have labeled the linear discriminant analysis, or the linear classifier, as a point of comparison. And you can see that in gray, you have the fully connected network, again, above the K-tree at the one tree level, and stays consistent throughout. But you can see that the gap between the fully connected network and the K-tree seems to narrow as you increase K. And actually, if you look at fashion MNIST, eMNIST, in the USPS data set, by the time you get to the K equals 32, that gap, it becomes statistically indistinguishable. They cannot be statistically significantly different from each other using t-test. And this is not necessarily the case for some other data sets. And CFAR10 was weird, so the fully connected network performance actually went down, making it so that the K-tree performance was significantly higher. But that is very odd, and I'm not sure how to explain that. So discussion and takeaways. This is the limitations and assumptions slide. So the input-output function, or a 1D vectorized image classification task, puts our neuron model out of brain context. It's not likely that you'll find this kind of task in the brain. Also, our model is an abstract model of a real neuron. The neurons are not feed-forward. Synapse location is not limited to the terminals of the dendritic tree, and our model is receiving real valued inputs, not binary inputs. But knowing all of these things that are biological properties of a neuron model that one might want are actually next steps in what we can do to make this model even closer to a neuron and be able to test each of these properties. We can test recurrence, we can test synapse location, we can test binary inputs. But for now, the takeaway is that our results provide inputs on dendritic tree structured constraints and repeated inputs. This is a special case of sparse neural network. This sparse network can perform at the level of a dense network using nearly the same amount of free parameters. And future work would be to compare key tree sparse network to other kinds of sparse networks, such as that in the lottery ticket hypothesis paper. This is also a relatively new approach. Considering bringing deep learning approaches toward neuron modeling, we judge neuron model performance instead of traditionally trying to match neuron physiology data. Also, I will acknowledge that some of the work coming out of Aydin Segev's lab also is along these lines, and I would definitely recommend checking them out. Our results suggest that repeated inputs to a dendritic tree could improve its ability to perform complex computations, and some future work would be to introduce more biological constraints and train these more biophysically realistic neuron models to do image classification. And with that whirlwind presentation, I thank everyone here. I thank the Cording Lab. I thank my advisor, Conrad Cording, the Neuroscience Graduate Group, and the University of Pennsylvania. Are there any questions?
Awesome. This is really interesting. We do have some questions. Let me get to them. So Daniel asks, to what extent are the electrophysiological processes at the branchings you model here observed to be plastic in the brain, not just the synapses?
Yeah, so I'm sorry, I'm like speedily running through these slides. I know people hate that. So the branchings are allowed to be plastic. I trained this model using a very biologically implausible process, the backpropagation algorithm. I do not use any neuroplasticity rules that have found in biology. That would be another way of trying to get this closer to what is biophysically realistic. Think of my work as kind of a first step at building this kind of, reference of, oh, this definitely has some impact on computation. That maybe not. This does, and so on. Sorry.
Awesome. All right. I think now we're gonna move on to our next talk.
Thank you so much.
Thanks again, Alana. So for our final speaker, we have Jorgen, and he is a postdoctoral associate at MIT in the lab of Michael Fee. And he'll be talking to us about a connectomic substrate of credit assignment in basal ganglia reinforcement learning.
Thank you so much for this introduction. So let's start the presentation. This works, right? So yeah, I mean, here in the background, you can basically see already that I'll talk about a lot of data. So about a basal or several basal ganglia connectomes, and I basically want to see what we can learn from this like static data about credit assignment in reinforcement learning. So let me first talk about what I mean with credit assignment. Well, this is actually a very general problem, as you know. So basically, when you want to optimize the behavior of a system, then you have to find the relevant parameters that need to be changed, and you have to understand how to adjust these parameters. And a very successful solution for this problem is, as we all know, the error backpropagation algorithm, which is already pretty old, but incredibly successful with its many variants. But the big question for backpropagation is basically, is it actually implemented in the brain? And as you know, there's a lot of recent work, especially theoretical work in this regard. But I think we still don't have like an idea whether an animal actually uses this approach. But there is a second class of algorithms, and I call them here the perturbation-based approaches, which are usually used in combination with reinforcement learning, and they're like even simpler conceptually. So you just like wiggle one of the weights or parameters, and then you basically look at whether the output changes in a positive or negative way. And the big issue with this class of algorithms, on the other hand, is that they're assumed to basically be too slow for learning, so that convergence for real learning problems is too slow. So I want to understand and study how the basal ganglia, which according at least to Kenji Doya is like the reinforcement learning machinery of vertebrates, solve this problem. And I think we actually have now reasonable evidence, especially in the songbird system that I like to study, that perturbation-based approaches are used for song learning. So when you look at how zebra finches learn their songs, so young zebra finches, they basically start out with very simple and unstructured song that we call subsong, and then over time they gradually refine this, so the song becomes more structured and more repetitive, because zebra finches have a very stereotyped and repetitive song, and then eventually you end up at a very stereotyped and but refined adult song. And what machinery do they use for this? Well, I talked already about the basal ganglia, and it turns out that in fact they have circuitry in their brain that involves the basal ganglia that is required and necessary to perform these this imitation learning here. And we have essentially here in the basal ganglia these medium spiny neurons, so they're filled up with medium spiny neurons, and then we have two different cortical areas that are projecting onto these medium spiny neurons, the area that is called HVC and another area that is called LMAN. And then we have a third non-cortical area that is called VTA that also projects there. So how do we think that credit assignment actually works there in the basal ganglia? And Michael Fee, my advisor, came already together with Jesse Goldberg already a few years ago up with a very interesting model in this regard, so that basically credit assignment combines action, copy, context, and reward signals. And when you look for these three signals in the songbird brain, well, then you can essentially find areas that almost perfectly match to these descriptions. So it turns out that HVC has these very statically relative to the behavior firing neurons, so they can convey the relative song time. And then it turns out that LMAN, on the other hand, this other cortical area just talked about, has more or less random activity when you watch the neurons fire during the behavior. And these random variations are also introducing little changes into the song. And this is, of course, exactly what we want for reinforcement learning, right? You want to have a behavior where you introduce small changes, and then you want to evaluate these changes and see whether they were actually positive for the animal or whether they were negative. And it turns out that there's this third projection that in just a few years ago, and Jesse Goldberg's lab, Vikram Gadagar, showed in a very nice paper that VTA is basically conveying a reward, an almost classical reward prediction error into the basal ganglia area here, onto the medium spiny neurons. Okay, so now with these three signals together, the question is, and you rightfully may ask at this point, what does all of this have to do with the connectome and anatomy? And it's actually fairly straightforward. So we derived three key anatomical hypotheses based from this model. And the first one states that in this model, it makes a lot of sense that the HVC or context synapses are actually the synapses that are being modified and identified with this credit assignment mechanism. Why is that? Well, because they're static relative to the behavior and the activity. So if you want to learn something, you want to base it on a stable context, but it's impossible to base learning on something that just varies and fluctuates randomly. So we predict that essentially the HVC context synapses target the plasticity compartments of the medium spine neurons, the spines, but that the LMAN synapses, which convey this information about the random change that just happened to the dendritic shaft. There's a second prediction that comes out of this, which is that pre-post, so synapse pairs between the same axon and the same dendrite from HVC are correlated in size, which can be taken as an anatomical marker of synaptic plasticity, because when you think about Hebbian plasticity, then it basically predicts that neurons that synapses that are similar in their pre-post activity should be similar in size. And the third prediction that we came up with, which is a more recent development, is actually that the basal ganglia should filter out the random activations that are coming in from these LMAN axons. And why is that? because the behavior should be biased by the context of a certain situation, but not by the random perturbations that are currently going on. Okay, so to address these questions, I've acquired two massive electron microscopy data sets of this brain area of area X, and had really help from hundreds of students in this analysis, because this is like such a time-consuming process to analyze these data. But since this was still not sufficient, even with all this help, I was fortunate enough that I could team up with Google AI, so with the team of Laura and Jane there, and they developed these amazing flood-filling neural networks that were then used in my data for automated neuron reconstruction. And I was also teaming up with two undergrads back then in the lab to basically solve the other segmentation and reconstruction problems to be able to then automatically analyze this data, which is essentially the identification of the synaptic junctions, but also synaptic vesicles. And then you want to classify all the compartments of these neurons automatically. You don't want to do this by hand. And of course, the cell types. Okay, so with all of this, I could then essentially access these connectomes and basically query them for these hypotheses that I've just laid out. So I can do these virtual experiments. And just to give you an overview of how much about how much data, like actual measured connectivity data I'm talking about. So the small data set I've acquired contains already roughly 400 medium spine neurons and over 250,000 synapses. But the larger data set is actually one of the largest reconstructed EM data sets in the world at the moment. It's not fully proofread, but even without the proofreading, we think it's extremely useful, and it contains now roughly 20 times the number of neurons, so over 4.5 million of synapses. So that's a lot of data to test the predictions. And to do this, I can now basically, so we wrote this little Python interface, and I can just here is like 2 lines of code that give you an example. So give me all the HBC synapses in the data set. Essentially when the cell type is HBC, and then I can calculate the mean size, for example, but only for those HBC synapses that actually target the medium spine neurons and where the morphology and the postsynaptic site are dendritic shafts. Okay, so with this tool, I can now directly address these predictions I've talked about earlier. So the test whether HBC terminates on dendritic spines and whether the action, so the context synapses terminate on dendritic spines and the action synapses on dendritic shafts. And it turns out that this is true. So the vast majority of HBC synapses terminated on MSN dendritic spines as predicted by the model, while there's a slight majority for the Elman synapses. And I can then could directly test the next prediction as well, so that synapses between the same axon and the same dendrite are correlated in size, but that this is the case especially for HBC, but not necessarily for Elman. And it turns out that there's a highly significant effect for HBC. It's relatively weak when you compare it, for example, to the same observations in hippocampus, which is interesting, but it's about similar effect size as in that is barrel cortex. And interestingly, this turns out to be not significant for shaft synapses at all. Okay, but now let's get to the third prediction. The third prediction was essentially that there is this feed-forward inhibition. And this is now a second-order connectivity analysis, but of course, it's very easy when you have this connectome available to just like search the connectivity matrix for these motifs. So this is what I did, and you can directly identify these Elman axons that are projecting onto interneurons, onto local interneurons, also onto medium spine neurons, as we know. And then these interneurons project again onto the medium spine neurons. So you get these triplets. And then on these triplets, I can now do quantitative anatomy. And so one of the first findings was, you may be aware of a recent study from Princeton, where they basically showed bimodal synapse size distributions for intracortical neurons, and it turns out that there are also bimodal synapse size distributions in the basal ganglia, but only for some connection types. So this is this is very important because it means that so that maybe something is special about these connections, and we're still like working on understanding what this could be. But I can also use these identified triplet motifs now to perform like more quantitative anatomy, for example, in terms of dendritic location of these synapses and whether they could actually be suitable to cancel out spikes. And this is what we find. So the inhibitory neurons are enriched here close to the soma, while the excitatory synapses are more far out. Okay, and with this, I already come to the last part. So we also did, based on these findings, we also did a small modeling study, like very simple firing rate models, actually with a local learning rule and eligibility trace, as Haran had like introduced in the beginning. And when you implement this model inspired by our anatomical findings, then you see that in a relatively small number of iterations, so less than 1,000, which is well within the number of repetitions that actually zebra finches use to learn their song, that you can copy a simple one-dimensional template. So with this, I'm already at the conclusions. So I think that EM connectome analysis is really a new, right? And new because it's actually the technology and theory is there since many years, but it was very difficult because of the technical challenges. A tool to test functional circuit models and ideas. And for this specific model here, to me, it looks like the data and simulations are consistent with this plausible, biologically plausible credit assignment mechanism that is based on converging action, context, and reward signals. And if you have, like, if you want to read up on this, then most of these results are already available as a preprint. Thanks.
Awesome.
Yeah.
Thanks so much, Jürgen.
Yeah.
Sorry. So yeah, I know we're running pretty short on time here, but if anyone has like a quick question, I'd be happy to ask it. I thought someone raised their hand, but maybe not. Okay.
Okay.
Yeah, I guess just a quick thing, I guess, like how simplified do you have to be when you do the, when you get these, you know, EM partitions, I guess, of the neurons, and they connect to a bunch of things, bunch of different neurons. How simplified do you have to be in terms of how you interpret the findings after that? I guess, if that makes sense. So I imagine there's a lot more complexity going on in there than what you're extracting.
Yeah, I mean, so the results that you saw, they were extremely simplified and targeted, right? I mean, so when you so I think there are in general two different ways of dealing with this kind of data. So the first one would be that you go in with specific predictions and just like almost exclusively look at what your model essentially says, and the other way is that you take a more explorative approach and just look at like all the different cell types and how they connect. And so in this case, I've taken like this like very focused approach, which can make your life much easier, right? Mm-hmm. Because of the amount of data, and there is some. I mean, there's some. So, I'm not sure I fully understand the simplified aspect of the question, but I mean, the data is like not really simplified, right? So we are measuring synaptic areas for the different cell types, and then look at where they end up on the dendrite. So we have like very detailed anatomy and theory available, but we are not like using all of this anatomy all the time. Yeah, it really depends on the question.
Yeah, I guess it helps when you have a very focused question. Great. All right. Well, thank you
so much,
speakers again. Really interesting talks, and nice to see where things are going. Thanks again for Arash for doing anything on the back end, and thanks everyone for attending.